ನೀವು ನಿಜವಾದ তারা ওরকম আবার
ಪವನ್, ಗುಡ್ ಮಾರ್ನಿಂಗ್. ಅದೇ 12 ಗಂಟೆಗೆ ಡಿಸೈಡ್ ಮಾಡಿದ್ದೀನಾ? ನಂದು ರ್ಯಾಪ್ ಅವ್ರ ಇಲ್ಲ ನಂದು ಮೀಟಿಂಗ್ ಆದ್ಮೇಲೆ ಅಥವಾ 3 o'clock ಆದ್ಮೇಲೆ ನೀನು ಕಾಲ್ ಮಾಡು. ನಾನು ಬೇಕಾದ್ರೆ ಸರ್ ಹತ್ರ ಮಾತಾಡಿರ್ತೀನಿ. ನೀನು ಏನಾದ್ರು ಡೌಟ್ಸ್ ಇದ್ರೆ ಡಿಸ್ಕಸ್ ಮಾಡಿ.
Good morning, sir. Krishna Kumar, sir. Good morning. Hello. Am I audible, sir?
You are audible.
Devpa, sir, can you please unmute? Hello. Am I audible, sir?
Yes, you are audible.
Hello, Celia. How are you? Not able to hear you.
Good morning, sir. How are you? Good to see you.
Good morning, sir. Am I audible, sir? Krishna Kumar, sir?
I think sir is not able to hear.
Okay. Good morning, ma'am.
Good morning. How are you? You are doing
Yes,
a very
ma'am.
good job.
Thank you, ma'am. Thank you so much. Good morning, sir. Krishna Kumar, sir. Am I audible?
Yeah, audible. You are audible.
Yes, sir. Thank you, sir. Happy to see you, sir.
Thank you so much. Shall I have sent my PowerPoint presentations to Devap also, but also I can
Yes.
share from my screen. Okay. So, Devappa Sahibre, namaskar ರೀ.
ನಮಸ್ತೆ.
ಕೆಲಸ ಎಷ್ಟೈತೆ ಅನ್ನೋದ
What you silly?
ಕೆಲಸ ಎಷ್ಟೈತೆ ಅಲ್ಲಿ ಈಗ ಅಷ್ಟು ಜೋಡಿಸೋ ಹಾಗೆ ಅವನು ಇವತ್ತ ಸರ್ ಇವತ್ತ ಸಂಗಮಟ ಮುಗೀತ. ಓಕೆ ಇನ್ನೇನ್ರಿ ಒಬ್ಬೊಬ್ರು ಒಂದೊಂದು ಫಾರ್ಮ್ಯಾಟ್
Good morning, sir. Krishna Kumar, sir. You are muted, sir.
Good morning, sir. Sir, Krishna Kumar, sir, can you please unmute yourself?
How I am audible?
Yes, sir.
Okay.
Yes, sir. Sir,
9:30 sharp start ಆಗುತ್ತಾ ಲೇಟಾ? Will you start at 9:30 or it will be late?
No, sir, we will start sharp at 9:30. Sir.
Okay.
I'll just confirm whether the chairman has arrived. Dr. M.S. Patil, sir, are you there?
Is with Shamrao Jagirdar. Professor M.S. Patil is online.
ಮ್ಯೂಟ್ ಇಟ್ಟಿರ್ತೀನಿ, ಯಾವಾಗ ಸ್ಟಾರ್ಟ್ ಆಗುತ್ತಾ ಹೇಳಿ ಅಷ್ಟೇ.
Hello, sir. So we'll start the program, sir, now. Shall we start?
I am okay with anything.
You can start, Rajeshwari.
Okay, sir, thank you.
Chairman is there.
Chairman is there.
Professor
A very warm good morning to you all. I welcome you all to the fourth day of our International E-Conference on multidisciplinary approaches in plant disease management for sustainability in agriculture. I welcome the chairman, co-chairman, rapporteurs, keynote speakers, invited and oral speakers, and all the participants to the 8th session on virus vector relationships. This session is chaired by Dr. M.S. Patil, co-chaired by Dr. Prabhraj, and the rapporteurs are Ms. A.S. Padmaja and Dr. T.
Rajeshwari muted. Please unmute.
Okay, sir. Am I audible, sir?
Yes, yes.
This session is chaired by Dr. M.S. Patil, co-chaired by Dr. Prabhuraj, and the rapporteurs are A.S. Padmaja and Dr. T.B. Manjunath Reddy. It gives me an immense pleasure to introduce the chairman of this session, Dr. M.S. Patil. He is the professor and head, Department of Plant Pathology, College of Agriculture, Dharwad. He is specialized in plant virology and transgenics. He has guided 7 PhD students and 16 MSc students, and he has almost 100 publications. Sir, I welcome you to the session. With the permission of the chair, I now welcome Dr. N.K. Krishnakumar, who will be presenting the keynote talk on recent invasions of Western flower thrips, Franklinella occidentalis, and tomato spotted witch virus into India and their possible implications to Indian agriculture. I welcome you, sir.
Thank you,
With
madam.
permission of the chair, I would like to introduce Dr. N.K. Krishnakumar, sir, to the session. Dr. N.K. Krishnakumar is Bioversity International's regional representative in South and Central Asia. In this role, he provides scientific leadership for research activities on the management and use of agricultural biodiversity being implemented in Central and South Asia. Within Bioversity International's initiative on effective genetic resources conservation and use, he works with partners and stakeholders at the global, national, and local levels in implementation of collaborative research activities. Before joining Bioversity International, Dr. Krishna Kumar sir was worked for ICR, where he served close to 40 years in a number of capacities, most recently as DDG Horticulture Science. Prior to this, he was Director, National and Principal Scientist at Indian Institute of Horticultural Research and Head Entomology and Nematology. He has contributed significantly in horticulture, pest management strategies, and insect vector studies. He developed DNA barcode for nearly 50 different species of insect pests. Dr. Krishna Kumar serves in a variety of associations in India concerning horticultural ecosystems, entomology, and technical management. He is associated with many national and international professional societies in various capacities and is a founding member of the Entomology Academy of India. Sir, I welcome you to the session. And sir, can you please
Thank you.
unmute? Yes, sir.
Yeah,
I now
thank you.
request the chairman, Dr. M.S. Patil, sir, to take over the session. Thank you. Dr. M.S. Patil, sir, can you
Dr.
please
M.S.
unmute
Patil.
yourself?
Okay, just he is unconnected. He is disconnected.
Okay,
Please, let us start. He will join. No problem.
Sir, I request Krish Kumar, sir, to proceed with his presentation. Thank
Sir,
you.
1 minute, madam. Sir is win.
M.S. Patil, sir.
MS Patil, sir, is there.
Sir, can you please unmute yourself? Okay, so Krishna Kumar, sir, you can proceed with your presentation. Start uploading the slides, sir.
1 minute. These are all things which we have to learn to, because I'm used to other one. Let us see how it is done.
We'll help you, sir.
I have set, you have the slide means you load. Otherwise, I'm going to share here.
Yes, sir.
Then
Please share, sir.
I'll see what I can do. Okay, just hold on for
Yes.
1 second.
Yes, sir.
Yeah, is it visible?
Sir,
Noted.
good morning. Very good morning.
Sir, it is same as the other platforms. You can
But I have set, in my screen it has come.
No, sir,
Now,
you open
Sir.
the PPT first and then share.
okay, just hold on. Share, okay. Is it okay now? Let's see.
Yes, sir.
Okay,
It is starting to share content.
okay.
Yes, sir. It is visible. Kindly
Yeah.
put on to the slideshow mode, sir, and present.
Okay. Okay.
Yes, sir. It is visible. You can
yeah.
proceed, sir. Thank you.
Very good morning to all of you. Dr. Choppa, I see the audience. Dr. Chaudappa, and so many of the esteemed colleagues, the chairman and the co-chair of this session. In keeping with the symposiums and the theme of the symposium, and also the importance of the viral diseases that are emerging in the country, partly because, am I audible? Am I
Yes, sir.
audible?
Yes, sir.
Yes, sir. You are audible. You can proceed,
Okay.
sir.
So I just thought that one of the problems which has just cropped up in the country and which is going to have a drastic implications on the future of viral diseases in the country, I just thought of to flag this issue and sensitize the national and the other audiences of the region to have a kind of a preparedness in tackling this problem, which in no way can be, its damage potential can be underestimated. This is a species of thrips which I worked for my doctoral dissertation in Hawaii as well as my postdoctoral fellowship in UC Davis. And the very name of Western flower thrips sends shivers across the globe because of its ability to transmit innumerable number of diseases, a huge range of host plants, and probably it is exactly similar to Apis gossypii that we are used to in aphids, or probably Anopheles mosquitoes in the human segment. So Western flower thrips is one of the top vectors transmitting almost. In fact, the very tospoviruses that we are talking today started with Western flower thrips. Way back in the 1914, it was reported in 1905 and then confirmed in 1914 in the Australian subcontinent. And it was Mr. Thrips, Mr. Pittman in Australia, who using the Western flower thrips confirmed the transmissibility of the tospoviruses through thrips. And later on, Dr. Sakimurai, a Japanese scientist, worked enormously on this pest and was instrumental in developing the first plant resistant to tomato spotted wilt virus. Originally, tomato spotted wilt virus was considered a monotypic virus. That means only one virus, which is of the tospovirus group. But later on, we have many tospoviruses. And the tospoviruses is a very unique virus in that all its cousins are animal viruses. The so-called many of the hemorrhagic fevers, hantavirus and many others. Somebody is speaking, can they mute their phone? So many of its cousins, there is an echo, many of its cousins are basically the viruses. Is there an echo? You are hearing an echo, or can I go ahead? Hello?
There's an echo, sir. Somebody has muted.
Okay. So I'll do one thing. I'll remove my headphone and speak. That I think may be better. Just give me a minute.
Am I audible?
Yes, sir.
Yes, sir.
Hello.
Yes, please proceed, sir.
Hello?
Yes. Yes, sir, you are audible.
Okay. So many of the species are animal viruses, but this is the only species of tospoviruses. And tospoviruses are one among the many vectors like whiteflies and aphids, which are very, very assuming a lot of importance. And most of the vectors are having a piercing and sucking type of mouthparts like mosquitoes and others, which facilitate the transmission, and thrips is no exception. It was originally called as rasping type of mouthparts, but recent indicates very clearly indicate it has a piercing and sucking type of mouthparts. What are basically thrips for the information of the houses? They are extremely small, 1 to just 2 millimeters, though some species can be large. But you can see that one of the characteristic features of the thrips are they are having fringed wings. Thysanome means fringed, Thysanome means fringed, and terra means wings. So their most characteristic features are the fringed wings, and Western flower thrips is no exception and is the size of about 2 millimeters. One of the unique aspects of the thrips is, though it does not come under the typical group, the nymphal stages are called larva 1 and larva 2. And I'll come to this why I'm explaining to this. There is a pseudopupal stage in the soil and in the leaf litters, and then the adult emerges, and the life cycle is completed in about 20 to 21 days, which is also, and the adult itself can live about 20 days, which is necessary in understanding the epidemiology of the virus. I won't go into the too many details of the taxonomy. There are two subgroups called Terebranchia and Tubulifera. You can see the tube-like structure on the right. That's not what we are interested, but the one on the left is the Terebranchia are more important. Now, many of them actually suggest that any vector that comes into it, you just spray any of the insecticide and manage. Most, I would say that whether it is western flower thrips or any other species of thrips, we should not assume that all thrips are alike, all mouth parts are alike, and their feeding mechanisms are similar. In fact, on the right side, this is one of the actual photograph of the scanning electron micrograph of the western flower thrips mouth parts. You can see that right in the corner there, it is the tip of the mouth part itself is loaded with so many of the sensors, which makes it to choose one plant over the other. You can also see how the compound eyes are organized. Hundreds of eyes, hundreds of the sensilla present on the antenna and mouth parts determine the host specificity, and that is what determines why a species of thrips is vector or not a vector. These are some of the emerging viral diseases associated with thrips. Sunflower bud necrosis is not a tospovirus, but there is a strong association of thrips. Tomato TSWV, originally this is a photograph from other places, but it could also be a tomato tospo. And we are also having thrips palmate transmitted watermelon bud necrosis. We are having, the list is endless, many other places. One of the unique features of thrips that Dr. Sakimura initially in 1940s is thrips are very unique as vectors, and western flower thrips is no exception. The one on the left side, you see the classical mouth parts of the western flower thrips through an scanning electron micrograph, and you can see the complexity of the structure. On the right side, I have specifically drawn the digestive system of the thrips, which is hardly 1 to 2 millimeters. Once the virus is acquired, the virus has to move through the blue structure, which is the gut of the thrips, and then at the barrier, that is epithelial barrier of the hindgut, the viruses actually emerges into the hemolymph, gets back into the salivary glands, where they tremendously multiply, and that is when thrips become systemic in infection. But the beauty of this entire thing, also looking into the previous slide, is only the larvae can acquire the virus in this case, but cannot transmit. The adults can only transmit the virus, but they cannot acquire the virus. And even among the larval stages, the first larval stages are very, very potential in transmission of the disease. So there is a tremendous understanding of the vector that is required in our management strategies, and to assume that whether it is thrips palmi or Certothrips dorsalis or western thrips are all alike in this process may not be appropriate, because that is telling that Culex, Anopheles, and Aedes mosquitoes are all similar in the transmission. So we have to be very careful about understanding a deeper insight into what we are talking. Why study vector transmission? It's a beautiful question, because most of us will only do diagnosis, and when it comes to management, the method advocated are very different. One of the things is, one of the things that I would like to flag the attention is whether vector control is feasible. Even when you want to breed for resistance, you have to go through the process of both insect transmission and in some cases mechanical transmission. For example, we all infect papaya ring spot virus through mechanical transmission, but in nature, nobody is going to smear the infected sap.
Department exam pass பண்ற மாதிரி
யாரோ தமிழ்ல பேசுறாங்க பாருங்க, please switch off. There is a vector and virus. Hello. Dr. Devappa ಯಾರ ತಮಿಳಲ್ಲಿ ಮಾತಾಡ್ತಿದ್ದಾರೆ ಸ್ವಲ್ಪ ನೋಡಿ. Vector and virus transmission both are essential. Now, another thing is, irrespective of what we do when the source inoculum is there, it is only the vectors which spread from one plant to another. Now, how do they spread? Why do they spread? What stages do they spread? What are its alternate hosts? And the virus survives when the plant is not actually there, the host plant, in the vector. So understanding this is very critical in our management issue. When we made this slide, we did not have in Franklinia occidentalis. We had only Franklinia schulzii, and of course, I see on the right side Franklinia occidentalis, Trabasi and thrips palmi. For those of you who want to critically understand how the Western flower thrips entered into this country, someone in the Tamil Nadu, in their anxiety to please somebody, brought chrysanthemum cuttings imported from Western countries. And those Western countries, chrysanthemum is one of the hosts of Western flower thrips. And as a result, the Western flower thrips entered into this country. And then what happened is, the chrysanthemums which were grown in the polyhouse were actually taken to the market, and the Western flower thrips began to spread. And it was only a matter of time. When we import the floriculture material, you can also be sure that many times you are also importing the adult thrips which are fully loaded with virus, and once they come to the environment, they are going to spread the virus. In a matter of time, we also had the tomato spotted wilt virus very close to Tamil Nadu. Many times what is really happening before the onset of molecular systematics is there are hardly few taxonomists. Luckily in India we have excellent taxonomists in the form of Vikas Kumar, Kumuthyagi and others. But many countries don't have this kind, and as a result, some species they wrongly identify. For example, Thrips flavus was attributed as a vector of tospovirus, but later on rectified. In fact, Thrips flavus was identified as this probably because of the mixed infestation that was sent to Dr. P.N. Krishna. Now, so the very critical determination, you must have seen in Delhi billboards, they put whenever dengue is there, now they don't put Anopheles, very critically they put the tiger mosquito. So it is critical even in our case. The management of viruses is extremely complex, whether it is aphid or thrips or any transmission, because unlike the pathogen, environment and the host plant, there is a fourth factor which is the vector, which in turn is used by n number of factors, so your complexity multiplies. Now, the thrips transmission of tospovirus is actually, and Western flower thrips also is no exception, and Western flower thrips, because I have myself studied, it is circulative and propagative. Please underline the word propagative. The movement of the virus inside the body is the same as whitefly, aphids and whiteflies, but the exception is they replicate in the midgut cells of the thrips as well as in the salivary glands. The infected plant, usually one thing that we have to understand is each sucking pest has got its own type of feeding, whereas aphids are basically phloem feeders and they feed through intercellular spaces, whereas thrips actually destroy the epithelial cells and in the process they begin to feed. But a destroyed epithelial cell, automatically the virus also gets destroyed. But anything that acquires and transmits without destroying the epidermal cells is actually the one which is going to transmit. Tospoviruses, for the benefit of those, nowadays everybody is beginning to understand the complexity of viruses with coronavirus. This is also a cell which is about 80 to 110 nanometers in diameter and is a tripartite. That means it has 3 RNAs and each RNA codes for different region, and it is a very complex, and some of my work involved on the blue and red dots like structure you see on the lipoprotein membrane, the glycoproteins which are basically responsible for insect transmission. Now this is just called the GP and the GP2. This GP1 and GP2 are actually also called as the amino glycoprotein and the carboxylate glycoprotein. And it is one polyprotein which is divided into 2 and thereby forms different type of glycoproteins. One of my work involved the suppression of the one glycoprotein, we categorically proved that the transmissibility or acquisition and transmission by the thrips is actually prevented. It was my postdoctoral work in the UC Davis. Now, these are some of the things I don't want to go into too many details, but here I begin to share with you the slides. You see the kind of problems that the Franklinella virus can transmit. At this point of time, I would only request the house to say, in Kadayam, we have one of the biggest floriculture nurseries. All our floriculture nurseries and other nurseries are importing the plant left and right from other countries. I am 100% sure it is only a matter of time before this spreads and we are going to hear new diseases. Now, the biggest question that why I flagged this issue was the Eastern Hemisphere is predominantly dominated by Thrips farming, which is another very important vector, as important as Franklinia accidentalis. Now we have both Franklinia accidentalis and Thrips farming, and we are going to see a dance of plasmoviruses in the days to come unless we take adequate control measures. Just to show the virus transmission as it occurs, that the only larvae can acquire, and then it is transstadium. Stadium means one stage to another stage. It is passed on to the restive stage of Thrips, and viriliferous adults emerge and disperse. It is the dispersing adults which are basically responsible for the transmission of the virus. And our studies very clearly also indicated, based on my postdoctoral work, real-time RT-PCR, and also the virus load was estimated in the Thrips, infective Thrips. We clearly established that the virus adults, as soon as they emerge, the first few days are extremely critical, and later on they begin to gradually lose their load. But even then, it becomes extremely critical in managing the freshly emerging viruses. This is just to show the salivary gland complex is critical to virus passage. I will not go into the details, but we are just telling the infected plant and the non-feeding stage, how it is there in the insect blood, which we call it as hemolymph, and then adult emergence takes place. This was one of our classical works, which we said, often a question is asked in many of our insect vector studies, are all the insect vectors equally capable of transmission of the virus? My work, including the Western flower thrips work which I did in postdoc, in the population there are three different categories. You can see that the top line, which are basically the ones which are what we call as perennial transmitters. On the inoculation axis, you can see the first day, second day, third day, up to 5 days, they are extremely potential in transmitting the virus. But there are also some groups which we call as ephemeral transmitters, which can transmit the disease only for first 1 or 2 days after adult emergence, and after that, they lose their ability to transmit. There are, in addition to that, there is what we call as non-transmitters, which is 50% of the population. So we have to understand, and this population trend may not be equal across from January to December, and it may vary depending on the potential of the species of thrips. So again, this question that I am flagging for these issues is management of viral diseases has a lot of inputs has to come from the vector side to make the management more clear. Further to that, there are a lot of questions. For example, in many countries, there are male thrips and female thrips. And many times they say male thrips are more efficient transmitters, which is still to be proven across. But thrips can multiply in such a way that they can only produce males or only produce females. And in most cases, they are only producing females, as in the case of Thrips tabaci in India, where 99.99% of the ones are females. But males are required in order to create the evolutionary progress. So now we are coming into a stage where both these species are merging in the Indian subcontinent, whether it is groundnut, whether it is watermelon, whether it is capsicum, whether it is tomato, whether it is chrysanthemum, or n number of crops, including impatiens necrotic spot virus, which is also transmitted by thrips. In fact, there is for those of you who are interested, like Siliachalam and Chaudappa, on 24th we are having a meeting arranged by the Entomological Society of India, and where Dr. Ramesh Chand has agreed to preside over the onion problems. The onion, what is that? What is that, Chaudappa, the name sometimes I forget. The virus associated with onion seed production is devastating. IYSV is becoming very severe, and we expect that its implications to be more in the coming Rabi season. And as a result, all the private sectors and the public sector and the policymakers will be there to discuss on 24th. And for those of you who are interested, can go to the Entomological Society of India website, and the link will be provided. This is just to show that the symptoms of TSWV is little different as compared to the symptoms that comes on GB or the tomato tasco on tomato. And our initial studies very clearly indicated that TSWV is more damaging even as compared to tomato tasco on tomato. For many reasons, I do not want to disclose too many details. This transmissibility. So it is also a number of crops are also there. Species are such as claritis and many others are also being reported by Dr. Kumar and others. Only thing is that these are the characteristics associated with the first incidence of the tomato virus. Many of us even do not know how to identify a vector-borne disease like virus and other problems associated with other wilt. Always the wilt associated with a vector-borne disease, you can be sure that the apical regions are wilty and the distribution will be highly sporadic. But even then, one of the close observations is this is the first symptoms that occurs on the infected leaf, and if you are smart enough and destroy that plant to a great extent, you may possibly prevent the secondary spread of the virus. Now, this is just to show the vector status of tomato virus. One of our own studies very clearly proved Pseudothrips dorsalis is also a vector of watermelon bud necrosis. We don't know whether it can also transmit PSWV or more efficiently or less efficiently, but these are some things. For example, what you see is the tomato tasco as compared to the previous slide, when you can see the symptoms of TSWV as compared to tomato tasco. Now, these studies also require a very patient understanding of the acquisition, the multiplication of the species, the indicator plants, as in this case of cowpea. Only C152 is excellent as far as the development of the symptoms, and also the maintenance of thrips under extremely hygienic conditions so that cross contamination doesn't take place. Choosing the right stage of thrips for transmissibility studies, and also knowing how to dissect or how to understand the replication rates of species. These are all some of the processes. I am very happy that the ORP on sucking pest, you were able to make a very significant progress with regard to a number of these issues, which has helped us to place at this level, thanks to Dr. H.P. Singh, that we are in a position to address many of these things much better, without which we would not have been in this position. One of the things that we also carried out in the process is we developed molecular markers for a number of species of thrips, inclusive of western flower thrips. And as I said, for many reasons, certain of the information on western flower thrips I am not in a position to share, but we have wonderful markers for western flower thrips. Of course, it is also available across the world. But what is very clearly different with this thing is, many a times we have to see whether the species that are the thing that we have is the same as what is elsewhere reported. Because in the case of Thrips palmi, we found Indian populations of Thrips palmi were vastly different in a few sequences as compared to the global sequences for Thrips palmi in many other countries. You can see that Indian population you can understand why we are able to transmit in India. Watermelon bud necrosis is a very, very serious problem. But in countries such as Taiwan and China, watermelon silver mottle is actually a serious problem. So, though we are telling western flower thrips and identified, it is possible that there are many biotypes which further needs to be probed deeper and deeper. So, this is about thrips hawaiians is another species of vectors, and these are some of the work how with the specific sequences and how the group is occurs. You can see in the case of Rankiniella occidentalis also, there is a group and there is a group 2. So we have to go deeper in our understanding of molecular basis of the species in order to have our management studies. Having said that, the future line of work should be immediately somebody pick up by sensitizing the authorities that how far it has distributed, which are the host plants that are there for the thrips as well as for TSWV. We have to sensitize the policymakers on the vector status. Floriculture movement both from outside and as well as within the country. You go to the airport, you will always find somebody carrying floriculture material. Host plant resistance itself, has to have a relook, because in case of tomato and many other crops, we have to look for tomato TASPO as well as for TSWB. Releasing one over the other may not really help. And virus plants has to be done. Other viruses that Western flower thrips can also transmit is something that needs to be seriously, and also the replication patterns and population. So I just thought my whole purpose of this presentation was to sensitize the Indian continent about the new invasive species. Thank you very much.
Thank you, sir. Thank you for your overwhelming presentation.
Am I audible?
Yeah. Madam, yeah.
Sir, please end the slide, sir.
I'll share the slides.
Thank you, sir. Thank you.
Madam, this is MS Patil speaking.
Yes,
Yes,
madam.
sir. Please take over the session, sir.
Okay, thank you very much. Good morning. Of course, initially I missed this, but I observed everything. So I thank our keynote address speaker, Dr. Krishna Kumar. He knows, everybody knows him very well, former Deputy Director General of our ICR, and presently he is the Biodiversity International Regional Representative of South and Central Asia, and he has delivered really a very wonderful talk, especially with the tips that vectors. This is the very appropriate time, because these TASPO viruses in India, major viruses in our part, the bud necrosis, which is making a lot of damages on tomato, which is called bud blight disease, and bud necrosis in groundnut, and also watermelon bud necrosis, which is the most important in our US Darwad. We have done a lot of work on these, and also this TASPO is most important in onion, Irish yellow spot, and other important crops, and he has covered very elaborative informations on those things, plus virus vector relations, and he has introduced the new vector to our all the delegates, Frankliniella occidentalis, which has been introduced, and this is making some of the adjustments now. I think the next, if time permits, next we'll go for the invited talk.
So shall we take a few questions, question answers?
Okay, then we'll keep it open so that we'll entertain one or two questions, because there may be limitations of time. If time permits, no problem. So questions from.
Hello, sir.
Yeah.
Yeah, there is a complexity of our thrips. What is the strategy to find out identify the thrips species or what is the biotypes?
Krishna Kumar sir, can you please unmute yourself? Krishna Kumar sir, can you please unmute?
Sir Krishna Kumar. Madam, any problem with the audio of Krishna Kumar?
There.
I am not able to. Am I audible?
Yeah, now you are audible, sir.
See, one of the best answers we have molecular diagnostics, and Dr. Ashokan will be able to help not only identify the thrips, but also if there are any subtypes in the species. Dr. Vikas Kumar also can do from ZSI. We are also training a number of postgraduate students who are equally competent in molecularly, in a very quick time, identify the species, and also if the biotypes are emerging, we'll be in a position to give. But what is required is a financial support to do this kind of work. And unless and until we empower the laboratories to this kind of work, we will not be really able to provide. This is something, but we have the potential to do whatever the types or anything. Thank you. Thank you.
Thank you, sir.
Thank you, sir. So any other questions?
We should also welcome Dr. Chodappa, who is in the audience. Chodappa. Okay. Okay.
Chodappa sir.
Okay, he was there. It's okay.
Patil sir.
Yeah.
So you can go to the next, on the next speaker, sir.
Okay, madam. So the next speaker, it is the invited talk, Dr. Vipin Palan. The topic is role of whitefly and plant proteins in the transmissions of apple scare skin viroid. So Dr. Vipin, please go ahead and we will have the 10 minutes time presentations.
Okay, sir. Thank you very much. Am I audible to all?
Yeah, you are audible. Go ahead.
Okay, I will share my presentation. Is everybody able to see the presentation?
Yes, sir. We are able to see your presentation.
Okay, please share your slides.
It is visible, sir. Slides are visible. You can proceed, sir.
Okay, okay. Thank you very much. Good morning to all. Good morning to all. First of all, I must thank the organizers for giving me this opportunity to share my work, especially Dr. Devappa and Dr. Shankarappa for inviting me for this presentation. So this particular work actually it deals with the role of whitefly and plant proteins in transmission of apple scar skin viroid. So there is a team of students and scientists who are working in this area, and we are working at CSIR Institute in the Institute of Himalayan Bioresource Technology in Himachal Pradesh. So this is where we are located. We are in the Kangra district of Himachal Pradesh, which is at about 1300 meters, and we are very close to the Dhauladhar range of Himalayas, which is covered with snow for the part of the year. Just to give you a brief overview of what actually the institute actually works on, there are many areas which include industrial crops, aromatic plants, medicinal plants, floriculture, nutraceuticals, and many other areas where microbial resources, diversified products we make. And we are specifically also looking into the role of how the plant pathogens, what kind of plant pathogens are present in the crops that we grow in the Himalayas. And I would be talking especially about one of the pathogen, viroid pathogen that infects apple plantations wherever we have the apple cultivation. So this viroid actually is a very, very small pathogen. It's a very unique pathogen with a size of only about 246 to 400 nucleotides. Does not code for any protein. It's single stranded RNA. and it is replicated by the Rhotic circle replication mechanism, and it causes losses mainly through fruit and flower deformation, tree barking, yellowing, and mortality. And whatever the characteristics of the viroid is that it spreads rapidly in the field. Symptoms are mainly seen during the fruit and flower development, and there are some unconfirmed reports which point towards the role of aphid and bumblebees in the role in the process of transmission. And on the right, actually, you can see the structure, how the viroid actually looked like. There are a number of stems and there are a number of loops where the single-stranded RNA, circular single-stranded RNA, is organized in this structural formation. Now, as you can see in the state of Himachal Pradesh, there are many places, many districts where we are actually growing apples, and apple scarskin viroid is a very serious issue, and it reduces the marketability of the fruits and the quality of nutrition as well as the quality of produce for the farmers. And if you really to an extent, it's very difficult. Now, we were actually involved in study of the apple scarskin viroid pathogen, and we were looking into its mechanical transmission into the various hosts, and we were trying to set up a diagnostic host for apple scarskin viroid. And we had prepared this infectious clone, and we were doing some inoculation experiments, and we saw that some of the host plants like cucumber and beans, they developed chlorotic lesions. But some of the unexpected results that we got was that there were some uninoculated plants which were also showing positive similar symptoms, and they were also positive for the viroid. And at that time, there was nothing known in literature as to whether viroids can also be transmitted by some insect vectors. So when we looked at the plants very carefully and observed it for a very long time, we found that there was presence of greenhouse whitefly, which is basically Tramidurus vaporium, and it's not Bemisia. So Bemisia, we don't have Bemisia because it's a subtemperate kind of area, and we mainly have this particular whitefly in our greenhouses. And we saw that this insect was actually associated with the plants. And basically, it's a phloem feeder, as with many other insects. And it is known that the viroid actually is translocated with the help of a plant protein through the plant, through the phloem, which is a phloem protein 2. And we actually tested a number of plants, and we found that there was many plants which were not inoculated, but they were actually positive. So wherever we saw that there was association of the whitefly, there was presence of the viroid. So we hypothesized, which was very unconventional at that time, that whitefly, it can acquire and transmit the viroid RNA. So this is for the first time we were thinking about it. And because viroid was not known to be transmitted by any insects, and people know that it is only through the pruning tools that these viroids could be transmitted. So the number of experiments that we did, we found that the viroid is actually very actively transmitted by the greenhouse whitefly. We prepared the infectious clone and we fed to the fly, and then we inoculated into the plant, and we see that the viroid is getting transmitted and it is developing chlorotic-like symptoms on cucumber, some of the cucumber cultivars. And when you actually transmit directly through the fly, also you get the similar kind of symptoms. So there were a number of questions like mechanism of acquisition of transmission of the viroid by the fly, how this is actually happening, whether the viroid is replicating in the insect vector, other cell types, and whether whitefly proteins, they are associating with the viroid, and what is the fate of the acquired RNAs. And this work was actually published in RNA Biology. We analyzed the insects which were actually feeding on viroid positive plants, and we saw that there were replicative forms of the viroid which were detectable in the fly. And as you know, dealing with white flies, you need a lot of material for analysis, and this experiment was actually repeated several times, and then only we could confirm that there were replicative forms of the viroid. We don't know whether these are coming directly from the plant or they are being replicated inside the vector. So that was yet to be established. So there were virus-specific amplicons, which we obtained from RNA and DNA of the Viroplicaris whitefly, because right now we using this new term because we only say Viroliferous. So because viroid was not known to be transmitted by any insect, so we gave this new term Viroliferous whitefly. And when we analyzed the whitefly RNA and the whitefly DNA, we found that we could actually amplify these specific fragments, PCR fragments for the viroid. In the whitefly DNA also, we could amplify in the infected and the positive. In the healthy, there was no amplification. In the whitefly RNA, there is another 200 base pair fragment which we could amplify. This was not to the viroid, but it was the 330 base pair fragment which was specific to the viroid. And this DNA form that we identified from the whitefly DNA is actually unique to the fly, and it is not present in the infected plants or in the healthy insects. And probably this could be because of the revert transcriptase-like activity which could be present in the insects. We are looking into the characterization of this activity at the moment. So further analysis of this complex, we worked out, we found that it is transmitted as a ribonucleoprotein complex by the whitefly. And this band was actually specific to the viroid. There was a very 200 base pair fragment which was always getting amplified, and we characterized this. We found that it is coming as the ubiquitin ligase gene of Cucumis sativus, which is always associated. And we see that this particular transcript is always associated with the ribonucleoprotein complex, and it was always amplified in the healthy Cucumis sativus also, but not in the whitefly. So there is a complex of viroid and ubiquitin ligase, and there could be many other components which we don't know yet. So the whitefly actually contained the CSPP2 protein complex, the Cucumis sativus floin protein 2 complex. We prepared the antibody for floin protein 2, which because we knew earlier from earlier observations published in literature that this protein is actually translocating the viroid through the phloem. And we prepared the antibody, and then we did Western blotting studies. We isolated the total protein from the whitefly, and on the left panel, actually, you can see that results of the Western blotting analysis where we identified the presence of the PP2 in a number of complexes, right from 1 to 4, which was varying in size at the same time. So 4 potential complexes we could actually identify where the floin protein 2 was also present. And because this protein is always associated with the complex of the viroid and ubiquitin ligase, we feel that there are a number of complexes where the viroid is actually getting associated in the whitefly, and this is happening during the passage of the viroid through the whitefly. So how does actually, this is the most obvious question, like how does the whitefly is actually doing it? We found that the whitefly contains transcript binding proteins, and through Western blotting and through Northwestern blotting analysis, we identified at least 3 places where this complex was existing, and we characterized this through multi-top analysis, and we found that there was a number of proteins which we tentatively identified as heat shock protein 20, cyclophilin, and the other bands were also like for threonine tRNA synthetase and somatomedin B, and we don't know whether it is getting replicated or not. So based on these observations, we actually worked out further. And I must say that these binding experiments, they were done with very high concentration of sodium chloride, so that the binding is not loose binding, it's a very strong binding that we were actually talking about. So what was like one of the obvious experiment that we did was to understand whether we can get the replication of the viroid in the insect cell lines, because working with whitefly, feeding them, and that experiment is we are still doing whether it is replicating in the whitefly or not. But we did some experiments where we transfected the viroid into a number of human and insect cell lines, and we saw that there was change in morphology of the cell lines, and they lose their intrinsic morphology basically, and there were cytopathic effects we saw throughout, and this was actually sign of globularization of the cells and shell shrinkage, and so it was reflecting that the viroid is actually replicating and having its effect on the morphology of the cell lines. And at the same time, the viroid was actually integrating into the RNA, into the chromosomal chromosome of the cell lines, so that it is replicated. This is normally happening in other plants also. So to integrate into the host chromosome up to 48 hours, because after that we did not see any amplification. So we did a number of experiments. The next question was like to understand, are there some proteins, and earlier observations show that there could be potential proteins like small heat shock protein or somatomedin B or some other proteins. So one of the proteins that we took up recently was small heat shock protein, which is about 20 to 22 kilodalton in size, and it is a protein which is very specific to the insects. This protein we could not identify from other organisms, and this was very specific. This, especially the small size of the heat shock protein, was very specific. We wanted to understand whether this protein, in order to validate also, we wanted to understand whether this protein is binding with the viroid, and through this EMSA experiment, as you can see in this photograph, that when we use increasing concentration of the protein, we see that there is a reduction in the mobility of the viroid in this agarose gel, and when you use a very high concentration, that leads to a remarkable reduction in the movement of the viroid and the protein complex. And this protein actually we had expressed in E. coli, and then we used the varying concentrations of the protein. And this actually, the lane 6 and 7, where we are having very high reduction in the mobility of the complex, shows that there is a cooperative binding. So as you add more and more proteins, so more and more complexes are being formed. So this was also confirmed by Northwestern analysis, where we identified two in vitro binding assays of small HSP with a big labeled viroid RNA, and we found that it is clearly binding with the viroid RNA. And one of the controls that we used is protein 2, because it is already known to be binding with the viroid RNA. This was also validated with the help of bioinformatic analysis, where you can see on the left and the right panel that this is the structure of the viroid RNA, and if you look at small HSP, it is able to bind to a number of sites, and one of the site that we identified was on top of the structure of this particular molecule, where the small HSP is binding very strongly. So they have very good affinity with each other, and this can be proved with the help of in vitro as well as in silico experiments. So in order to actually validate and understand whether this protein is really required for the process of transmission, we did transient expression of the truncated small heat shock protein in cucumber, because it is known if we generate the small RNAs against this particular transcript, this particular gene in the insect, in the plant, and then feed the insects on that. So while feeding, they actually take up the small RNAs, and there is a silencing of that particular gene in the insect. So this slide actually shows that we use the tobacco retrovirus vector, and we could see clearly that this particular small HSP is actually expressing in the plant where we actually use this TRV2 vector, and it is transiently expressing. So there was some morphological effect also, as you can see. And when there is a viroid and TRB1 and TRB2 vectors associated with that, there is a reduction in the size of the inoculated plants, while on the healthy or the small HSP, only expressing plant was normal. So we actually also analyzed as to how the silencing is actually happening in the plant. So if you look at this data, it shows actually there is an increase in the silencing. This was done through real-time PCR, where from day 1 to day 7 there is increase in the level of silencing. This is not the expression. This is increase in the level of silencing. And when the whitefly were actually fed on the small HSP plants, they showed normal growth and development, as I told you earlier. And the silencing of the small HSP in whitefly assessed by real-time PCR, the relative expression of small HSP was gradually increasing. And what we found is, like, when we use these whiteflies which are with compromised expression of small HSP, there is a reduction in the transmission of the viroid by the insect, and it was about 40% reduction that we could get. It was not 100%, it was only 50%, and it shows that there are many more factors, many more transcripts or many more other proteins which could be actually responsible, and small heat shock protein is one of the component which could be responsible for the transmission of which is part of the complex coming from the plant to the insect. So there are a number of questions that still remain. What is the replication potential of the viroid in the whitefly? We are still doing this experiment, have not confirmed it yet. And what is the source of the reverse transcriptase activity, whether it is coming from a retrovirus which is present in the insect, or it is coming from the retrotransposons present in the genome of the fly. Now, what is the composition of the complete ribonucleoprotein complex? Because we could get only partial reduction in the transmission. There are some other whitefly proteins for critical for transmission. So, which are those proteins? And those proteins, there was a bit of lacuna initially because we were not having the genome sequence of this particular fly. So, we are also developing transcriptome data as well as the genome data, so that we can specifically characterize and analyze the role of these proteins of Ephorarium whitefly in the transmission process. And what is the fate of other plant transcript? Because the plant phloem is actually full of many other transcripts which are translocated through the phloem. So that is all being sucked by the insect. What is the fate of that particular transcript that is taken up by the fly? So there are a number of questions that we are looking into, because this is altogether a new area which we actually identified in 2015, and we are trying to characterize this particular transmission process and the unique role of the insect we are trying to work out. And this has huge implications, because there are transcripts which are present in the phloem, and the insect may be responsible for transmission of the transcripts from one plant to the other, which may not be happening, which was not known earlier as well. So this is a unique observation, and this could actually lead to the role of insects as a vectors for gene transfer of genes from one plant to the other. So this is a team which worked. It was initially started by Dr. Yashika and Sunny. They were doing their PhD at that time, and new students who took up this transmission work, Savita and Swati. Dr. Anand Raj, who is working as a research associate in our lab. Dr. Rituraj was responsible for the bioinformatic analysis, and most of the cell line work was done in the lab of Dr. Luke. Thank you very much for your attention.
It's shall I go ahead, madam?
Sir, question answers if any, sir.
Yeah, thank you. A fantastic presentation. and it is the beginning of the work showing the viroids can also be transmitted through vectors. So it is requires
Thank you,
a
sir.
lot of
Thank you.
work
And
in your institute or somewhere. The work has to be carried out, especially you have the potato, you can see the potato tuber viroid and others.
yeah,
Conclusion of course requires some more work, but anyway, congratulations. It is a
yes,
very good beginning of so far what we say viroids are not transmitted. It's because of the protein coat which is not there. So
yeah,
the lot of questions, a lot of clarifications and controversies
yeah,
may come later. You please see that you continue the work and make the
yeah.
conclusion and see that you will be remembered in the history of viruses. So with this, I think I encourage one or two very specific questions to Dr. Vipin.
I have only one question to ask. Are there any parallel work going on which show that the transmissibility of genes from one plant to another through the vector? Any other parallel work going on elsewhere?
Of especially of X.
Yeah.
That I am not aware. I can only say that there was some work done earlier where people tried to implicate the role of insects in the spread of viroid, and this was an indirect kind of experiment that they were doing where there were bumblebees, and through the bumblebees the pollens were actually getting spread from one infected plant to the other, and that is how the viroid was shown to be associated. And in some cases, they also found that viroid was actually encapsulated within the uh, protein of the capsid protein of the virus. But in our case, we specifically tested a number of viruses which could be present in the plant, and we found that there was no virus, and it was only the viroid which was actually getting transmitted by the fly. So directly, there is no other work going on, to the best of my knowledge.
Okay, thank you, sir. Patil, sir, can you please unmute yourself?
Will go for the madam. Will go for next speaker.
Yes, sir.
Okay. So the next invited talk is from Dr. Rajkumar E, and the topic is engineering chimeric peptides or proteins as vector management. Dr. Rajkumar, your talk can be for. Please go ahead.
Okay. I can hear me. Hello.
Yes, Dr.
Okay. Okay. Hello. Today I will discuss on recent advances in use of heterologous peptides or proteins for management of insect virus vector. Pardon me?
Dr. Sorry for interruption. Can you please put your slides in slideshow mode?
Okay, thank you.
Thank you. You can proceed now.
Okay. Today I will discuss on recent advances in the use of heterologous peptides or protein for insect. Actually, I am not from this field. I am actually protein chemist and structural biologist. If I make any mistakes. Okay. Following are the shortcomings of the use of chemical pesticides for insect pest management. The vast majority of the chemicals target within the nervous system of the insects. And indeed, chemical insecticide interact with just one of the main targets, voltage-gated ion channels, glutamate receptor, GABA receptor, acetylcholinesterase receptor. As a result, the use of agrochemicals with such a few targets has promoted the evolution of resistance to the vast number of insecticide families, which were highly effective controlling the insect pest in the recent years. In addition, industrial use for the control for the pest management represent less than 1% of the total pesticide market. Then it becomes the secondary consideration for the most agrochemical companies to develop a new generation's insecticide for vector control management. And they also have off-target these insecticide effect on beneficial insects also. Then is there any alternative strategy then can be the natural products as a potential alternative sources for novel in recent years, has been renewed interest in the natural products as a potential source of novel insecticides because many of the most successful second and third generation insecticides are natural products or their derivatives. For an example, ivermectin and spinosad, as we know that these are produced by polyketide synthesis. These are all modular proteins which produces these antibiotic-like compounds. And also we know that delta toxins, also known as bacillus thuringiensis toxins or cryotoxins, which are mostly effective against lepidoptera and insects. And also terpenoids, the lacks of the terpenoids have been discovered. Many of them are having insecticidal effect or insecticide repellents. And we know that pyrethrinides, and I will discuss in the next subsequent slide, spider and scorpion peptides, they as a insect products. And in particular, there is a growing interest in potential of insecticide peptide derived from the venom of spider toxins. Yes, they are rich source of insecticidal peptides since these are all very compact peptides and they have insecticidal properties. And they can modulate several receptors. They target several different type of receptors which cannot be targeted by the chemical insecticides, such as ion channels and also the modulation of neurotransmitters and G protein coupled receptors. They target many important enzymes in the insects, such as chitinases, phospholipases. And also, as you know, that these are environmental friendly, which are alternative to the conventional agrochemicals, since these peptides and proteins can be produced through transgenic crops or plants. Now, I will discuss two case studies where they used toxins or protein toxins or peptides successfully used for management of hemiptera and insects through the transgenic crop productions. At the end of the my talk, I will discuss on our work on engineering chimeric toxins fusing different peptide or protein toxins which through genetical fusing, and these can be purified and can be used for production of transgenic plants. First case study I will discuss on seminal work by Brownie Boning Group from the Department of Entomology, Iowa State University, where these insect neurotoxin peptides, though they offer as alternative to the insecticides, but have drawback. What is the major drawback? These neurotoxins has to be delivered to the hemocells. Then here authors used P. enunciation mosaic virus coat protein to deliver this toxin to the hemocells. These are the control constructs they use it for the productions of the toxins. One is GFE, it's a green fluorescent protein. So this is used for tracking the toxins, and that is fused with the H is histidine tag for the purification purpose. Then this green fluorescent protein is fused with the coat to track the coat protein movement in the insects. Then this one fused with the histidine tag for the purification purpose, and also expressed in the E. coli and purified from the chromatographic technique. Then they standardize several construct. One of the construct by inserting polyprotein peptide between the coat protein and the GFP that will lead to the productions of higher quantity of the toxins, and also this toxins is highly soluble. And then these constructs these construct they tested on the epids, different species of epids through the injections or through the feeding. As you can see that, as you can see from these images, these green fluorescent proteins is localized to the hemocells, especially in the pericardial cells. Only if you fuse it with coat protein, but not alone. That means what is the conclusion from this slide is CP delivers the fused protein peptide from gut to the hemocyl. Okay, upon standardization of the delivery of coat protein to the hemocyls, then authors went and fused these insect toxins. That is insect toxin derived from the spider, that is exotoxins, and they want to test it whether that can be delivered to the hemocyl or not. This is the omega exotoxin from the spider. As you can see, this is a very which has beta sheet structures, and also these strands are linked by the disulfide bond, and it is very stable insecticide. And these are mainly blocks mid and low and high voltage activated calcium channels, and this paralyze the insects. And in addition to this one, they fused with signal peptide, GNA secretory signal, which is signal peptide. What it does, it takes the protein from the insect to the phloem of the plants, so that this insect can be this toxin can be delivered to the plants. Then, in addition to that one, this is the toxin they fused with the core proteins, and also they fused with the GST tag. This is the purpose of GST tag is purification purpose. You can produce in the E. coli and you can purify. And also, as a control, they used inactivated exotoxin. That means they mutated some residues, they generated the variant so that that variant cannot be activated. Then the of these fused proteins on the different epids vector, as you can see that you can see that in nearly 90% of the mortality, in the case of where the core protein is fused with exotoxins, and in the control where is the inactivated exotoxins, that mortality is very less. Conclusion is core protein delivers exotoxin to the hemosyl, and exotoxins highly toxic to the epids if delivered to the hemosyl. Then authors went and generated the transgenic Arabidopsis plant expressing the core protein fused exotoxins, core protein fused with inactivated exotoxins, and core protein fused with only the signal peptide which delivers the toxin to the phloem, and core protein fused with GFE. As you can see that this populated epids on the plant transgenic plants, as you can see that there is only less than as you can see here, there's less than 50 epids can found on the transgenic plant where that is expressing exotoxins, but in the controls, these populations has increased to nearly 300. That, and also they plotted a number of populated epids versus its days. As you can see that populations of epids has increased exponentially in the control fused peptides, inactivated and signal peptide and green fluorophore proteins, where in the case of hexapeptide, which is toxic, this population is within the 50. That means what it indicates that these transgenic plants expressing these exotoxins can be used for controlling the epid pests, which act as a vector for the many of the viruses. The conclusions of this work is this work demonstrates the use of core protein derived from the spider can be used for controlling the insect vector which are transmitted by the emepteran insects. Then they can make orally active, and core protein mediated delivery of neurotoxin peptide proteins can be used as a general strategy to deliver the several toxic peptides to the hemocyls. Here, core proteins or any other signal peptide is required to deliver the toxin from the guts to the hemocyls. That means this can be used as a general strategy. Unlike core protein, you can use some other signal peptides also for the delivering these type of the toxins. Okay. Because of time constraint, I don't discuss this very novel and seminal work done by Professor Pradyuman Singh group in National Botanical Research Institute. What they did, they isolated the fern, that gene from the fern. Well, this is a chitinase. Then they expressed this ones in the cottons through the transgenic cotton productions, and they can then these transgenic cottons protect the cotton from the whitefly infestations. And this is the first time insecticidal protein used in genetically modified crop plants effective against whitefly. And this is considered the best patented innovative technology from India. And this is listed as one of the best technology by the Nature Publishing Group. Agriculture background, I did BSc and MSc in agriculture and trained biochemist and also structure biologist. And then I thought I can use my expertise to design the chimeric toxins. Those can be effective against both hemipteran and lepidopteran insects. As I recently noticed that resurgence of white flies, especially hemipteran insects, in the BT cottons because of there is not much competition from the lepidopteran insects. Then I thought that can I design the chimeric toxins, fusing different toxins. Those can be effective against both lepidopteran insects and hemipteran insects like white fly and aphids. In this endeavor, what is the my protein toxin through the gene fusions against and white fly. Then through the literature search, I found out the one endotoxins that is everybody know that ones. And this cryotoxins unusually fused with lectin. And this cryotoxins is not only effective against lepidopteran insects and also some of the hemipteran insects. Then I found out one of the toxic peptide is a spider toxins, toxins. Okay, the spider, which is very compact toxins, which has parts between the beta strands, and this is effective against both insects. Then there is a problem. Can you see the slide?
Yes, not yet. It's showing starting. Yeah, you can see now. You can proceed.
There is one more lectin. It's called GNA lectins. This is called snow lectins, and this lectin actually take the any fused proteins from the gut of the insect to the hemocytes, and itself it's effective against both hemipteran and lepidopteran insects. Then I thought, can I genetically fuse these different toxins and make one more chimeric toxin that can be effective against both lepidopterans and hemipteran insects? Then I went ahead. Slides are not okay. Then we entered, we genetically fused these endotoxins, that is crate 41 toxins, with spider peptide toxins and also GNA. And you can notice that in between this endotoxins and SFNA, we introduced protease cleavage site. Why you introduce protease cleavage site is so that this toxin can be cleaved into two parts. One part, which is endotoxin, that can be effective against, this is effective in the guts, and another toxin, SFNE and GNA, this is effective against hemocytes. That means it can be delivered to the hemocytes, that can be neurotoxins. And especially this proteins, these are 16 proteases, they are present in the insect gust, and they can cleave any peptides or proteins contains these signal peptides. Then you entered and synthesize the genes which fused with endotoxins and SFNA and GNA, and then you can clone these ones and express it in the E. coli, and you purified these proteins. Okay. Then after purification, this is the STS page. You can see that they can purify the chimeric toxins. We did a lot of computational things to standardize the construct. We tried several constructs fusing with the different linkers. That means linkers are very important to assemble this protein as a soluble protein. It should be active. Then afterwards, we purified by histidine tag using chromatographic technique. As we can see that this toxin is this chimeric toxin can be purified and you can produce it in large quantity, and that is intact. Now you are looking for the collaborators, especially entomologists, to test these toxins, whether it's effective against lepidopteran insects and also hemipteran insects. And advantages this fused toxins is it may effective against two different class of insect pests, and it can have two different mode of action actions. That means insect can may slow in developing the resistance. And this is my group in Indian Institute of Technology. Mainly you are protein chemist and structural biologist. You work on the protein structures, and most of the work is carried out by Mr. Naveen, and currently he already completed his PhD. Currently he is a postdoc in Dundee University, Scotland. Thank you for questions now.
Hello, sir. Hello, sir.
Can I stop the slide sharing? Yeah, please go ahead. Please go ahead.
You can start.
Madam, shall I ask Madam.
Yes, sir.
Please go ahead. Go ahead.
So, thank you, Dr. Rajkumar, for your very nice presentations, especially of the transgenic aspects and the most important part of our management.
Sorry, your voice is breaking. Sorry, your voice is breaking. Can you repeat the question?
Okay. Thank you very much for your very nice presentation and that this kind of transgenic work, you know, the viruses and how we have to manage. This is the only best option and very good work of yours. And since time is the limitation, I entertain one or two questions. Please ask very precise questions to Dr. Rajkumar.
Hello, Rajkumar sir.
Please go ahead. Please go ahead.
Go ahead, please. Go ahead.
Myself, Shankarappa, your junior at UC level.
Oh yeah, I know you.
Yeah, thank you, sir. Thank you. A nice presentation. I have one doubt. What is this? How we can improve solubility of a protein? One.
There are several strategies we can use. You can add the tag that can increase the solubility, especially GST tag increases the solubility. And also there is a called ubiquitin. That ubiquitin again, it's a protein. If you fuse with another protein, it increases the solubility. And also you can the residues. You can generate the variants by computational approach. You can identify mutating some surface residues. You can change some hydrophobic residues to hydrophilic residues that can increase the solubility. And this is again, it's a $1 billion industry questions. You cannot directly say that you can change this residue. It can increase the solubility. You can go by trial and error method, but there are clear-cut strategies are there. Those strategies can be used to increase the solubility of the protein. You are not audible. Sorry.
Soluble protein is very important for study, no, sir. Exogenous application.
Yeah, that is the reasons I told you, in that construct, I tried different constructs, different constructs. Out of several fusion constructs, only one of the construct was soluble.
Okay.
And you have
what about in expression level in a transgenic? Expression
I didn't go for transgenic. Just purified from the E. coli bacteria.
Okay,
now we have purified protein. Now we want to collaborate with entomologists. Those entomologists can feed this one on the insects. If they are effective, then only we can think about transgenic through the collaborations. We are not in that stage. Now, currently we have only chimeric toxin, purified chimeric toxin.
Okay, okay, sir.
We don't know whether it's effective or not. That somebody has to test it.
Okay, sir. Thank you, sir. Thank you.
Can I add one suggestion to him?
Please, sir. Please, sir. Go ahead.
We did this kind of feeding the insect protein.
Krishna Kumar, sir?
Yeah.
Can you please switch on your video, sir? Please.
Okay, I'll just switch on my video. Okay. We did this type of work in feeding the glycoproteins. In fact, we cleaved the glycoproteins and then fed it to the insect vector. What we normally do is, a little bit of you have to add a little bit of some kind of a sugar, something or anything that makes the insect to very easily accept it. You can use a small tube. On top of that, you can stretch 2 layers of paraffin. After the first layer of paraffin, you place the solution that is of interest. Cover it in the top with one more paraffin layer. And leave the insect in the glass at the bottom. After they are starving ones, you will be under the microscope. You can very clearly see through the paraffin. They will be able to suck up the whatever the thing that you want. We have one or two papers on glycoproteins and phytopathology, and we were exactly like you said, were able to demonstrate that the GN protein, glycoprotein, if it is fed to the vector, it can actually block the transmission.
Okay, thank you for your suggestion, sir. But anyway, I am not going to do that work. I am collaborating with entomologists to test that once. I will take your suggestions and tell him.
Rajkumar sir is only he was entomologist.
Krishna Kumar sir, can you please unmute yourself?
Yeah. I cannot hear you. Sorry.
You want me to repeat or what is it?
No, sir. Done this.
Okay. Contact Dr. Ashok and he will help you.
Okay, thank you very much.
So, Madam,
Patil sir. Yes, sir.
shall we conclude his talk?
Yeah,
Yes, sir.
that's a very good talk, very good information.
Thank you, sir. Thank you for the invitation. Okay, it's my pleasure to deliver a talk in this forum. Thank you very much.
Madam, shall I continue with the continuation of theme 4?
Sir, we have another speaker, sir.
Yeah, that climate change and ecosystem.
Yes, sir. One keynote talk by Dr. G. Ravikant, sir.
Yeah.
It is a continuation of theme 4, climate change and pathosystem.
So
So with the permission of the chair, I would like to introduce Dr. G. Ravikant, sir. Can I?
Yeah, sure, madam. Please go ahead.
Yes, sir. I welcome Dr. Ravikant Ji to our international conference. Welcome, sir.
Thank you.
So I am very glad to introduce Dr. Ravikant. Dr. Ravikant is a senior fellow, A3, and is in the biodiversity monitoring and conservation planning. He has over 20 years of research experience and has led multiple research projects focusing on forest genetic resources. He applies molecular tools in the field of tropical forests, conservation, and species recovery. Course for number of medicinal plants and assessed the extent of adulteration in the medicinal plant trade. A core research focus has been on enhancing the conservation and sustainable use of forest genetic resources and in developing tools and research outputs to manage forest genetic resources of Western Ghats. In recent years, he has been part of the National Mission on Biodiversity, Principal Scientific Advisor, Government of India. As part of NBMHW, he is coordinating the component on biodiversity and agriculture, which aims at ensuring food and nutritional security by incorporating sustainable use of biodiversity. He has published more than 100 research papers in leading journals and led more than 20 projects funded by DBT, DST, NMPD, and other international funding sources. We are glad that you are the part of our conference, sir. I request you to present. So, Ravikant, sir, can you please unmute yourself?
Can you hear now? Hello? Yeah.
Yes, sir, you're audible, and the slides are also visible. You can proceed.
Yeah, thank you very much, respected chairman, co-chairman, rapporteurs, and all online listeners. Good morning. And what I'm going to talk to you today is very different. And this is across cross-domain theme, which is about plant pathogens and human health and need for inclusion in World Health Program. So before I talk about this, the importance of plant pathogens in human health, I would also like to talk about One Health Program. So I'm an associate professor or senior fellow at A3, Bangalore. So what we have seen in recent years is that global changes and human activities, large-scale anthropogenic activities, have led to global warming, migratory movements, habitat destruction, and all this has led to ecological disequilibrium and loss of biodiversity. This loss of biodiversity and ecological disequilibrium has led to increased risk of infections, and also because there is increased toxicity in the air, and all this have led to disruption in the biotic interactions between plants, animals, as well as between mutualistic organisms, as well as between different kinds of across kingdom organisms. And all this has led to emergences of new diseases as well as loss of immunity among people, especially to cope up with these emerging new diseases. And one important thing is there is interdependence of human health, animal health, and ecosystem health. As we have seen in the last 2 decades, that there are a number of new diseases that have emerged. bird flu, Ebola, Zika, swine flu, and now recently the COVID-19. And all this have been largely transmitted from animals to humans. In fact, what we have seen is that 60% of existing human infectious diseases are now from zoonotic. That is, zoonosis is a disease transmissible from animals to humans. And what we have also seen is that at least 75% of emerging infectious diseases to humans are of animal origin. And 5 new diseases seem to appear every year, and 3 of which are of animal origin. And what is also important is considering all this and considering the emergence of new diseases, in 2004, One Health organization was formulated, One World, One Health. It was a concept where, hello, yeah, it was a concept where they incorporated the ecosystem health, including that of wild fauna, into human health. And this was done when European ministers as well as World Health Organization met on June 15th in 2017 and paved the way for ambitious integrative initiatives in the World Health Program. And this, in the last 3-4 years, there has been a lot of changes, and there have been more stress with the emergence of this pandemic. There's been more emphasis on this One Health Program. This One Health concept includes common issues such as zoonotic pathogens. It looks at niche adaptation, epidemiology, vector transmission, culture resources, food safety, biosecurity, as well as pathogenicity mechanisms. And what is important is that it considers that human health, animal health, and environment health cannot be separated, and they should be looked at a wholesale level. And in recent years, last couple of years, there's also been wider coverage of this One Health program to include not just zoonotic diseases, but looking at antibiotic resistance, looking at food safety and security. I'll come to this a little later, and looking at vector-borne diseases, environmental health, chronic diseases, as well as occupational health and plant health. So in this, there was an emphasis on plant health. However, what has happened is, though the importance of plant health has been recognized in One Health program, the specific role of plant health is not clearly defined and often sidelined, largely because plant pathologists are not being included in this One Health program. And that is the one major reason why, though there is inclusion of plant health, because there are no plant pathologists in this group, that there is very little, specific role of plant health is not very clearly defined. But why should we consider plants? And it is important that at this juncture, we also discuss that plants are important, not only because they are source of food and nutrition, and and in the recent years, the emergence of genetically modified crops, and both of these have important concern with respect to food safety and security. The other important thing that we need to also look at this point is that plants also are susceptible to pathogens. They harbor human pathogens. I'll give examples of pathogens which are plant pathogens, which can be and indirectly cause plagues also. And there is also pathogens which can cross kingdoms and affect both plant and animal kingdom. And both of these can cause severe damage to human and animal health. So this is what we need to stress upon. But what I will first talk about is that the plants are important source of food and nutrition, and food safety and security is very important. So when I talk about plants, as we all know that plants are some of the major sources of food and nutrition, and directly and indirectly related to human as well as animal health. So when I talk about food safety and security, there are two issues here. One, when we talk about food safety, it means that there should be absence of harmful elements in the food, including absence of all kinds of pathogens, all kinds of microbes from the food. Second is, when I talk about food security, it means that there should be consistent availability of safe and nutritious food. So, but if you see, malnutrition and obesity are the two major pandemics, that the world is actually facing today with respect to food security. Malnutrition, on one hand, it's for many of the third world countries, it's one of the major issues, and malnutrition and lack of nutritious food to sustain life and major concern. The other major concern is increasing obesity rates in both adults and children, and this has also been termed as the first epidemic of the 21st century. In India, for example, it's supposed to be the capital of diabetes, and other physiological disorders are quite high. And if you look at malnutrition in India, there are about 7.7 lakh children under the age of 5 who died in 2017 just because of malnutrition. And what we see is among the Asian countries, if you look at Indian statistics, more than 61.7 million people are under the age of 5 are undernourished or malnutrition, and because of that, they have a stunted growth. The other major problem also is obesity. So both India has both the problem of underweight as well as overweight. So India has alarmingly about 9.8 million people who are overweight and about 20 million obese Indian women. And if you look at underweight, also there are 102 million Indian men and about 101 Indian women who are underweight. And it is said that India is about home to about 40% of global underweight population. So that is really a cause of worry. And the general cause of obesity is the imbalance between the food we take and the reciprocal to the energy we outlay. And largely, this is largely because of the wrong eating habits, less physical exercise, and lack of awareness. And if you look at the Indian diet, if you will see that the ones in darker colors indicate the deficiency, and the ones in lighter colors indicate that we are actually consumption is with reference to the actual with reference to the reference diet. So here, if you see, the vegetable consumption in India is very low. The fruit consumption is also very low. Legume consumption is also comparatively low in northern India, and animal source protein is completely low in northern India. In fact, only Karnataka seems to be very good with respect to Karnataka and Maharashtra seem to be little okay with respect to consumption of legumes and fruits, whereas most of the states within the country seem to have very little consumption of legumes, fruits, and animal source proteins with compared to the reference diet. And all this is important because all this leads to both malnutrition as well as obesity. The second important thing that we need to consider is about the food safety. And if you look at worldwide, about 1.5 million people have died in the last year due to contamination of food and water. And contamination of fruits and leafy vegetables by Salmonella and E. coli are the major causes for deaths as well as gastroenteritis diseases. In fact, they cause a significant burden on the healthcare systems every summer in India. And almost 10 lakh acute cases of diarrheal diseases and gastroenteritis diseases are reported, and about 3.6 lakh deaths every year occur only due to foodborne contamination. And foodborne outbreaks largely are caused by fruits and vegetables, and they are the major causes where because of and this especially the green leafy vegetables account for more than 20% of foodborne outbreaks of E. coli. And we also have outbreaks of Shigella and Campylobacter pathogens have also been observed. And this is largely due to irrigation with sewage water or with contaminated water, or washing these fruits and vegetables with contaminated water. In fact, salmonella contamination, which is known to cause about half of the known outbreaks, has been linked to fresh produce, especially tomatoes and melons, especially cucumber, and largely because of washing the fruits with contaminated water. And what also is important is that many of these salmonella, I'll give you the next slide, can enter into the plant system. It's not just surface on surface, but if the plants are irrigated with contaminated water, they can actually thrive inside the plant itself. In fact, what we in the recently study, they showed that 6% of beetle palm or palm leaves were screened, and it had 6% of them had salmonella contamination. And this is a study where they show that Salmonella enterica are found inside tomatoes and melons. No, it is inside. It is internalized in the plant tissues, and it was high. It was found in high percentage. In fact, this is the different cultivars, and what we have seen is not just in the external surface, but also internally. Also, many of these Salmonella strains were found inside the plant. So this is very important because this can cause, even if you wash it well, you are still going to get gastrointestinal diseases because of this contamination. So that's one important factor about food safety and security. Coming back to the main topic about plants being also susceptible to pathogens and harbor human pathogens and can cause directly plagues. So I'll talk about plagues, and though the number of pathogens shared between plants and animals or plants and humans are less compared to pathogens shared between animals and humans, many times plants indirectly play a role in the spread of pathogens harbored by animals. For example, we have all heard about the Nipah virus, and this Nipah virus seemed to have actually hypothesized to have been transferred from bats to humans due to the consumption of date palm juice. In fact, there has been now a very detailed study where they have shown that because of contamination of bat saliva in the date palm sap, and which was consumed by humans, that led to the Nipah virus transmission from bats to humans. And it also was transferred from the pigs, which ate the fruits infected with Nipah virus, which were eaten by the bats. And when the pigs were consumed, the humans got Nipah virus, both from date palm consumption, as well as by eating pork, that humans got the Nipah virus. And this Nipah virus became man to man, was transmitted from man to man and became a pandemic. So that is an important. So in this again, the plants were the major transmitters of the Nipah virus. So similarly, if you go back to history, we will see about the bamboo and plague being often associated, bag and rats plague. And this, is a very recurring problem in the Northeast, as well as we've also seen it in Western Ghats, where, when there is a bamboo bloom, a mass flowering of bamboo, there is a lot of seeds of bamboo, and because of this, there is a rat population boom. And because the rats boom, and then there's a large number of rats, and once the seeds of bamboo are over, they start moving into human population settlements in search of food, and with them bring the plague and other diseases. In fact, it was one of the major problems that happened in South India in the last century. And we also know about the Great Irish Famine, which was largely because of Phytophthora infestans, which led to starvation and death of millions of humans because of the infestation it caused in potato. And it was so severe that in Ireland, that it was declared as a Black 47, where the population of Ireland was actually fell by 20 to 25%, and it also led to a lot of migration. What we have seen in recent times. Similarly, there was a lot of migration, and almost 1 million people died, and 1 million more immigrated out of Ireland into other parts of Europe. So again, here too, the potato and Phytophthora infestans was the main cause of this particular thing. So crop pandemic would be as devastating for biodiversity as well as food security as COVID-19. And this threat is not just theoretical. Already we have, there are a number of reports of a dozen varieties of native and wild species are in danger of disappearing due to temperature changes and loss of habitat, and a number of new diseases emerging. And it's not just in animal diseases that are emerging, but also a lot of plant pathogenic diseases are emerging, and that are endangering popular crops, including varieties of wheat, potato, and bananas, and a number of other wild relatives. And therefore, there is a link between plant, animal, and human health. But what I would like to stress upon is that there are pathogens which can cross kingdoms and can cause diseases. And these can cause severe damage to human and animal health. The major question that I'm asking is, do pathogens really exist in plants, or do they have a wider host? And is there a possibility of these pathogens to jump from plants to animals? And both of these answers are true. And what is important is that we need to recognize this. And for a pathogen, the mechanism involving adaptation, colonization, and virulence are common across hosts, whether it's plant host or animal host. These the adaptation mechanisms of adaptation, colonization, and virulence are common, and they can overcome host defense system pathogens by disrupting the host cell wall in case of plants and producing virulence factors such as toxins and enzymes. The virulence factors in turn trigger expression or repression of host genes. And one important thing about plant pathogens is they have evolved evolutionarily to breach the plant cell wall, a significant barrier which the animal and plant, animal pathogens or human pathogens never encounter. So plant pathogens in that way seem to be much more virulent compared to animal and human pathogen. This is an example of a cross-kingdom transfer of Salmonella enterica. So here, this is a recent study which was actually done in US, GKVK, and IAC together, where they have shown that there is a cross-kingdom transfer of Salmonella from the rodent feces in the soil to the roots of Arabidopsis plants. So if the plant is infected with the rodent feces, and the plant has, or the animal has Salmonella infection, and if the Salmonella infection is transferred through the feces, it can go into the plant, from the plant it can enter back into the rats, and from the rats it can go back to the plant. So the entire cycle has been shown, and here is a study where they have actually shown that in the roots as well as leaves and fruits, there seems to be organ burden of plants with Salmonella enterica, and this is a very novel study where they have shown that it's not just between plants and animals, but the entire cycle can repeat and it can complete, where it can go from plant to animal, and from animal it can come back to the plant. So there is possibility of this particular species, Salmonella enterica, to actually have cross-kingdom hosts and can transfer between hosts. But what I would like to stress today is to give examples of plant fungal infections in humans. And I'll show about few examples, not just about fungal, but also bacterial and other diseases which have actually crossed the kingdom barrier and have actually, from plants, they have actually infected humans. This is one case study where, not just one, but there are a number of studies where they've shown that in this case, for example, Alternaria infectoria, Clausus blossom blight, and other diseases in a number of crop plants has been shown to infect immunocompromised individuals, and more than 20 cases of human infection have been reported because of Alternaria infectoria. And this is largely from the European US, whereas there are very few studies in India or in other developing countries. And all all the plant fungal or plant bacterial infections are all mostly reported from Europe and US, whereas in very few studies have been done in India. And so, what is also important is to see that apart from this fungal infection, and there are a number of other fungal infections that are actually seen, which are all these plant pathogenic infections, and all these plant pathogen fungus are seem to infect humans, and they have been reported to cause number of diseases, and largely blood infections, eye infections, respiratory tract infections, and surgical site infections. And there have been a number of case studies where they've shown for all these funguses that they have been able to transmit and cause human diseases, and all these are plant pathogenic diseases, funguses. Similarly, a number of plant bacterial infections are also being reported in humans. For example, this is a Rhizobium radiobacter, which causes crown gall in a number of eudicots. It has been actually shown to cause bacterium fetal death, and there are more than 5 deaths have been caused because of this particular bacteria. And what they've also seen is that has been able to infect model organisms, including C. elegans and Drosophila, and largely it was found in urinary tract in patients who had urinary tract infections or catheter-related infections. So, and there are a number of other bacteria, all these which are host for, which are seen in many plants, including maize, onion, tomato, and rice. All these bacteria have also been shown to produce blood infections or cervical lymphadenitis in humans. Similarly, viruses also are known to cause infections in humans, plant viruses, especially the tobacco mosaic virus, TMV, which is supposed to be stable in tobacco products, and has been found, in active state, or the TMV has been found in the lungs of both active as well as passive smokers and patients with problems in the lung. So this particular virus has also already been shown to actually have crossed the plant, from plant kingdom has already entered the animal kingdom. Similarly, there are a number of plant oomycetes infections also in humans. For example, this particular oomycetes has also been reported to cause blood infections in humans, and it can cause not only in humans but also in large number of domestic animals that this particular oomycete has been known to. And this was first reported from a person who was injured in the Afghan war, and he was infected by this particular oomycetes, and then later on people started reporting in many other places. So phytopathogens, whom do they actually infect? So with few exceptions, phytopathogens have largely infected immunocompromised or those who had less immunity, or post-surgical patients, or post-traumatic patients, or those suffering from severe wounds. And infection has largely been seen through damaged skin or respiratory tract. But what is important to know also and realize that plant pathogens are generally opportunistic pathogens, some of them, and they lack specificity for humans and animals. But however, when there is active immune disruption, or when the immunity is less, they can actually infect both humans and animals. And there have been also studies where, for example, in case of syringe microphage phagocytosis suggests that opportunistic pathogens have the potential to develop specialized infection strategies and during cross-kingdom infection. In fact, another important question to ask at this juncture is: can these pathogens, after they infect humans, can they spread from human to human? Though there have been no reported studies, no case studies, but there have been studies to show that they can multiply from human to human and spread. For example, a Providence virus, an insect virus, was able to infect cowpeas and 2 mammalian cell lines, and multiplication of this virus was confirmed both in the plant as well as in the vertebrate cell lines, as well as in the insect host. So, and replication of plant viruses in animal cells is possible, raising the possibility that these viruses may be able to cause human infections in future. And as we know, that human viruses have a high mutation rate, and it's only a matter of time before they gain upper hand in the evolutionary arms race. And this eventuality should be considered, and this should be studied. And the last one I would like to talk about is about the genetically modified crops. And as we know, I'm not going to talk much about it because there is a lot of gray area in this, because we know that GM crops are also associated with health risks because they can cause toxicity, allergicity, and genetic hazards. And though we don't have so many studies, but there have been also, in recent years, there have been also studies which have been coming forward and talking about the health risk associated with GM crops. In fact, one of the classical study or classical case was the Starling maize study, where the environmental group reported detection of the gene carrying, in the teco shells, which was one of the important gene that was inserted in the corn, and people reported adverse reactions because after eating these corn products, and allergic reactions included rashes, swelling of mouth and throat, vomiting, as well as diarrhea, and which actually led to recalling of the entire food boxes, and about 2 million food boxes were recalled and had to be destroyed because they cause important allergic reactions. So, in conclude, what I would like to stress and I would like to highlight is that there is a possibility that animal and human infections can be caused by plant pathogens, and they may become critical and can have critical impact on human health and safety. And recent genomic data reveal cases of horizontal gene transfer not only between bacteria and plant genome, but also between bacteria and other eukaryotes. And this is an important thing that we need to consider at this point of time. And with shared genes, the transmission and sharing of diseases across kingdoms will likely to become more prevalent. And understanding the disease processes and outcomes across kingdom would help us stay prepared to contain an outbreak. And this can be achieved by building network of understanding and crossing the kingdom barrier and creating more collaborations across animal kingdom, plant kingdom, and which is much needed in times of outbreak and other crises. And with this, I end. Thank you very much. Thank you very much.
Sangeeta, can you listen?
Yes, sir.
Madam,
We can
can
listen. Yes, sir. You're audible.
So, thank you very much, Dr. Ravikant.
Yes, sir.
For your very informative and very good talk of pathogens, plant pathogens making or infecting humans and animals. This is a very scary information, is what I think, because in our system, in the market, we eat every day with the infected materials.
Yes.
Take the vegetables, take the fruits. Every day we eat the bud necrosis.
Yes.
Every day we eat the papaya ring spot.
Yes.
Every day we eat the tobacco ring spot or tobacco mosaic. Tobacco mosaic is the most common. This is very scary, but anyway, thank you very much. It requires a lot of further research and confirm that normally plant pathogens do not make any kind of infections to human beings. But you have given a very good information. So in the long way, in the long run, probably we have to be careful with the plant viruses while taking the food. Thank you.
Yes.
Thank you very much.
Yeah, thank you.
Shall I conclude this session?
Sir, I have one question to Ravikant, sir.
Yeah, please.
Okay.
Sir, how safe are we plant pathologists working with the
Oh, that's a very good question. In fact, now, if you are especially working with viruses, you'll have to be really careful because, you have to take precautions. Not just with viruses, but with any pathogens, because if your immunity is low, there are chances of getting infected. But in fact, now, people are advised, the cancer patients who have undergone chemotherapy have been advised not to consume any fresh fruits. And lastly, that is because there are possibilities of plant pathogens entering their system also, especially because their their immunity is very, very low, they are advised not to consume any fresh fruits and vegetables. So it's an important, and we have to be careful. And another thing is that as we have not restrict ourselves to looking at only one kingdom, but it's time now to look at cross kingdom hosts as well as cross kingdom pathogens which can transfer across kingdoms, and people need to study and keep their eye open to look at these kind of pathogens that are emerging. Thank you.
So I think
Sir, I have one question. Ravikant sir.
Yeah, thank you. Yes.
I have one question. See, for any infection, specificity plays a very important role.
Yeah.
Even we are handling so many pathogens, you are very difficult to get infection. But with respect to specificity, how can define this infectivity of the plant pathogen to the human beings?
Right now it is too early to say, but there have been now studies and reports where they're showing that largely, but till now, whatever people who have got infected are largely people who have had who are in the post-operative care, or those whose who have undergone chemotherapy and whose immune system has been very, very low. Only those people have been infected till now. But viruses are very fast mutating, and in no time, they can cross kingdoms. In fact, all the animal pathogens are now being infected humans, and in later time, and because of all this climate change and change in the atmospheric toxicity, there is possibility that even plant pathogens can also infect humans. I'm just putting an eye opener, and and providing some case studies where people have actually shown that plant pathogens can also infect humans. But viruses are very fast. No time, they can cross. In fact, all the animal pathogens are now being infected humans, and later time, and because of all this climate change and change in the atmospheric toxicity, there is possibility that even plant pathogens can also infect humans. I'm just putting an eye opener, and and providing some case studies where people have actually shown that plant pathogens can also infect humans. But these are very time, they can cause. In fact, all the yeah.
Kumar sir,
Shall I-
please
shall- Madam, shall I conclude the session because time is almost running out? Okay, I think it is the time to. Sorry, if there are some questions, you please contact Ravikant maybe later through your mobile or through email. Because of the time shortage, I will make the conclusion. This was the very excellent session actually considered by our organizing secretary, Dr. Devappa. Virus, now everybody, even the uneducated, the village persons, everybody is aware of the word virus. Very excellent. And we had the invited speaker, Dr. N.K. Krishnakumar, renowned entomologist, delivered talk on the Aspergillus transmitted through thrips, very important. And another most important was the Vipin. He initiated the another area of our transmission. Bagmo viruses are normally transmitted through whitefly, but now he is giving the information that viroids are also transmitted. And another important, most important topic, it was the protein or the toxic peptides that they can be used to manage the virus's vector through transgenics, and altogether the entire health management of humans and animals. Initiated the transmission. Please, you can just talk to anybody later. We'll make the conclusion. When Ravikant has given again the very important informations, we have to be very careful with the infected materials, especially with the viruses or the fungal infected materials or the bacterial. So I thank all these speakers for their excellent presentation, and I thank the co-chairman, Dr. Ashokan, and also rapporteurs, Padmaja and Manjunath Reddy, and last but not least, our beloved Dr. Devappa, organizing secretary of this e-conference. Excellent way of organizing. And I thank him for having given the opportunity to chair the session. And also Dr. Sangeeta, who is doing excellent work in conducting all the organizers for conducting this e-conference. Once again, I thank you all.
Thank you, sir. Thank you, Kapil sir, for chairing this session. The co-chair, Prabhurat sir, and the rapporteurs for your comments, compliments, and suggestions. My special thanks to Dr. Krishna Kumar sir. Krishna Kumar sir, are you audible? Is it audible? Can you please unmute? Sir, and your video also, sir, please.
Which video? I am on the video, but the only thing is I'm hearing a lot of crosstalk.
Okay. You have given a keynote talk. Thank you so much. Thanks to all the participants for patient hearing. Dr. Krishna Kumar sir. Krishna Kumar sir, are you audible?
I am audible.
Is it audible?
I am able to hear.
And your video also, sir, please.
One.
Okay, sir. We will next move on to the next session. I welcome you all to the 9th session of this international e-conference, that is Integrative Plant Health Management. I welcome the chairman, co-chairman, rapporteurs, all the keynote invited and oral speakers and the participants to this session. This session is chaired by Dr. P. Chaudappa sir, co-chaired by Dr. S.T. Enjirappa sir, and the rapporteurs are Zahir Basha and Dr. Raghunath Reddy. We have one keynote talk, one invited talk, and 3 oral presentations. It gives me immense pleasure to introduce the chairman of this session, Dr. P. Chaudappa sir. He is the former director of CPCRI Kasargod. Is specialized in molecular plant pathology. He has 30 years of research experience in molecular characterization and management of Alternaria Colletotrichum Phytophthora associated horticultural crops. He has published almost 120 research papers, 8 books, and has guided several PhD and MSc students. He has held several positions and is the recipient of several awards and is the fellow of IPS and other societies. I welcome you, sir, and I hand over the session to you. Over to you, sir. Thank you. So kindly unmute yourself and proceed, sir.
Rajeshwari, one clarification from Dr. Krishna Kumar.
Yes, sir.
I have sent in the chat, there is going to be a meeting on problems associated with onion seed production and also iris yellow spot virus. The session will be chaired by the Honorable Member of the Niti Aayog, Dr. Ramesh Chenji, and all the others are there. I have given the details and the link in the chat box. So those of them who are interested to join this meeting on 24th can make use of it. Thank you very much.
I'll read to all the participants the Krishna Kumar sir's message. There will be a webinar on onion seed production and iris yellow spot virus on 24th, organized by Ethological Society of India and NRC Onion and Garlic. Yes, sir. Is it okay? Okay, sir. Thank you so much. Over to you, sir. Chaudappa sir. Hello, sir. Sir, Chaudappa sir is not audible.
Chaudappa sir is here.
Yes, sir. Over to you, sir. Please proceed, sir.
And Enjirappa. Chaudappa and Enjirappa. Enjirappa is the co-chairman. Chaudappa is the chairman. Co-chairman is there. Yeah. Most probably Chaudappa will join. You can take the session. Chaudappa sir is also there.
Chaudappa sir is there. He is unmuted, but we are not able to hear him, sir. He's talking, but we are unable to hear him. Can you please unmute?
Oh, again, I don't know what's happening.
Please unmute, sir. Yes, sir.
No, it's automatically, okay. Okay.
So Chaudappa sir is visible, but we are unable to listen to him, sir.
Talk to him over phone and then see what he can do.
Calling. Wait few seconds.
Meanwhile, let me introduce the co-chair of this session. Chaudappa sir is visible, but we are unable to listen to him, sir.
Talk to him over phone and then see what he can do.
Dr. Enjerappa sir is the professor and head Department of Plant Pathology, College of Agriculture, US Raichur. He has total 29 years of experience and he has guided 8 MSc and 4 PhD students. His thrust areas of research are management of bacterial blight of pomegranate and onion. He has established plant health clinic and conducts trainings to farmers and extension functionaries. And we are glad to have you here, sir. Enjerappa sir? Are you there, sir?
It's audible to
Okay,
me.
sir. Yes, sir. Thank you, sir. I welcome you, sir, to the session.
Okay, thank you.
Chaudappa sir also join. He will join.
Okay, no problem, madam. Proceed.
Yes.
So can
Professor Enjarappa, you can start the program.
Okay.
Chaudappa sir will join.
Wait. Delegates, I think I'm audible to you.
Yeah.
Yes, sir, you're audible.
Audible to me, no? Okay. Okay, the session is on the integrative plant health management. Now I welcome Dr. K. Prabhakar for the keynote address. His topic of presentation is the recent trends in biological control of post-harvest pathogens. Now I welcome Dr. K. Prabhakar to give the keynote address.
Enjarappa, you will join later because he is having one meeting in Coimbatore. You will join later.
Okay, and then I invite the next speaker, that is Dr. Dinesh sir. The topic is management of bacterial diseases of soil nature vegetables. Now I welcome Dr. Dinesh Singh sir. Welcome sir for the presentation of this integrative plant health management this one session. Welcome you sir. I think Dinesh Singh sir is audible.
Yeah, he is there.
Yeah, he is there. Hello sir, good morning.
Good morning. It is audible?
Yeah.
Okay.
Please address sir. Management of bacterial diseases of soil nature vegetables.
My slide is that visible?
Okay,
yeah,
visible, no problem.
Okay. So good morning to all. I am going to speak on biological control of bacterial disease of Solanaceous crop, Solanaceous vegetable crops. And this is that why we are going to this managing these bacterial diseases, because that will go for the not only the production but it also affect the quality of the produce. You can see this is my first slides. It's about that these vegetables, particularly the Solanaceous crop, is called as the protective food. Because of that, it has the rich source of vitamins, minerals, and anthocyanin, phenols, and amide proteins, which is that's called it is a protective food. In case of the tomato, because a rich source of the lycopene and phytin, neurosporin, and the carotene, which is also one of the provitamin A and vitamin A. Likewise, potato, it is a good source of that carbohydrate. Besides that, it is also of that essential amino acids like lysine, which help to enhance the protein utilizations. And capsicum, it is also vitamin source, vitamin C, which and vitamin K, carotenoids, flavonoids, which is the rich source of that, which is also called as a protective foods. And chili is also, which have the folic acid vitamins, which can use as arthritis and asthmatic problems. Besides that, eggplants, which is also have that phenolic compounds, and particularly caffeic and chlorogenic acids and flavonoids, which has a main role for the controlling the high blood cholesterol. So that's why I have the, not only in this case corona period, this is the one of the develop the immune system to that human being. Come to that bacterial disease of Solanaceous crop, 12 diseases I have listed here. And you can see that this is some disease of bacteria, particular specific to the a particular crop like potato. It has the common escape, ring rot, and pink eye disease, which is specific to the potato. And in case of the tomato, you can go for the bacterial escape and bacterial the important disease, and pith necrosis, which is specific to the only the tomato. But like bacterial wilt, you will find that it infect all the solanaceous crop, tomato, capsicum, chili, eggplant, and potato. And bacterial canker, which infect tomato, capsicum, and chili. And bacterial spot, which infect the tomato and capsicum. So these have the, some have the specific, some have the wide host range, including that solanaceous crops. So I go for the first disease like bacterial type tomato and capsicum. You can see based on that importance of the disease, it is impossible to control the disease when it's present in the field. And the losses caused by this is go to up to 52% as reported in Sarma 2011. And based on the symptoms and the colony of that bacteria, you will see in this slide that spot, brown circular both on the leaves. And also on the young fruits, lesions appear as a small dark slight red dots. And on mature green fruits, you will find the brownish black elevated scapes. This causal agent earlier that is reported only Xanthomonas compactoria. It is divided into 4 groups. Now it is group A, B, C, D. Now give the status of the species that is now divided into 4 species: Xanthomonas ubicicatoria, Xanthomonas vesicatoria, Xanthomonas proferens, and Xanthomonas gardneri. So now it is reported 4 species. And group A, B, and D that infect both tomato and pepper, where is the group C will only infect to the tomato. There is some reports are there where it is the ubicicatoria present, then perforance is not infect to the host, but now when dominated the perforance, ubicicatoria is not present there. So that there is some reports are there like. Next disease is the bacterial speck of potato, that is called tomato, caused by the Pseudomonas syringae of tomato. These are the foliar disease and infect the leaves and the fruits also. Small black spots are not present, but it is smaller to that spots, and these spots on the fruits are very small, and it is like a pinpoint like and do not penetrate very deeply into the tissues. So in unripe green fruits, you will find the spots often have darker green halos, and ripe fruits, the spots can leave subtle yellow halos. So next disease, bacterial canker. This is also that important. Now it's South India, this and some North India is also this disease is reported, particularly in the Karnataka and Uttarakhand area, states of India which have been reported. And that is caused by the Clavibacter mechagenensis. Obviously, mechagenensis are the losses due to disease incidence of 25 to 48% by this disease. And in fact, you will see that the spots on that leaves, and that you will find on the leaves is curling leaflets and browning of the leaves. Petioles remain green and firmly attached to the stems. On the fruits, you will find that dark brown center of the spots, and rays surrounded by the distinct yellow. That is one of the peculiar symptoms of this bird's eye spots on the fruits. You will see that on that fruits you will find such type of the symptoms. You will find fluorescent colony and the cells of the bacteria, and this is the colony of the bacteria. So this bacteria is the gram-positive bacteria. Next is to just say it is a very minor disease. It is somehow it is reported and it is sometimes confused with the bacterial leaf spot, but it is the Pseudomonas syringae pathovar syringae. Next is the pith necrosis of tomato. It is mostly that reported from that greenhouse, and this disease caused by Pseudomonas corrugata, and this occurs mainly in greenhouse, and symptoms typically develop in the fruit, first fruit or closely mature green. And also we see that this is the pith area which is rotted by these pathogens, and you will find also rotting on the fruits. Now this is the one disease, the zebra chip disease. It is a non-culturable bacteria, that is a Candidatus leverivector solanacearum, which infect the potato, tomato, and capsicum, all three crops. And this is the found on that you will find on the leaves and also the stem. Branches of that also go for proliferations, and these stunted shoots, certain internode and solar internodes, aerial tuber formations you will find here. And also tubers may have enlarged lenticels, necrotic flaking of the vascular tissues, stick along with the medullary rays, and that is when slices of the potatoes are fried. So that give that the like a zebra, that is give the symptoms like that one when it is fried. So that is called, this is the zebra or chip. And then it is on tomato, we will find that stunting, typical growth can be spiky and chlorotic, and leaves may be mottled, chlorotic, and in the purpling of the midvein and leaves in some cultivar. So that is the one which is the symptoms on the tomato, and this capsicum is you found also the same type of shortening of stunting growth, certain internodes of the stems, leaves can be pale and green, chlorotic, and then it may be leaves may be cup shaped, and this flower abortion may be that absorbed and tip necrosis. So this is that the symptoms of the zebra chip disease in capsicum. This next very important disease, and it is affecting all the solanaceous crops, like bacteria built of solanaceous, which infect by this Ralstonia, that is called by Ralstonia solanacearum complex now. And these symptoms of the potato brown rot is found in tubers. You will find this is the vascular disease, and in the pith that also vascular system is browning of that one of the important characters, and also oozing. When you cut the stems, you will find ooze will flow from a cut infected stems. You will find the yellowish and grayish bacterial which come out. So that is one of the typical symptoms, and also go for the diagnosis for that. And this is the typical colony of Ralstonia solanacearum infecting that. And on the SMSA media, you will find this typical colony for that. And go for that recent classification of Ralstonia solanacearum, because this is the now divided. Earlier it is Ralstonia solanacearum. Now it is go for the species level. 3 species have been now classified, that is Ralstonia pseudosolanacearum, Ralstonia solanacearum, and Ralstonia CGP. So this is the distribution, and now it is divided into 4 phylotypes. Phylotype 1 and phylotype 2, A and B, it is only the American, and then this phylotype 3, African type, and this is type 4, phylotype 4, it is from that Indonesia. So that is go for different phylotypes, sequivars, biovar, and the ecotype and races. For that Ralstonia, you know that this is the 5 races have been characterized, and this based on that which race and what is the combination, based on that given the status of the species. So now it is 3 species in place of only 1. Next disease is the common scape, which is specific to that only potato. This is the one Streptomyces scabies caused by this, and now it is reported 13 species of the Streptomyces which caused that common scape based on the conditions, soil conditions, environmental conditions of that infect what type of the species to the cause the disease. And this is the colony of this, and this is the symptoms of 3 types of symptoms. We will netted type, or it may be escape, superficial, or maybe pitted. So 3 types of the symptoms you will find on the, and this is the go for the quality, reduce the quality of the produce. Next is the ring rot of potato, although it is not reported from that India, but these have the also significant importance and also have the quarantine problem for that. So this is that the disease is caused by the clavivector Clavimetiagenesis septodonicus, which is causing the disease of ring rot of potato. Is generally confused with whether it is a ring rot or it may be the caused by the brown rot of potato. So this is go for typical symptoms of in potato tumors. That is, and also it is also we for the circle of that vascular systems. And then this is find because this is also gram positive bacteria, and this colony of bacteria we find on the medium. So you will find that different pathogens causing different type of symptoms on these graphs. Next is to black leg and soft rot disease. This is caused by earlier it is Irvinia group, and also now with the soft rot is not only the Irvinia or Pectobacterium. Now it is reported so many the bacteria which is Irvinia Carotobora group, or Irvinia Carotobora subspecies Carotobora, Irvinia retroseptica, Bacillus, Clostridium, and now it is a Dickia, new name of come to stem leg, black leg, that is called Dickia soleni. So that is also is an important disease in the potato. This cause the disease, and this is the symptoms of the black leg of the potato, and this is the symptoms in potato is causing soft rot, and this is tomato. So this is tomato and potato is important disease. This is it is not much common, but it have reported academic point of view. You can see know that this is the Pseudomonas fluorescens which are infecting that Parky patches. And this is the called as a pink eye disease of potato, and this skin appearing in the affected area is a diagnostic feature of pink eye. You will find this is the margin of here the eye is there, that is in that. So that is a subtle pink discoloration noticed below the affected area. So now go for the management. Management of bacterial disease like other fungal and bacterial disease, it is very difficult for the particularly the disease it is soil borne like bacterial wilt or ring rot or common escape, but foliar disease is quite possible to manage the disease. So for the management of solanaceous vegetables caused by these bacteria, culturable practices, resistant varieties, chemical control, and biological control. So in bacterial diseases, chemical control is very less options like in case of other fungal diseases, you have so many fungicides, but in case of the bacteria, we have very less number of options for managing the disease. Only the copper oxychloride or somehow the streptocycline antibiotics we can use for the management, but no other options are there available to manage these bacterial diseases. We go for the combining of this is integrated disease management, best option to managing the disease. We go for only biological control, because why we are going using this, because of the many advantage over the chemical methods. And biological control, I will discuss only the microbes and resource resistance, and source of resistance, and then botanicals. So I will discuss one of these. So, and this why we are going for the biological control, because of these hazards of the pesticides and development of particularly bacteria using the antibiotics, that is a very chance of to develop the resistance against the particular pathogens. So we are avoiding these antibiotics and also this hazardous to the human beings by grave dangers. Consumption of glass, we will be here to pesticides in India, and this 7,717 tons which are reported 1516, and class 1 is the 2,254 which is insecticide. Out of that you can see in the diagram cases of accidental intake of pesticides and death other than suicides. So 2014 and 15 you will find the red color of that which have show the number of deaths, which have number of poisoning cases are reported, and how many of the deaths are by using these pesticides. Death of pesticide farmers continue to die as a Indian plots global norms, and it is first report that in 1958 in India the first report of poisoning due to the pesticide was from Kerala in 1958, where over 100 people died after consuming wheat flour contaminated with the parathion. And next is to the worldwide death and chronic disease due to the pesticide poisoning number 1 in million per year. So that is one of the what is the danger for that human beings. Not only India, it is go for the in the world also for the using these pesticides. Recently, that have been go for the banning of this pesticides like insecticides, herbicides, and fungicides. But in the list of that fungicide, we will carbendazim, tinopat, tiram, or mancozeb, methyl, gineb, and peptide. But there may be what happened, we don't know. But in this list, there is no that which is used for the management of the bacterial diseases. But because we don't have the much option in that managing the bacterial diseases also. So we go for the microbes using it as a biocontrol agent. We have the many advantages of why we are going for, because it is less costly and cheaper than any other methods, and we have protected to the crop throughout the crop period. Once you have applied, it can be go for the longer period, and its application of biocontrol agent is safer for the environment and also to the person who applies to them, and they multiply easily in the soil and leaves no residual problems. Besides that, have the so many advantage of that. Why we are going for the biocontrol part? Because it's microbes in the rhizosphere. We find that a 3 category of the good, or it may be the bad, or it may be ugly, which is found in the rhizosphere or phyllosphere. Go for the good, it may have because of that tolerance to the abiotic stresses. It have the physiology and metabolism reactions will induce resistance, growth, and development, helping that nutrient acquisitions like siderophore extract that and protection against the pathogens, antibiotics, and development immune to disease in the plant systems. So they have the because this is the role of these microbes, and then bad because it will cause the disease and food contaminants. As in the previous that lecture I heard that this is how these are now go for the issue for that using these microbes go to infect to the human beings, which is earlier we don't know think about that. So this is also one of the area where this have to play important role, which come under the category of now ugly. This for the management of this using biocontrol agent like a tomato, we have the long list of that different group of like actinomycetes, bacillus, penibacillus, enterobacter, pseudomonas, streptomyces, A. virulent strain of Ralstonia solanacearum, yeast like Pichia and Candida, and mycorrhiza like Glomus and Trichoderma hargianum or viridi. They are in combination with the Pseudomonas fluorescens, which is using people for that controlling of this bacterial disease. In brinjal, you have using the people the Bacillus megaterium, Pseudomonas, and also in chili and potato. These are the bacteria and fungus can be used for the managing of this disease. We have also studied some isolated the antagonistic bacteria from the rhizosphere of chili, and it is found that out of 59 bacterial isolates, 33.6% isolates show the strong inhibition zone against Ralstonia solanacearum, and they have that causing the bacteria built of chili. So that we see that inhibition zone how for the testing of the in vitro conditions to inhibit the growth of that. This is the growth of the Ralstonia solanacearum, and this growth inhibition zone found by this antagonistic bacteria. So that have the it is not whatever the you have isolated from the rhizosphere all will go to antagonistic have the ability to inhibit the growth, but 33% isolates have the positive for the inhibiting the growth of the pathogenic bacteria. Not only the inhibiting, this go for when go for the isolation of that, we also think about that production of the PGPR activity of that stem, like this is the indole acetic acid productions of phosphorus solubilizations, siderophore production. So these are that have the positive for that have the ability of which strain you are going selecting for the biocontrol agent have both antagonistic and plant growth promoting ability. So that can be chosen for that use as a biocontrol agent. And for that the proportions of the isolates showing that when out of 59, the maximum go for that 58% isolates so that production of that phosphorus solubilization, siderophore productions is the only 6%, And then ammonia production 6%, 11% is for the ammonia production, 19% for the indole ester process. So that isolates so have different proportion for the have the PGPR activity. Out of that, you can be choose the best one for that use as a biocontrol agent. We have used for this biocontrol agent Bacillus subtilis, Pseudomonas, and Trichoderma, and it found that in the field study in Haldwani area, it found that a combination of Pseudomonas fluorescens and Trichoderma harzianum give the better result to control the bacteria built in the field conditions. And data say that data can be see that the more biocontrol efficacy is go more than 50% in case of the combination of the biocontrol of the Trichoderma and the Pseudomonas fluorescens as compared to other combinations. Likewise, we combined with the effect of bleaching powder and antagonist to control the bacterial build, and it is under glasshouse conditions that so that Pseudomonas fluorescens plus bleaching powder give that better result. It reduce that and disease control, and in alone it have the lesser. So in combination of the less this affect the disease control by using that resistant variety and susceptible variety. So if you have the combination of resistant variety plus bleaching powder and the biocontrol agent, it gives the better result as compared to use the susceptible variety. Here is also one of the field studies. We have shown that this is the variety go for the combination of the resistant variety like bacteria built Arca samrat we developed by the IHR, and Pusa ruby, the highly susceptible to the bacterial build. It gives the better result, disease incidence reduce, and some we have found in the field condition there is no disease in up to that 60 days after that. So after infection, so that give the better result. You go for the field. In combination with the integrated disease management. Besides that, bacteria built common escape is also using biocontrol agent. It is give the example of antagonistic fungi Trichoderma harzianum, Penicillium digitatum aspergillus flavor give evaluated for biological management of common escape, and among them that Trichoderma harzianum was highly effective to lower the disease incidence. Besides that, Pseudomonas moselli and in combination with the vermicompost give the best plant growth and yield and reduce the disease incidence of the common escape. Besides that, it is also use of non-pathogenic streptomyces. Biopesticides is also like a plant, the Granium retains plant state give the promising result to control this common escape in the field conditions. For the soft rotting, they are also using different type of the microbes for the controlling by Streptomyces, Pseudomonas fluorescens, Bacillus subtilis, and Pseudomonas aeruginosa, which is reported Salem et al. 2018, and it gives that better result in the protecting that black leg disease, which is caused by Dickia soleni, by Serratia polymitica, which is give the better result in the, and also not only to reduce the disease incidence, it is also to check that further spread of the next generation progeny to that. So that in case of the protective plant, by reducing the black leg development on average by 58.5% and transmission to the tuber progeny as latent infection by 47 to 75% by this Serratia. So that have not only to control the disease, but it also check that further transmission of the disease to next generations by using these microbes. And the next example of that bacterial leaf spot, which are used for the Pseudomonas fluorescens and Bacillus subtilis and Bacillus pumilus, for that both it provided significant suppression of bacterial spot in the field conditions. G et al. Reported 2006. And combined application of the Pseudomonas fluorescens of the and PGPR strain 89B61 provides significant control of bacterial spec and spot of tomato. So when applied that this biocontrol agent, it is not only controlling the one disease, it may be the how the mixed infection in the field that can be also checked. Seed bacterization of the Bacillus subtilis, which are reported in the tomato to control bacterial leaf spot by Kavita and Umesa 2007, and Penibacillus ilgi is also give that controlling that bacterial leaf spot, and it was comparable to oxalic acid, which is controlling this disease. So this is reported in Lee Dew et al. 2020. So that he give that better result. And we have also studied this, develop a liquid formulation for based on the Bacillus subtilis, and it was found that it is not only control the bacteria wilt when applied in the field, that is a bacterial leaf spot, it is also controlling the Alternaria solani and bacteria Phytophthora infestan in the field conditions. So that is also give the options, the advantage of this biocontrol agent. It is not only to specific to the fungi or the bacteria, it is controlling the both pathogens, fungal, both group of the pathogens, fungal and bacteria both. And in biological control of canker, some reports are also for it not much work has been done on this aspect, but this some potential biocontrol agent like Bacillus subtilis, Bacillus thuringiensis, and baby Bacillus brevis have been reported to control the disease, and some formulations of Bacillus subtilis and Trichoderma harzianum has also been reported to have controlled this bacterial canker disease. Bacterial speck of tomato is not much work had been done. One I have got the one difference, which is the Romero et al. 2016, which reported that Pseudomonas and Bacillus amyloidosis are methylotropicus and Pseudomonas veroni, which have found potential to control these bacterial speck of tomato. Go for the mechanism, why these are the effective, why these microbes used for the controlling. So the controlling of this bacterial disease by the mixed path antagonists, by have the different mechanism to control the infection, by antibiotics, use of the antibiotics, or it may be lytic enzymes, unregulated waste products which have been used like ammonia, carbon dioxide, hydrogen cyanide, and physical chemical interference, the blockage of the soil pores, germination signals, consumption and molecular crosstalk, and that confused to the pathogens also. By indirect antagonists like competition for the food, that is produced of the exudates, leachates, which is used by this because they are the fast growing as compared to pathogenic bacteria. So that have more, and also production of sideropore scavenging, which is iron sparsity surrounding the rhizosphere. So that it go for the used by this biocontrol agent, and induction of host resistance by contact with the fungal cell wall, or it may be that detects a pathogen associated molecular patterns, phytohormone mediated induction induced resistance to the plants by using this biocontrol. So here we have go for the mechanism of bacterial suppression by tomato plants by polypeptide genes. We have targeted 3 genes and cloned these genes in Bacillus amyloidosis, and then the function of these polypeptide genes, and that is the 3 genes we are targeting: bacillin, macrolactin, and diffidine. And this we see that in photographs, that is the this is the parent, and this is the clone product of this one of these all 3 genes, which have inhibiting the growth, but it was lesser than the parent strain. Because of that, how all these the gene in this parent, but it is an individual which is clone that. And this is the mechanism for that. Why go for this? The mechanism for that this product is go for the lysis. This is the electron microscopic study, so that this is that untreated, and these are the cells of Rhizostonia solanacearum treated by these polypeptides, and this so that they have the lysis of the cell wall, and this inhibit the, because of that they are inhibiting the growth of the that bacteria, pathogenic bacteria. And this induce one mechanism for the not direct for the lysis, but it will go for the induce to the plant system to, because you will see these photographs, you will see that there is no inhibition of that bacteria directly, but we have go to reduce the disease incidence because of the mechanism induced resistance to the plants. And we see that the data 12 days, 20 days, and 30 days, when you this is that induced strain which have to go for reducing the disease by as compared to other. So that is the one of the induced resistance mechanism involved in further using the microbes. And this is the some examples so done by these other workers on this bacterial build by using this. And next to resistance source of using some different centers of ICR and also the universities have developed a resistant against the bacterial wilt like a tomato, brinjal and chili. But in case of the potato, there are no much work has been done in this direction to develop resistant variety to against the bacterial build. But the resistant source are available which can be used by the breeder to develop resistant variety against bacterial build. So these are that different in case of so many work has been done in case of the tomato and brinjal to control the bacterial build. Bacterial canker, they are not much work has been done for the develop of resistant varieties, but only the source of resistance are available. And bacterial leaf spots, some are reports are there which lines are have to show resistant to the disease, like they are Ohio 4012-3, Ohio 40, so and varieties are there which are available. So and Sarma 2011 reported that resistant to bacterial spot in both Hawaii 799 resistant and CRA6 for modality resistant was given by Polygen. So there's some work we initiated in India also, but not much work on this aspect of the developed resistance variety. And pink eye, there is some reports in the USA, and common escape is also no resistant varieties are there. Only source of resistance are available, so that can be used for the developed resistant varieties. Grafting, one of the techniques which are used for, because soil-borne pathogens, not only the bacteria built, it can be fusarial built also, and also go for the resistant to the nematodes. So because nematode is also contributing to the cause the infection to the plant by the fungi or bacteria. So this source of resistance are there which can be used as a root stock like Hawaii 7996, which can be used as a root stock to control this in tomato. And also in the eggplant varieties used as a root stock by EG2, for example, for 203, EG195, ES69, which is moderately resistant to control this bacterial build. So this is one of the grafting techniques are there, which some ICR institutes are using this technique to develop that grow this tomato and brinjal crops for that against the bacterial build, because this is a very severe problem in the coastal areas. So that is one of the techniques which can be used, but it is laborious and labor consuming, which can be used for that. Then botanicals, not much work has been done on this aspect of using the botanicals for control of these bacterial diseases, but some examples are there. Although these botanicals are have the importance for because these have the safe, eco-friendly to control the diseases, and have especially the essential oils which exhibit the different level of antimicrobial efficacy to various ranges of fungal and bacterial pathogens, and they efficiently reduce the major disease in the crops. Like in one example of tuber treatment with the jute leaf extract, so the more pronounced inhibitory effect on the soft rot growth both in the in vitro and vivo and during the storage. So that have been reported Rahman et al. 2012. And one example in bacteria with thymol oil, which derived from the thyme plant, reduce the bacteria build in tomato plants. And one example for so that in Bexa orellana, which is the bark can be used as leaf, bark, or flower fruit and fruits or seeds. So inhibitory effect of bacterial leaf part. So that is not much work on this aspect have been done. So this is the scope, new scope to can be try for to develop that one that identify scheme that which how the botanicals are effective to control the bacterial diseases. And because these are the botanicals are the constituent of several classes of substances like monoterpene, hydrocarbons, phenol and methyl esters, monoterpene, alcohols, and sesquiterpene, electrons, and these are the ketones, aldehydes, ether. These are have the active principles which can be effective to control that bacterial and fungal diseases. By using this, the active principles are effective, but have the some limitations why these are not much popular, because of they have the specific constants regarding the efficacy of the botanicals as a biopesticide. Because of that, you have so that constituents of these active principles may be changed based on the part of the plant or maybe age of the plant, which affect their compositions. They require greater application rates. What rates are 20% to 20%, 10% of that concentration you are going to use. So that active gradient require higher rate, and that frequent reapplication when used as a outdoor materials. So that is one of the constants which is using for that. And then also the what type of you go for using final product for the formulation as ready to use as well as the extraction yields could be limiting the factors. So these are the some constants which are not much popular of the botanicals using the bacterial diseases or any other disease in the plants. Cultural practices which support this the biocontrol by using like you can be reduce the inoculum of the pathogens present in the soil or in the plant residues. So use only disease-free seeds obtained from certified nurseries. Remove disease plants as soon they are detected. Hand tools which is used for in the cutting of the tubers or other activities. Support wires should be disinfected. And also crop rotation for soil-borne, particularly soil-borne pathogens like common escape, ring rot, and wilt, which is also important for that. Roguing out of wilted plants and infected plants, which is a soil amendment, which have improved the efficacy of biocontrol agents which suppress the disease in bacteria wilt and common escape, and also have the new area by using the biofumigation like cabbage and mustard, which can be used to control the soil-borne pathogen like common escape, or else can be used with bacterial wilt, which can be controlling these soil-borne pathogens using biofumigations. So that is one of the area which can be go for the using as a biofumigations in place of the chemicals. And other culture like a whole seed pot, in case of the potato, you can use the whole seed potato for that, and also the managing the irrigation to control the disease, and also mulching. One of the also have the recommended for practice for the common escapes, and delay in the planting in the southern eastern plains decrease the incidence of the common escape. So that is how give the examples of the cultural practices which did not directly affect the disease incidence, but it is manipulate or reduce the inoculum of the pathogens present in the soil or in the pleural spheres. And the management study zebra disease, which is not much problem in India, but it has the some because this is the problem transmitted by the psyllid vector. So that because it's a bacteria which is not culturable but transmitted by the psyllid vector. So that is managing the insect. One is the important part using the different follow the crop rotation of the insecticides of different mode of actions. Use of neo-green traps is also reported to highly efficient to monitor the vectors. So with this, I thank to that organizer giving me opportunity to present some my works and also part collected from different sources, and also thanks to the organizer giving me opportunity for that. So thank you all for the patience hearing.
Thank you, sir. Sir, Dr. Chaudappa, sir, can you please unmute yourself?
Can you hear?
Yes, sir. You are audible, sir.
So thank you very much for giving me an opportunity to chair the important session, integrated plant health management. At the outset, all of you know very well, we have a 30 to 35% of the crop loss is being occurred in this country, which is causing a tune of 60 million tons per year, which is equivalent to 1,40,000 crores. If you could be able to manage 50% of these diseases, you could be able to save the around 60,000 million rupees. So, though the centuries have kept the loss, but it has got an importance in the disease management. I don't want to take much time here. Already Dr. Dinesh Singh has presented how to manage the bacterial diseases. But here, I would like to make a few comments on his presentation. But he made excellent presentation. But thing is that while recommending for management of bacterial diseases, we should be very careful. Better to avoid bleaching powder and antibiotics. This is what my personal feel. And we can promote a grafting, particularly that soil-borne bacterial diseases, and we can give more emphasis on biocontrol agents. This is very important. Plus botanicals and biofumigation. So with this, I compliment the Dr. Dinesh Singh, who has made an excellent presentation and given a overview of the bacterial disease in this country. Now, I request Dr. Vinayak Hegde. I think Dr. Prabhakar is not here, no? Dr. Prabhakar
No,
is
sir.
available?
No, sir. He will not be presenting, sir.
Okay, now I request Dr. Vinayak Hegde. And Dr. Vinayak Hegde is head of the division at CBCRA Kasaragod. And he was working on classical class. Now we request Dr. Vinayak Hegde to take presentation.
Good afternoon. Am I audible? Sir, am I audible?
Yeah, audible, audible.
Audible?
Yeah.
Okay. I will try to share my good afternoon.
Not yet, sir. Not yet. It's not showing. Your slides are not visible yet, sir. No, sir. Still, it's not visible. So start sharing, sir. Yes, sir. It is visible.
Yeah. Is it visible?
Sir,
No?
please put it on. Yes, sir, it's visible. Please put it onto slideshow mode and proceed, sir.
Okay. Good afternoon, everybody. Respected Chairman Chodappa sir and co-chairmen and organizers. I first of all thank organizer Dr. Devappa for providing me this opportunity to share my views and present on the coconut disease and arecanut diseases. I think this is the first presentation on coconut and arecanut in this conference probably. So I will just give the disease management and arecanut using the technologies, advanced technologies. In that, my presentation will cover mainly on coconut and arecanut scenario and status of major diseases and emerging diseases and diversity analysis of pathogens and diagnosis and challenges or issues in managing these diseases and need for advanced technologies to address the issues and conclusion. So with this, go one by one. Compared to coconut and arecanut, as we all know, these are the more than around 100 countries coconut is grown, and arecanut is also grown, and it is mainly have India is the largest producer and consumer of these crops. These produce provided livelihood security to millions of people in Asia and Pacific regions, and it has got cultural and traditional importance also. Mainly cultivated by small and marginal farmers, increasing demand as health things. You know that coconut now it is increasing, and if you see the scenario areas coconut, it is grown in most of the states. As you can see here. And presently, around we have got area of around cultivated 16 states, and mainly 90% of the area and production comes from 4 states: Kerala, Karnataka, and Andhra Pradesh. And this is the latest as per the Coconut Development Board report. And you can see the high variability in the productivity of the coconut and though area is more in Karnataka and even Kerala, the productivity is comparatively less compared to Andhra Pradesh as well as in Tamil Nadu. Then regarding betel nut, again, it is an cash crop, you can say. It is distributed in southwest and northeastern parts of India, and now it has got economic backbone for more than 16 million people, and largest producer and consumer again, India. So these are the states where now arecanut is being cultivated in spite of a lot of problems you are maybe knowing even in the news as well as frequently coming on about these crops, diseases, loss as well as pest attack. So in spite of that, we are the leading in the production of arecanut. Coming to the diseases as issue in arecanut and coconut, mainly diseases of coconut, about 173 fungi, bacteria, and viruses and phytoplasma are reported to cause the disease in both these crops. These are the three major pathogens which are known to cause the disease. Phytophthora, phytoplasma, and Ganoderma diseases are the major importance. And both the crops, the diseases are the almost similar. And emerging and re-emerging pathogens, I can say Colletotrichum and Laccidiplodia. These are the two pathogens. Diseases caused by these two pathogens are re-emerging in these crops. In case of coconut, if you see that bud rot is Phytophthora palmivora. It is killing around 5% of the coconut trees every year in the endemic area, high rainfall area. Weather stem rot is another major disease, fungal disease caused by Ganoderma. So important disease, aggravated especially in the drought conditions. It is again more severe. So then root wilt disease, of course, is a disease of more than, as you all know. Still, it is of course, though no much spread, it is spread to adjoining districts. So it is a little bit concern of preventing the spread of this disease to other areas. Then, of course, leaf blight. Leaf blight is another Laccidiplodia. It is an emerging disease, especially in the Pollachi area, here the coconut hub. That is a major problem. And of course, stem bleeding is a common in most of the places. It can be manageable disease. In case of arecanut, again, there is a lot of Phytophthora, the major problem, as I told. Phytophthora disease, about 30 to 40% every year, there will be loss, and maybe 100% loss individual palm wise if you take many times. And bud rot and fold rot, the same pathogen, leads to a lot of problems. And inflorescence dieback and Colletotrichum leaf blight are again emerging and creating problems. Local stem rot as Ganoderma, and yellow leaf disease, which is also almost old now, is also a problem.
I think your mobile also.
Okay.
Switch
Mobile is off on
mobile. Yes, you can proceed, sir.
So please proceed, sir.
Yeah. I will just go through one by one. Phytophthora disease is coconut bud rot. It is a major problem, and it has been Phytophthora palmivora, and palms of all ages are susceptible, as I told. It is a disease spread from wind-blown rains and slug. This is known to disperse the pathogen from ground to the top of the palm. I think some problem in the. Hello?
Sir, you are audible, sir. You can proceed, sir.
Okay. This is with this Dioceous slug is another importance. It has been long known. Then fruit rot or mahali, again Phytophthora disease. This is It is a major problem in Arakanat, where our 10 to 90% loss has been reported. And 2018 also, we have taken survey. You can see here, even in Chikmagalur, around 52% loss.
Sir, I think there are two machines, computer and mobile here.
Okay.
Please switch up your speaker. There is voice is echo.
Oh, Dr. Sankar, I think other than speaker, all the people should be in mute, except the speaker.
Sir, done. That was done.
Okay. I think I have closed all my other gadgets. Still, I think some problem. This is the fruit rot severe. You must have seen so much severe dropping. Then the major reasons, higher incidence of Phytophthora diseases, mainly due to difficulty in taking up timely prophylactic measures. Actually, as you know, this is the problem of skilled climbers required and the monsoon season, and we are not able to take the timely prophylactic measures. Climatic changes, erratic rainfall, and non-adoption of scientific cultivation, as well as improper inadequate fungicide use, has led to the increased incidence of disease over the years.
Please echo.
Sir, this is the problem with the speaker.
There is some problem with the kindly check it out.
One minute, sir. 1 minute. Hello?
Yeah.
Echo is there. Check it out.
Dr. Vinay, today, if you get to the live streaming should not happen.
Dr. Hegde is using live.
Sir, Hegde sir, please unmute yourself.
Now we can start. Dr. Hegde.
Okay.
You can start now.
Yeah.
There is some problem.
Yeah, till echo is coming, sir.
The echo is coming.
Post site, absolutely there is no problem.
Hello.
In the host has to check. Host and the co-host should check that live streaming may be in their computer, not in Vinay Hegde's computer. Host and the co-host should check.
Sir, host and co-host, there is no problem. That is the problem with the speaker.
Oh, 1 minute.
Where is the problem?
Sir, maybe he has switched on both YouTube and Alexa.
Ask the speaker to disconnect and connect again.
Dr. Hegde, you disconnect and reconnect once again. Somebody is suggesting. Yeah, you disconnect and reconnect.
No, this is Ranganath. For me, my presentation also happened same thing. I did disconnected and again connected. That's why, sir.
Yeah. He is doing.
Now there is no echo, sir. Please proceed.
Dr. Hegde, now you proceed.
Okay, I will share.
Now you start.
Okay, Vinayak sir, please start. Vinayak Hegde.
Okay, I'll start. sharing because I stopped sharing. Yeah, please start sharing. Hello. Is not visible to them still? It's,
No, the slides are not visible.
yeah, it is loaded. Just it is still saying.
Yeah, you start now. Start now.
It is coming. I think it will come.
Stop sharing and again.
You're starting. It is there now. Stop sharing. No, that's what I don't know. Yeah, stop means again it will take. Again it will go here.
Yes.
This one.
Now we can share it.
Yeah. Yes, I am sharing now.
Yes.
I think it is visible now. Okay.
Yes.
I will go ahead as I was. I will go to continue as Phytophthora. I was there in Arecanut. I told more than 50%. Even 2018, we have recorded many much loss, about 50% loss in overall in certain districts. So it is the severe disease where we have characterized the different this Phytophthora medae, which is collected from different places, and we use different genes. This aspect also we have done. This is the Phytophthora species associated with Arecanut fruit rot and rubber abnormal lippy. We have seen, collected, and try to check with the different primers and amplified and checked. All lion Phytophthora isolates associated with rubber abnormal leaf fall also has been P. medae, and Phytophthora isolates 23 identified as P. medae, and 2 we got Phytophthora heavy also in Arecanut. And this is the regarding management. If you come again, there is a integrated management has been recommended, as well as for as we all know, prophylactic measures are the major importance in case of Phytophthora disease. So even in case of bud rot, we are using for this crown cleaning and phytosanitation and Bordeaux mixture spray to the crown, as well as even trichoderma, which has been developed here in CPCI earlier. So we are evaluating, and it has found to be good in some of the cases to reduce the disease incidence in case of bud rot incidence in Phytophthora, due to Phytophthora. In case of management of fruit rot of Arecanut, as it has been done for the many years, it is not now Bordeaux mixture, as we are told that 2%, whatever the chemicals or fungicides earlier screened, Bordeaux mixture was found to be still holds good, and many farmers are using Bordeaux mixture spray now. And there was earlier Dr. Chaudappa sir has only developed this. You must have seen that poly covering of the bunches was really very used to be good, but later due to skilled required and climbers problem, and this has been slowly discontinued now. But farmers are depending only on spraying with the Bordeaux mixtures, and they were asking for Bordeaux mixture alternative. So we did from 2015 to 2017, around 12 fungicides, new fungicides of which are specific to oomycetes has been screened, and we found that under that one fungicide, though there are many oomycete specific fungicides, only one fungicide, which is mandipropamid, was almost giving good control like Bordeaux mixture for this Phytophthora, in reducing the Phytophthora disease. So mandipropamid 0.5%. Then we continued again to see in the larger area, not in the experimental plot, only with comparing with the Bordeaux mixture. Again, we have seen that 2 years, next 2 years, 17, 18, 19 also, we saw that mandiprobamid sprayed plots, there is a less incidence, even 1% Bodo mixture. Though there is not much significant difference, but there was definitely a reduction in the incidence. Mandiprobamid was found good. So this even the residue level with the IHR Bengaluru, Dr. Chodappa sir's suggestion only, we tried to get it done. Though we found that up to 60 days, this is this residue. This is a semi-systemic fungicide. We have found that it is in the kernel as well as in the rind of the arecanut peel. It was there up to 60 days. It could be detected. We have this was done from the IHR's help. Then difficulties in managing Phytophthora disease overall, if you see, the timely application of prophylactic measures, as I told, that is the one of the major difficulties, though this is very important, and lack of suitable mechanized devices. As you know, most of the Phytophthora disease, we have to go for prophylactic spraying, as it is a very difficult in the crops, both in arecanut as well as coconut, during the monsoon season to climb as well as spray. So that is the one of the major problem. Then cleansing erratic rainfall, as you know, nowadays suddenly certain years there will be erratic rainfall. This year, even now, it has started, though there was no much Phytophthora disease earlier during July. Now suddenly there are a lot of rain, though much Phytophthora is not there, but some gardens there are sudden dropping of nuts have been observed. Then early diagnosis and monitoring, as well as lack of resistant varieties. There is no, of course, arecanut, though we have seen that other areca species, Areca consina, was found to be resistant to Phytophthora. So, but most of the cultivated varieties, Areca catechu species, all are susceptible. Then lack of reliable forecasting models, though we tried earlier also, people have, scientists have tried here to best whether we can develop a forecasting model at least within 10 to 15 days in advance, because it depends on rain as well as sunshine hours. So it could not be, there is no proper models like potato. We cannot forecast only rainfall. Based on the rainfall, we could able to tell whether there will be this will come or not. So address these issues with, of course, Dr. Chodappa sir knows he himself, his own guidance. It was developed at CPCRI. We wanted to go for with the auspice, this one. A sprayer was designed at CPCRI, tractor mounted blast sprayer to both in coconut and this was arecanut. We tried for evaluated, but we felt there is a need for some sort of modification. Now our engineer has made little bit modification of this, so it should go directly to the bunches, especially. So we are trying that, as well as we are trying to go for unmanned aerial vehicle. Again, Dr. Chodappa sir's, I must thank. He only has introduced this and he asked us to start with. So we have initiated this work. Now we have taken up the project on disease surveillance also. I will come out for that later. This is in arecanut as well as coconut. We used this for spraying the Bordeaux mixture to check whether we can able to prevent. So, but though the results for 1 year screening was not very use this one, we request there is even the General Aeronautics, which we have collaborated for the drone. They themselves felt that there is a request for, there is a need for modification of the sprayer, especially for arecanut, where we have to target mainly the bunches. Otherwise, it will be a problem. So they are also working out this. So hopefully in coming years, we may have to do this, because there is in coming years, there is no climbing will be a problem, and arecanut or coconut, we need to go for this unmanned aerial vehicle based application with the safety of keeping in mind of the environment. That's what I wanted to say for Phytophthora disease. Then another major, this is Ganoderma wilt or basal stem rot. This is again a major disease in the many areas in Ganoderma. As I told, it is a integrated management has been recommended. I think many of you who are pathologists are knowing about these things, and we tried, but still in certain areas, this biocontrol where we are telling that Trichoderma hergianum, which has found to be very good in some areas, but in some dry areas, again, this is the creating problem. It is not able to survive. So we are looking for some of the trichoderma isolates which can be used in the drier tracts for managing. Even in arecanut, again the same problem. Arecanut also, Ganoderma is a major problem. It is also in the northeastern region, both arecanut and coconut. It is one of the major issue of in the arecanut. So this is the type of severe symptoms which we have noticed. And we tried to another major issue in Ganoderma diseases. By the time farmers or we ourselves have to notice the field level symptom, already damage has been caused to root as well as the pathogen has been caused the infection in the basal portion. So we are not able to, so that's why we tried to go for LAMP assay. PCR of course has been done for Ganoderma identification, but nothing was at the field level. We are not able to diagnose. So we felt LAMP could be one of the technique where we can take for field level diagnosis. So we initiated this work and we could able to do it at the laboratory level. We are trying to make it as trying to find out whether we can use it for the field level diagnosis using this LAMP reader. We have a LAMP reader. To that, we can able to do it within half an hour. We can in the field itself, we can carry that and we can able to detect it. That's what we are trying to do. As well as also we are trying to do with the this one, the LAMP assay we have done. But again, this is also in the field level. We have to check whether we can able to detect. So then this is again another important disease is root wilt of disease of phytoplasma diseases. There are root wilt disease, Tatipaka disease, yellow decline in different this one as well as Sri Lanka. It is Veligama wilt we call. In coconut root wilt, it is now known to be the phytoplasma 16SR 11th B group, whereas now Tatipaka is not at all there now. It was there earlier, but now even I have not seen anywhere Tatipaka disease. So then the root wilt disease, as we have known that it has been identified as a phytoplasma and the vector has been identified as protista as well as. And this is one of the concern is that it is slowly spreading to Tamil Nadu area. That is the only it was restricted in, of course, in the Kerala up to 8 districts, though it was a little bit in the other district, but not a major problem at all. Whereas in the Tamil Nadu, which is the adjoining district of Kerala, slowly it is preventing. So that action has been taken. Of course, CPCRI is trying to, with the CDB as well as TNAU, the efforts have been made continuously to prevent the spread by creating awareness as well as removing the infected farm immediately from the border area. So this is the yellow leaf disease of arecanut is another major problem. As I told, the same phytoplasma again in the coconut also. This is again a problem especially in Kerala as well as in Karnataka, major areca growing areas in Chikmagalur and Dakshina Kannada Sulya area. These many hectares have been affected and farmers are always asking what is the solution for this major disease. So dissection of RWD, again, we are trying this, this has been done, PCR technique has been developed for detecting, but Dr. Manimekala has developed this one earlier for both for root wilt as well as for arecanut yellow leaf disease. This PCR based real time as well as these techniques are available. It is already published. But the problem again, this is there is no consistency in the detection of this at the from these protocols. That was the problem for us. Even the universal protocol as well as the species, the latest protocol developed for this will not able to detect this disease. That's what we are working out now. I am taking the help of Dr. Selvarajan, whether we can go for nanopore sequencing to find out what is the difficulty in detecting through normal PCR based process, this process. Hopefully Dr. Selva has got now nanopore sequencer. So he told he will help in find out what could be we can do for identifying. Of course, we have tried to find out the weed host where in the coconut plantation where it could be whether it is a host for phytoplasma in coconut. The weed host, all we could able to detect. These are the weeds which we have detected, most of the weeds, and this could be easily using the universal P1, P6 primer as well as this we could able to detect, as well as these most of these are belong to different groups of phytoplasma, not to the coconut which has been reported in the coconut is phytoplasma. And of course, apart from this one, as phytoplasmal disease, as the farmers will be asking for the management. So CPCRI has done several years management works as well as based on the, as we all know, phytoplasma disease, either there is no cure, we have to go for managing the disease with other agronomic as well as nutrient status of the soil to increase the life of the palm. So this is what has been done, and Dr. Chodappa only initiated as a frontline demonstration in these of the districts of this all, and it has been very successful. And Dr. Krishna Kumar, at the time we were introduced, and it has become very successful in reducing the disease, and farmers are very happily, they were adopting these techniques to increase the yield level in the affected area. Of course, there are CPCRI identified 3 varieties like resistant to root wilt. So this is also being trying to popularize these varieties in the newly established garden as well as in the root wilt affected area. So then challenges, again, to set, having said all these things, again, there are the challenges in case of root wilt is, as I told, detection is the major problem. Still, we are not able to make it as a consistent as well as a reliable field level detection techniques. Selection of tissue samples, again, as you told, sometimes we are getting selection from the bud region or very young plants, sometimes in the root. But universally, we cannot say that you take only this region, we will get the detection. That is the problem. So we have to solve it. This is what I am telling, that there is a need for this one. Then other minor diseases, as we all know, in the most of the leaf affected palms, this is the major problem, is leaf rot. All the leaves will be, nothing will be there. You can see that this is all mainly, there are different fungal species because it becomes susceptible to fungus, and most of the fungus cause the leaf rot. So one of the management is to control this leaf rot, at least save the palm to a certain extent. Otherwise, this leaf rot will cause complete reduction in the yield. So stem bleeding, of course, it is a common in most of the places. It can be managed. There is no issue. But whereas this leaf blight has become a severe in some of the places in Pathology, where the coconut is the major area, as you have seen, that it was reported earlier, long back in 1984 itself. This fungus has been reported on coconut as well as disease has been reported. But this has emerged in this area last few years in this coconut belt of Pathology area, and other area also we are seeing, but the severity is more in this area. So this has been done as well as through AACRP center. The management trials have been done, as it has been recommended to go for root with hexaconazole. So of pathogenous leaf spot or blight disease, as I told, Placidiploria, we have characterized this fungus also using molecular this one, especially ITS region as well as other genes. So it has been identified morphologically also. And other minor disease is grey leaf spot. Though it is a minor disease, some pockets, especially in the nurseries and other places, it is becoming severe. So we have isolated and characterized this also. It is a Pestalozziopsis palmarum, now it is called. Earlier Pestalozziea we used to call. Then another major, this one noticed was a lethal wilt-like disease in certain parts of Tanjavur area. It was in 2014 onwards, they were noticed. Though there was a report by Dr. Bhaskaran from Tamil Nadu Agriculture University. He was in ASRP of our palms. So he reported that there was a new disease type in 2007, but it was not a spreading. There was very few, this one only. But in 2016 onwards, there was severe incidents, especially some in few palms were going regularly. Suddenly, it used to be within a week's time, they used to see this much dropping of nut and yellowing as well as this one. So, with the help of CPCRI Vipangulum as well as CPCRI tried to work out what is this. So, when we checked, it was these are the two blocks where we are checking, mainly the lethal wilt was observed. So, then detection of Phytoplasma in lethal wilt disease, we could able to detect, and it was found that Candidate Phytoplasma asterix, for the first time we have checked, this could be easily detected compared to root wilt. We are not able to detect here, but using this primer set, we can easily detect out of, you can see here 60 palms, this we can able to 59 palms, we could able to get positive identification easily. And even the this where we have used is again the spindle leaves as well as from the spear leaves as well as inflorescence actually we can able to detect it. So, but it is not it is first time that it is belonging to SR was 16 SR B group 1. This is not reported in coconut. Even in this lethal yellows which has reported is 16 SR4 group. So, but we have recorded this as a belong to the first group. So that has to be, of course, this disease we have taken this one to reduce from there itself, to taken all the precautions to destroy the area. It should not go to other places. So all meetings was held as well as even the concerned people have been informed. So it has been for the first time it was reported. Now we have to see whether that will be spreading or restricted. So inflorescence dieback in areca it was reported. It's also recent years again we are seeing that loss of up to 60% due to this disease has been seen in certain districts of Karnataka. In Kerala also, this we can see here this much drying of the inflorescence. So this is again we are asking. We tried for different fungicides for this because earlier Mancozeb used to recommend for this as well as Mancozeb plus carbendazim. This was also working, but again we tried different fungicides and propiconazole was shown to be good. Now we can try. We have 3 years trial. We could able to get that. Then leaf blight disease. This is the another disease. Who are probably in last few days you must have seen in TV also even in the Kannada newspapers. A lot of news is there, especially in the Kalasa and Chikmagalur area. Around 150 acres they are telling completely it has been. We noticed in 2019 we visited and seen that this is the major problem like this complete drying of the leaves. When we even in the same thing in the Tripura in 2016, we visited and checked that it is due to Coletotrichum. We could able to isolate the pathogen from Coletotrichum from this. Then we try to see whether all are same as inflorescence dieback pathogen only. It is causing same one, same type of blight. So we went for multi-locus sequencing of this also, these isolates. So based on our results, we could able to find that this out of 12 Coletotrichum isolates from leaf spot and blight disease, 5 were identified as fructicola, as one as the karsti, Coletotrichum karsti, based on the Karnataka and Kerala, they were listed. Whereas all the isolates we got from Tripura was identified as Coletotrichum siamis. So this has to be further we are working on more isolates we are collected now from the Kalasa and other area. We would like to check it whether there is any difference in the different species level complexity in the this Coletotrichum emergence. So this is the only Coletotrichum iotiora is associated with inflorescence dieback. As we know, inflorescence dieback is there in most of the places in Dakshina Kannada as well as in Kasaragod area also, but this is not causing that type of leaf blight. Is not noticed here in this area. Leaf blight is noticed only in certain pockets where suddenly that too as an outbreak. So this probably there may be different species as we told. These are the species which we have noted. This we are continuing with the recent outbreak again, and we have to see whether they are similar or not. Another emerging disease is the bacterial stripe. This is especially in the newer area of where the areca nut, now most of the districts are going for areca nut cultivation. So there, especially in the other area where earlier maize and these crops were there, now they are going for areca nut. Those area we are seeing the bacterial severe bacterial stripe disease, which is you can see here the undersurface where the bacterial oozes are coming out. So bacterial disease was noticed earlier in areca nut, reported long back in 70s, especially in the Tumkur area. But that was reported like Xanthomonas. And after that, there was no much incidence has been reported. Only in that area, few area it was located. But in the recent areas which we are seeing, this is all reported in the Shimoga area where we are seeing this type of this is the 2 places where we have collected all the samples where it is a major problem is emerged, and we isolated this one, and we felt that petiole as well as from the leaves, all the isolates we characterized, and we found that this is based on our analysis with the 16S RNA we found that it is Pantoia species. It is earlier, it was Xanthomonas based on the morphological identification in 1970s. Aragonite, it was reported. This we are now getting as Pantoia species. We suspect probably in the new area where they are growing maize as well as other crops. They are intercropped also with Aragonite. So we don't know how whether it has Aragonite it has come up. So that has to be worked out. We don't know. But we are getting mainly Pantoia species from this. And we have proved the pathogenicity also with the same isolate. So this is the pest and disease surveillance. As I told, another important area in Aragonite, which our coconut required is go for monitoring of the disease using UAV. And so we have taken up this work and able to, what our objective was to develop a surveillance system so that if we can fly the drone, we should be able to get for whole area, one district or 10,000 hectares or whatever maybe. At a time, we should be able to get what are the diseases prevailing in that area in the through using machine learning algorithm as well as ICT tools. We should be able to get it in our mobile. That is our idea. But now, as of now, we have started with rhinoceros beetle damage, which are easy to locate from the even RCB images. So we tried, and machine learning algorithm was developed, and we could able to make it up to 90% accuracy. We have developed this one. We are trying to validate because of this corona. We could not validate that under field condition. We are working out same way for root wilt also. We have taken the pictures from our Triangulum center. This is all our center of this one images, drone images. So you can see here we have developed this is the GPS tagging will be there. We can able to tell how many palms have been infected, how many are healthy, how many are showing the symptoms of what type of symptoms it is showing. Based on that, and it should be tagging will be there. We can see which area, which palm has been affected. So with that, we can able to go for spot level delivery of the pesticide or fungicide for particular palm. That is our objective, ultimate objective. Once we standardize this, we want to go for the spot level delivery of bioagents or fungicide for particular to the crown, where we can able to apply. Otherwise, there is, as I told, there is a difficulty nowadays farmers are facing. Whatever the crop protection, we are integrated management or apply the fungicide, they are facing the difficulties. I think unless this type of things comes, it will be, it is the future. I think we have to go for this. That's what our idea. And Dr. Chodappa sir only initiated this one and supported us. So we started this work. Hopefully, sir, we will be able to come up with another 1 year. We will be able to come up with some sort of system where we can able to go for at least survey system. We will be able to perfect it so that we can able to detect the diagnose the infected palm in a larger area, and then we can suggest for particular control measures for that one. This I wanted to tell in brief about. This is not my work alone. This is overall our institute, which has been done last few years, even many years, most of the diseases. So what I concluding, I would like to say that there is a sensitive and specific molecular detection are available. But there is no field level diagnosis, still a major problem. So we need to go for field level diagnosis, which is very essential. And as even for the root wilt and yellow disease, it is a major problem. We are working it out. As I told, we need to go for other molecular techniques and how best we can able to make it a reliable detection. Otherwise, unless we go for a reliable and a consistent detection technique, we cannot move forward. Of course, management trials for YLD as well as RWD using different types of, as I told, even for YLD, now we have felt that mulching of the films, I am not showing that the trial is going on. We felt that mulching will help in reducing that leaching of nutrients, as Chodappa sir himself suggested this one. So we tried, sir, that has given very good result. Dr. Bhavishya from Vittal has taken up trials many places. So hopefully in another year we will come up with that package where we can able to reduce the incidence of the severity of YLD. Then I feel the sensor-based diagnostics with the artificial intelligence and UAV, which the ICR is telling that we have to go for. I think plantation crops are the first one, coconut and arecanut, which where we need immediate these aspects, because that these are the major areas which is facing the problems. I think this has been the initiative with the surveillance system. I hope we will be able to go ahead with further to develop this aspect, and mechanized sprayers as the farmers are demanding, and lot types of farmers themselves are making, but still there is no mechanized way to from the ground level we can apply to the farms these biocontrol agents as well as this fungicides. So I think these are some of the issues which I considered as to be taken up. So with this I conclude this one. I thank all for your patience hearing whatever the disturbance in my audio for this one. I apologize for that. So then I thank Chodappa sir for all his encouragement during his tenure here. He helped us to work on this aspect. And if I have missed, I think he will be able to supplement the work which I have missed here, as well as the what the suggestions required for future. He will be able to elaborate a little bit more, I hope. And I thank one and all for your patience hearing. Thank you.
Thank you.
Thank you, Dr. Egde.
Thank you, sir.
You have made an excellent presentation, and you have covered all the aspects of the plantation crops, starting from the existing diseases and emerging diseases. And actually, recently we have heard also regarding bacterial blight and cultivar lichen, that also you covered. And you also covered a lethal laying of coconut in Tamil Nadu. So, Dr. Egde is known for best presentation always. Today also he made an excellent presentation. Though initially there is some hiccup is there in the electronic system, I compliment him. Now you can take one or two questions because time also running fast. If any one or two questions can be taken.
Sir, excuse me, sir. Can I talk, sir?
Yeah, please.
Sir, you said there is a fungicide persistence for 60 days, no, sir. Can you actually we worked in ACR cases, they used we used potassium phosphonate. Can it be used as alternative because all these are phytophthora diseases? Because foliar application we went for.
I could not hear some. What is the question?
Hello.
Hello.
Sir. Fungicide for 60 days, no, sir. Can we use potassium phosphonate, sir? Because we worked in ACR, sir. They use the foliar application with potassium phosphonate and soil application with Trichoderma hergianum.
Yes, sir. Potassium phosphonate is known. I am not saying that potassium phosphonate many of the tree crops also they are using, but as of now, I think even in the CIB, potassium phosphonate has not been registered as a fungicide or even in the agriculture use. I don't know, but it is being used by many companies or many people as a tonic, plant tonic. They are being circulated, but potassium phosphonate has been tested even in pepper and even in we have also in the in vitro all we have checked even for these things, and even farmers field also. And it is not very this one, but definitely there is some way that is control is there, but not since it is not recommended as such, we didn't continue much on this work.
So what about
actually, Dr. Shesha?
Yes, sir.
We have better fungicides for management of phytophthora compared to the potassium phosphonate. So that's why potassium phosphate is not much effective. So if you don't have any further questions, we can call upon next speaker, Dr. Nidhi.
Yes, sir, I am here. Sir, am I audible?
Dr. Nidhi.
Sir, am I audible?
You are audible.
Okay, sir, I am sharing the screen.
Yeah. Just I will make a small introduction. Dr. Nidhi will be making a presentation on a novel and eco-friendly solution for bull eye pathogen infecting a Lycobriskin esculentum tomato. So we request Dr. Nidhi to make presentation. Dr. Nidhi?
Yes, sir, I'm trying. It's connecting.
Yeah, you can make presentation now. Dr. Nidhi,
Yes, sir.
please load your slides.
Yes, sir, I'm sharing.
Please, please. Because time is also running.
Sir, is it?
Can I help you, Nidhi? Dr. Nidhi, can I help you?
Yes, yes, ma'am. Please see. Is it visible on the screen?
No, it is not visible. It is not sharing the screen also. Can you please click on the icon next to the video?
Yes, ma'am. I've already clicked and it's showing connecting.
Okay. Is your PPT open?
Should I mail you the presentation, ma'am?
No. You just open your PPT presentation and then start sharing. Maybe you have poor connectivity.
Dr. Nidhi, any problem?
Yes, sir. It's showing connecting only. Maybe some network issue is there.
I don't know. It is not displaying here. Now it's coming.
Yes, sir. It's not coming. You are sharing your screen.
Yeah. Now it has come. Now it's make presentation.
Okay.
Now it has come now. You can mode into the presentation mode.
Dr. Nidhi?
Ma'am, I'm not able to see the screen actually.
We are able to see the screen.
No, we could able to see your slides and make it a presentation format. Okay.
Is it right now? Yeah.
Yeah. It is being seen.
Okay, sir.
And you make it to the presentation form.
Good afternoon, everyone. The topic of my presentation is a novel
Dr.
and
Nidhi?
eco-friendly remedy.
Dr.
Yes,
Nidhi?
ma'am. Yes, ma'am.
Kindly put the slides onto the slideshow mode, please.
Ma'am, I already did.
Okay. Proceed.
Okay, you can proceed.
Okay, ma'am. Okay, sir.
Please proceed. Now, if possible, make it a presentation mode. Otherwise, you can proceed.
Okay, sir.
Please
the
tell
topic of
me.
my presentation is a novel and eco-friendly solution and remedy for bullseye pathogen infecting tomato. And our motto is lab to land and eco-friendly approach. That is, the research we are doing in labs should reach to end users, that is farmers, at a low cost and in a more effective manner. So the injudicious and wide use of fungicides has led to serious environmental problems besides affecting health of users and consumers. We already know this. And so it is necessary to minimize the use of agrochemicals for controlling plant diseases. And bioformulations definitely are a potential alternative to currently used agrochemicals. Tomato is our target crop, which is known as superfood, and have been on fire for last few years due to price hike, and one of the reasons being pests have become immune to general pesticides and insecticides. And during our survey in and around Faridabad in Haryana, it was seen that 40 to 80% losses of tomato crop was due to early blight caused by Alternaria solanae, that is bullseye pathogen. So our objective is to develop an IDM strategy or bioformulation to be distributed to farmers ready to use. First of all, a detailed survey and surveillance was undertaken in the year 2012-13 and in and around Faridabad, Vallabhgarh and many villages with heavy losses due to Alternaria blight. And the data was collected for 20 villages. This you can see, this is the survey report where person PDI was calculated, and then the AUDPC was also calculated for different genotypes. Now we all know that microbial resources are the biocurrency for next generation. So,
Dr. Nidhi, there is a small issue there. Your slides are not moving. We change the slides.
okay, sir.
Yeah.
Dr. Nidhi?
Yes, ma'am.
Kindly re-upload the slides. End the presentation and try to re-upload.
Okay, ma'am. I'll stop sharing and then again.
Dr. Nidhi, kindly unmute yourself.
Not able to share it, ma'am, now. I don't know what is the problem here. Should I mail you, ma'am? Because yesterday also there was a problem with the speaker, so...
Sir, Chaudappa sir, can you please unmute yourself?
Yeah.
Sir, she is unable to see.
No, otherwise we'll ask her, let her make a presentation, big presentation. So no, if she is unable to upload the slides, Dr. Nidhi,
Yes, sir.
you can make presentation.
Sir, or can she mail us the PPT so that we'll present and then continue with the next?
No, sure can be it because time is there. We have no objection. Let her mail it. It may take some time, you know.
Sir, it's sharing now again.
Please share it.
Sir, yesterday also there was some problem and it was like we made the PPT and it was smoothly running then.
Yeah, now what you do, now you share it now. Now, could you able to share it, Dr. Nidhi?
Yes, sir, it's sharing, it's connecting.
It's connecting? Okay, please.
Yes. Please allow me to mail it.
We will do one thing. We will ask
Yes, sir.
next presentation somebody is there.
Right, sir. Right.
But by the time you can resolve your issue. That is better, no?
Right,
Huh?
sir. Thank you so much. Thank you so much,
Ah.
sir.
Now we can request Mr. Satyaprakash. Mr. Satyaprakash is available?
Yes, sir. Yes, sir. Yes, sir.
You can make presentation.
Yes.
By the time she will be ready.
Okay. Yes, sir. Okay, sir. Sure, sir.
Please carry on. So Mr. Satyaprakash will be presenting on assessment of bacterial wilt resistant Solanum rootstocks for yield and yield attributing traits of grafted eggplants under acidic under laterite acidic soils. Dr. Satyaprakash.
Yes, sir. Yes, sir.
Please be it.
Yes. Wait, sir. Yes. Hearing. Sir, sir, the screen is visible.
No, it is coming actually starting a share to content. They are not visible.
Okay.
Please share it, huh?
Yes.
Yeah. Now you can make it to the presentation mode. Hello? You can convert into the presentation mode. Dr. Satyaprakash. Yeah, okay. You carry on. Make presentation now. Yeah, it's clear now. Yeah, you can make presentation.
Okay. Yes. First of all, sir, actually, somebody is from the commerce background.
Satyaprakash. Slides
Sir,
are clear now. You can please make presentation.
Yeah.
Yeah, time is running out.
Thank you for giving this opportunity. Yes, sir. Giving this opportunity to present my PhD research work. My topic is we are going to discuss about assessment of bacterial resistant Solanum rootstocks for yield and yield between traits of grafted eggplants under laterite acidic soil. Now, in coastal region of area from where I belong, near Odisha and West Bengal, these regions, the crops, especially the solanaceous crops, are very much prone to bacterial disease caused by Ralstonia solanacearum due to due to prevalence of a very high temperature and high humidity. So, eggplant suffers nearly 50 to 100% loss due to this particular disease. Now, this pathogen, as we know, enters to the plant through either secondary root infection or primary wounds, and it colonizes in the vascular system of the plant, so that the plant is unable to take nutrient and water from the root to the whole plant zone, and so ultimately the wilting and death of the plant occurs. So to mitigate this disease, several attempts have been made to mitigate this disease, like crop rotation and chemical management, biological approach, and host plant resistance. So, but it still had some major drawbacks due to which the control has not been a total success yet. So first of all, in any soil-borne disease, to control any soil-borne disease program, so first attempt is generally preferred for crop rotation, in which we generally take alternate host, so that it won't affect the our regular susceptible host program. Susceptible host. So, but the Ralstonia solanacearum generally stays in the field for more than 3 to 5 years in dormant condition, and it has a special survival skill which called as VBNC, the viable but not culturable state, in which its metabolic rate is very decreased, so that it remained in the dormant stage. So for 4 5 years it will remain, so that crop rotation has not been a success till yet. Then second, when you approach for chemical management of that one, this particular disease, due to localization of bacteria inside the xylem vessel, it enters to the plant through the root portion, and then it colonizes inside the vascular stage. So exogenous application of chemicals like antibiotics are not very effective in controlling this disease. Then after eliminating chemical management, when you come to biological control in the use of by use of Ralstonia piqueti or etc. So that user friendly formulation preparation is a major problem, especially when it comes to that grassroots level, when farmers generally go for a biological control. That particular formulation preparation has been a major constraint. So commercial acceptance has not been yet done. Then when all these things fail, then scientists go for host plant resistance. Generally, if we produce a directly resistant that particular bacterial resistant variety, then the farmers to go for chemical treatment or biological treatment or any cultural applications. So, but two major problem is that one is linkage drag. There are a lot of bacterial resistant sources are available, but incorporation of that bacterial resistance to that commercially accepted genotype is a major problem due to linkage drag. Along with the desirable trait, along with bacterial resistance, a lot of undesirable traits have been also been transported. So that's not desirable. And second one is
Slides are not moving.
Here it's moving.
He is not moving here. Please check it up. Slides are not moving.
Now in 5 slides are running.
Okay. Carry on.
Okay. Then we go for that. Then another problem in development of resistance and population is that acceptance of that particular bacterial resistant variety. Now, brinjal or eggplant is a very much region specific acceptance. When we go for eastern region like Odisha and West Bengal condition, generally consumer prefer round type and large and round type of brinjal. But when you go to that South India areas, then generally the long type brinjal are very much preferred due to in preparation of sambar and etc. So when even though there are a lot of varieties available public domain. Disease resistant varieties are available, but they are not commercially they are not accepted by the consumers. So so ultimately the grafting has been gaining a recently grafting has been getting a lot of attention to combat this disease. Generally in India condition now, VNR company is the major producer of grafted brinjal, producing a lot of grafted eggplant and distribution among the farmers. However, they are using Solanum taubum as their bacterial resistant rootstock. But Solanum rootstock has a lot of problem regarding their germination issues and growth regulation. First one is generally Solanum taubum seeds only take more than 1 month for germination, and again that very germination is very non-uniform, very erratic germination is observed. And second one is after germination also the growth rate is very non-uniform, so which is a major constraint in the production of grafted eggplants. And another is there is no, and so we need to exploit available other bacterial resistant genotypes to be incorporated for for bacterial resistance. So keeping these points in view, this experiment was conducted. Uh, so fixed objective, like first one is we'll screen the sound is coming. First one is we screened available germplasms, germplasm for bacterial resistance. Then out of the germplasm, screened germplasm, we selected some highly bacterial resistant genotypes and rootstock, and we used that in grafting, and we evaluated those grafted explants for bacterial resistance as well as yield and yield attenuating traits. So during this experiment was conducted in on a regional research station of IHR, Central Horticulture Research Station in Bhubaneswar. Here the experiment was conducted due to several factors. First one is for sir, the sound is coming. The favorable climatic condition like high temperature and high humidity is available here. Second one is the soil conditions. The soil is low temperature soil, which is a problematic soil with a very high acidity. As we know, Ralstonia, the bacterial infestation increases many folds in sir. Sir, sound is coming, sir. Please mute.
Proceed, Dr.
Okay, sir.
You only requested to mute all the other speakers.
Sir. So due to highly acidic soil in nature and sir, please mute, sir. The sound is coming, sir. Too much sound.
Hello.
Sir, actually, background sound is coming very much, sir. So, will everyone move it?
Please proceed. The time is running.
Yes, sir.
There is another session also is there.
Okay,
no, what is the problem?
Sir, actually, sound is coming from background, sir. Too much noise is there. So,
no, we could able to hear. We could able to hear well.
Okay, sir. So,
please make it faster.
Okay, sir. So soil being very highly acidic in nature and environment being so the screening. So the experiment was the site was selected for carrying out this experiment. So first one is plant material. So for screening population, screening process, we procured 30 genotypes from all over the India from different SAUs, including IRI, IHR, Kerala Agriculture University, then UAT, etc. And some wild sonalam species like Torbum, Cicidipolium, Ethiopium, and we used in a screening program. And for grafting purpose to be used as scion, we selected two high yielding genotype with having a very high bacterial susceptibility reaction, like Arca nilachala syama, developed from Central Horticulture Research Station, IHR Bhubaneswar, and the second one is one private sector hybrid BNR 212. First one we did screening. First we carried out the screening program. So for that one we first isolated Ralstonia solanacearum pathogen. So plant sample, the bacterial wilt plant sample is collected from Bhubaneswar only from sick blood. Then 1 centimeter small length portion was taken, connecting root and stem portion from the basal, and it was cleaned and sterilized with surface sterilized with sodium hypochlorite solution, then followed by repeated rinsing. Then it was macerated by the help of water pistol and streaked onto TTG media. And then after an incubation period of 48 to 72 hours, the bacterial colonies were appeared. Then from that particular colony, creamy white growth with pointed pink color at the center portion was selected, and it was subcultured, and that pure culture was prepared and stored for other inoculum preparation. Then for inoculation, we took two freshly cultured in a nutrient solution, and then it was put in a shaker for 24 hours. Then after 24 hours, we centrifuged it at 4,000 RPM for 15 minutes at 4 degrees Celsius, and the supernatant were discarded. Then actually one bacterial pellet was formed at the end of centrifuge tube, which was then again distilled sterile water is added to make a final concentration of 10 to the power 7 colony forming unit per ml. It was calibrated by the use of spectrophotometer, in which OD 600 nanometer value was obtained 0.3. Then after inoculation preparation, then inoculation of before planting into the main seed plot, seed plot, the seedlings, just 30 days old seedlings, were root tipped in that bacterial suspension before transplanting in the seed plot. Now, one thing we did, we withdraw irrigation for 24 hours so that to send the plant to stress and for quick build up, means for 20 to 30 seconds deep into the bacterial suspension. So the plant was first preliminary stressed by withholding water for 24 hours. Then after transplanting in the sick plot, observation was recorded at every 3 days interval. So whenever a bacterial appeared, and after 10 days it appeared, whose test was done for confirmation of bacterial disease, and then it was scored from 0 to 5 and was classified. The bacterial percentage was calculated by total number of plants died by total number of plants. Then accordingly, the score was given 0 to 5. So the group was classified according to immune, highly resistant, moderately resistant, moderately susceptible, and highly susceptible, according to the percentage of infestation observed in the population. Then the germplasms were grouped into, as I told, 5 groups. So Solanum tabon, Carry 1, Utkal 3, Arka Nidhi, Arka Anand, and Arka Harsita from IHR. Then IHR BNE1, IHR BNE3, and Sweta and Surya were found to be highly resistant. Now another observation we took is Solanum sessimifolium, which was reported to be bacterial resistance in most of the studies, was found highly susceptible in our study due to very optimum amount of due to optimum temperature and humidity, as well as the soil conditions. So then out of the 11 highly resistant genotype, we say, 7 genotypes for to be used as rootstock: Solanum tabum and Arcasita collected from IHR. Then Carry 1 from Carry, Andaman Nicobar Island. Then Utkal Anusri from OUT, Bhubaneswar, Odisha. Then IHR B, NE3 and NE1 from northeastern part of India. And IC series selected from local region of Odisha. So these during the screening population, as you can see, during screening program, we transplanted Pusas purple lung, which is used as susceptible check in every 2-3 rows interval to give to have a clear cut distinction between resistant and susceptible genotypes. And you can see the Carry 1, which was identified the highly resistant genotype, was against the Pusa purple lung susceptible check. Then after that selection of rootstocks, resistant rootstock, we went for production of grafted eggplants. So first one is for production, we 40 days old rootstocks and scion were made. Care was taken to select those plants having the same thickness of stem, so that a proper graft union can be obtained. Then after selection, then beheading of the rootstock was done, and one vertical cut was given on the rootstock, generally adopted in cleft method of grafting. Then scion was given similar cut so that it will fit properly within that rootstock. Then scion was inserted into the rootstock. Then specially available grafting clips were used to hold the rootstock and scion in position. Then after that grafting procedure complete, were transport to a healing chamber with high humidity and temperature of 25 to 26 degrees Celsius. Then they were kept in the healing chamber for first 4 to 5 days, after which the grafting clips were removed. Then they are kept for another 2-3 days within the healing chamber. Then after then it was transferred to into a polyhouse chamber for hardening, little bit hardening. Then after another week, it was transplanted in the main field. So this one is the graft union of the successful on a successful graft union is observed, and the grafting success was minimum to be found to be 85%, which extended up to 100% too. Then after grafted plant product, then we went for the evaluation plant bacterial two replication in 40 replications for in each replication, and RCBD design it was laid out in RCBD design. So the bacterial screening was performed both in field condition as well as in normal field condition as well as in sick plot condition having high bacterial inoculum load. So in field condition, in grafted plants, in whether the same may be BNR 212 or Arcanilla chama, a very minor amount, negligible amount of wilt bacterial infestation was observed. When we come to bacterial infestation screening in sick plot also, as compared to the non-grafted plant, nearly in which in VNR the non-grafted 80% and also in Arcanilla more than 80%, nearly 80% plants are completely due to bacterial infestation. Now the grafted plants performed far better and showed very much highly bacterial resistant reaction. All the and the carry one being the highest, carry one showed the highest bacterial resistant reaction when suppressed even Solanum taupum. And all the grafted, and if you consider the effect of grafting, all the rootstocks, identified rootstocks performed statistically on par with also carry one. We also in like in a program or screening program. Are you able to see the slides?
No, make it fast because time is running. You come to conclusion now. Hello. You can conclude now.
Yes, sir. Coming, sir.
Yeah.
Yes.
Please.
Then it and yield attenuating plates. Then generally the plant weight was non-significant. The grafting did not affect plant weight. However, the grafting had significant and also when you sort fly, it says the non-grafted plant produced early flower as compared to grafted plant. Then when you come to when upon investigation on yield rate, so number of the carry one of BNR2 and Arcanilla chama, and the yield increase over yield increase over non-grafted control. 20. That's a non-grafted plant. And another observation that grafting had no impact on the rate, which is a positive. Right. Affected the fruit shape and size. So we need as eggplant acceptance is purely dependent upon consumer acceptance. So we can see that grafting had no effect on fruit shape and size. So it's another positive point. So in conclusion, so what we did was to mitigate bacterial disease, we screened 30 genotypes, out of which 11 highly resistant genotypes we found. So from our 11 resistant genotype, we selected 7 resistant rootstock for grafting, and out of which carry one, IHRB, NU1, and Utkalanusri and I3 05984330 performed very nice, and carry one was found to be the best. Now these rootstocks can be used as an alternative for conventional use Solanum taupum in case of grafting to successfully combat bacterial disease. Thank you, sir. Thank you, sir.
Thank you very much. I think time is running. Hello.
Yes, sir.
Hello?
Yes, sir. Yes, sir.
Anybody has got a quick question? If no questions are there, we request Dr. Nidhi.
Yes, sir.
Please make presentation.
Sir, I would I've got the Nidhi's slides. I'll be sharing for her. So, is the presentation visible, sir?
Yeah, visible. Make it visible. You go ahead.
Nidhi, can you please present?
Yes, ma'am. Thank you, ma'am. Again, very good afternoon to everyone. The topic of my presentation is a novel and eco-friendly remedy for bullseye pathogen infecting tomato. And our motto is that we should have a lab to land and eco-friendly approach. That is what we are doing in our research labs. It should reach to the farmers because they are the end users and at a very low cost and in a very effective manner. Next, please. Ma'am, next slide. So the reason and the motivation for the research work is the chemical pesticides, the agrochemicals we are using in our agricultural fields, because we need to minimize the use of these chemicals for controlling the plant diseases, because they are causing so many health problems, environmental problems, pest resurgence, pest resistance, and many more. So bioformulations are the need of the hour, and they can be a potential alternative to that. Next, please. So need for the research, we targeted the tomato crop, which is known as superfood, and we have a kind of this Economic Times article that is showing that there is a price hike in the last few years, and one of the reasons being pests have become immune to general pesticides. So during a survey in and around Faridabad, that is Haryana region, we found that 40 to 80% losses of tomato crop was due to early blight disease caused by Alternaria solani, which is known as bullseye pathogen. Next, please. So our objective is the development of IDM strategy or bioformulation, which is ready to be used by the farmers to be distributed to the farmers. Next, please. First of all, a detailed survey and surveillance was undertaken in the year 2012-13 in and around Faridabad. Many villages were surveyed with heavy losses due to Alternaria blight. Next, please. The data was collected from the 20 villages on person disease index of early blight disease. Next, please. This is the survey report you can see where a percent PDI was calculated, and it was seen that the percent disease incidence was very high for the early blight. Next, please. So AUDPC was calculated for different tomato genotypes. Next, please. So now we know that microbial resources are the biocurrency for next generation. So various isolates of Trichoderma harziana were evaluated against Alternaria solani. Next, please. Both in vitro and in vivo conditions, and we selected a few of the isolates. Next, please. We selected a few of the isolates, and among them T2 and T5 were taken for the further studies. But as Nayak sir stated that in the inaugural session, that Trichoderma harzianum could not be a panacea and cannot alone solve the problem. So keeping in view after the preliminary studies, we took a number of botanicals, de-oil cakes. They were evaluated both in vitro and in vivo. Next, please. So next, please. So here you can see the photographs, the effect of plant extracts on growth of mycelium. These are some of the selected photographs only. Next, please. So many plant extracts were evaluated, and eucalyptus was found to be the most effective in vitro conditions with 80.7% disease inhibition. Next, please. And here in de-oil cakes, Karanj cake, that is Pongamia cake, was found to be the most effective in case of early blight in Faridabad region with 55% disease inhibition. Next, please. Fungicides were also screened against different strains of Alternaria to be used as a part of IDM strategy, and Vancomycin was used for the further studies, which was found to be most effective. Next, please. The selected components and the selected treatments were evaluated under net house conditions and then in the field conditions with different combinations also. Next, please. Now, following the integrated approach against the pathogen, the best effective and compatible components, these were selected with the kind of proper right scientific attributes which contributed towards the overall objective of the project. So we tried all the kinds of combinations for the compatibility and other things to be studied. Next, please. Next, ma'am. These are all the treatments we used with the different plant extracts, different isolates of trichoderma and different kinds of oil cakes. And again, T5, which was selected with eucalyptus and karanj cake, the integrated treatment, which was taken forward on the basis of the data on germination percent, plant growth parameters and percent disease inhibition, disease severity, etc. Next, please. So we developed the bioformulation keeping in view all the above points and farmers' aspirations. We formed a kind of tech-based bioformulation. Next, please. And it was named as bioelixir. Next, please. So we distributed this bioelixir, but before distribution, we again studied the in vitro effects and the net house impact of this bioformulation on various aspects of the disease. Next, please. And that too on different isolates and different strains of Alternaria. So the impact was seen in the farmers' fields also. In net house conditions, it was with the different tomato genotypes, and percent inhibition was seen for all the tomato genotypes. Next, please. So here, IDM strategy on growth of plant under field conditions was seen, and it was with the Sinova Lyco, that is 26.1% PDI was seen with this tomato genotype. Next, please. So now it was also discussed in this conference that we should make this biofertilizers and biopesticides to be a part of the bioeconomy or a circular economy, and we should encourage entrepreneurship. So I am representing an example that we, what we are doing in lab, we are also taking it to make it a startup, and we are in now collaboration with IOCL, and it's a all women startup. We are now doing the further studies in collaboration with IOCL. Next, please. So we prepared a kind of bioformulation taking into account their agro oil, which is totally organic, and they are trying to invade this agriculture market. So this agro oil, in consideration of the stability, shelf life, costing, compatibility, different emulsifiers were used in different ratio, doses, and sequence of adding ingredients to make it a smart formulation. Next, please. We are very thankful to different premier agricultural universities with which we are collaborating, and the renowned scientists which we are working with them. And you can see it's IRI, University of Horticultural Sciences, BCKV, and SQST, and I'm very thankful to them to be a part of this project. Next, please. Next, please. These are a few photographs only. Yeah, the recommendations for the reduction percent in percent disease incidence was given by different agricultural universities for our bioformulation, and they have taken into account all the 4 agroclimatic zones so that we can see the impact or according to in relation with the climatic conditions also, and it was seen that it is at par with the chemical pesticide, and we can at least replace 50% of the chemical use in case of early blight disease in tomato. So the treatment T3 was found to be best in most of the agroclimatic zones, but in case of Srinagar, it was 25% chemical plus 75% oil bioformulation, which revealed the best results for the farmers. Next, please. We started with the early blight disease, but in case of bioformulation, it was found to be broad spectrum and on the late blight disease and other diseases also. Next, please. So there different diseases were studied like target leaf spot, septoria leaf spot, cercospora leaf spot. So folio and fungal diseases were mainly targeted. And again, it was 50% chemical plus 50% oil bioformulation, which was the best effective treatment. Next, please. Increase in yield was also at par with the chemical pesticide, and the recommended dose was 50% chemical and 50% oil bioformulation for further taking into account. Next, please. That, as we know that we are talking about integrative plant health management, lycopene was also analyzed and it was found to be better in the treatments with the oil bioformulation as compared to chemical pesticide. As you can see here, T12 is giving the highest lycopene content, followed by T11. And now the conclusion. We can say that trichoderma or BCA based bioformulation must be upgraded so that they can replace chemical and overcome the shortcomings of the past. And this is an example. It is a proven component of the IDM, that is bioformulation, which is a safer alternative, and it's providing a number of other features also like early flowering and fruiting, no residues, and better quality tomatoes. And we are having the higher shelf life of more than 1 year for this bioformulation. Thank you. Next, please.
Thank you, Dr. Vijay. We have made an excellent. Hello.
Two more slides only.
We have made an excellent presentation, and we brought out a product also.
Yes, sir.
My suggestion is that in tomato we have the early blade, we have the late blade also.
Yes, sir.
Yeah, if you could able to evaluate this product against the late blade also.
Yes, sir.
It may be more meaningful.
Yes, sir.
Okay, thank you. Any questions are there? If no questions are there, now we'll request Mr. Vinay. Vinay is available?
Yes, sir.
Please make presentation. So you will make the presentation of zinc nanoparticles. Dr. Vinay?
Yes, sir. Yes, sir.
Please make a presentation.
Yes, sir. I'm uploading, sir.
Yeah. Dr. Vinay?
Yes, sir. Yes, sir.
Please.
Is it?
Its slides are not visible.
But
Make it fast because many people may getting hungry.
yes, sir. But
Time also running very fast. Please take 10 minutes or not more than that.
yes, sir.
Yeah. Now we make presentation.
Yes, sir.
Please carry on.
Welcome to the presentation green synthesis of Saccharomyces cerevisiae based zinc nanoparticles and their antimicrobial activity against plant pathogens. Nanotechnology manipulation of any compound at an atomical size range of 1 to 100 nanometer, and it is a tool in agriculture, especially in place of plant action. Green synthesis of nanoparticles has great importance because the toxic chemicals are involved. So, and these are very stable nanoparticles and with a efficient in action and cost-effective synthesis. In this, Saccharomyces cerevisiae extract were used as a green reducing agent. It is a flaw of research. We have isolated the Saccharomyces cerevisiae as a different fruit sample, and they are screened for the zinc solubilization ability, and a strain showed the solubilization zone was used for the synthesis of the zinc nanoparticles. This solution was prepared, was added under the receiving condition, and
Dr. Vinay,
zinc nano and regular shaped zinc nano tomato at 5, and then the zinc are evaluated against the major plant pathogen. Citrus root of the bacterial and minimum zone of if against the filial growth effect, then zinc nanoparticles also suppress canker and bacterial wilt of tomato. The particles also then both antibacterial and
मेरे को डायरी के ऊपर तो मैंने भी किया था इसको ये करना था
Thank you.
तो मैंने विंडोज सभी क्लोज कर दिए थे लेकिन क्लोज नहीं किया था क्योंकि वो फिर कनेक्शन प्रॉब्लम होता है ना.
Thank you, Dr. Vinay. Dr. Vinay,
ठीक है चलो.
Dr. Vinay, thank you very much.
Yes, sir.
Do you have any questions, sir? Anybody has got any questions or queries, suggestions? If no queries are there, at the outset, I am highly thankful to the organizers, particularly Dr. Devapa, Dr. Sankarappa, and Dr. Sangeeta, who has given me an opportunity to chair an important session on integrated plant health management. So, though its session is the end of the symposium, but this is very important, particularly in the field of plant pathology. Whatever you do, molecular work and all those, finally, the disease management is most important and critical. So, I compliment all the speakers. Today, we have the 5 presentations listed. Out of 5, 2 are keynote lectures, 1 is invited lecture, and 3 are oral. Out of 2 keynote lectures, only 1 is presented, that is Dr. Dinesh Singh. Dr. Dinesh Singh, all of you know very well, he is a well-known plant bacteriologist in this country, and his contributions are well-known. And I compliment Dr. Dinesh Singh, who has made an excellent presentation, and he has given new thoughts, particularly the grafting, biocontrol agents are the way to manage the plant bacterial diseases. So I compliment Dr. Dinesh Singh, who has given the new lead for the management of a plant bacterial disease in this country. The second lecture was invited lecture, Dr. Vinayak Hegde. Dr. Vinayak Hegde is also known for always good presentations, and he is highly committed, and he made a really wonderful presentation. And I compliment Dr. Vinayak Hegde that till today we are recommending a borders mixture for management of a fruit rot of aragonite. The farmers always often ask questions, since how long we recommend border mixture? But thing is that though we have a large number of fungicides for management of phytophthora diseases, but we cannot recommend these fungicides, particularly in the high rainfall areas like the west coast of India, because the chemicals can be washed out. That's why we are recommending border mixture only. But Hegde came out that Mandy bromide, this also in contact fungicide only, and if possible, we can go for alternate. The border mixture alternating with Mandy propamide can be recommended. This is one of the best thing he has identified. Beside this, and he could able to promote new solutions. For example, spraying a tall coconut aragonite is very difficult, and he brought out a technology for tractor mounted spray and drones. And these are the actually new thinking. I compliment him for a better thinking. Besides this, we have seen the newspapers recently. There is a Contotrichum blight affecting the aragonite in many parts of the Karnataka. Besides this, we have also heard the bacterial disease on aragonite. I think Dr. Dinesh Singh may also visit these areas and collect samples and compliment the CBCRI, what Dr. Hegde is doing. And this is actually, it was there long back, it has reappeared, re-emerged disease. So I compliment the Dr. Vinayak Hegde for the wonderful presentation. And Dr. Nidhi, and we had compliment her. So she wanted actually reduce the chemical inputs for management of early blight in tomato, because this is required, because one side spraying is going on, other side harvesting is going on. Actually, the consumer will be affected because of the residues of the fungicides, but she could able to identify the product called bioelixir, and she brought out to the level of commercialization. I compliment Dr. Nidhi, and I request Dr. Nidhi that she has proved that this product is highly valuable, highly effective for management of the early blight of tomato, because tomato particularly in the monsoon season, in addition to the early blight, the late blight also will come. Then other bacterial diseases also will come. So you should be able to show that this product for efficacy against all the leaf species of the tomato, that will be the great contribution. So we compliment her also. Next, Dr. Satyaprakash. I think Dr. Satyaprakash is working on the lines of Dr. Dinesh Singh only, and he could able to identify a CARI1 as rootstock of the brinjal. In addition to the what we call Solanum torum is already rootstock is available. Solanum torum is being used as rootstock for grafting of the vegetables, solanaceous vegetables. Besides this, you could able to identify one more rootstock, the CARI1. I compliment the Dr. Satyaprakash for making a inroads, identify the new rootstock. And last one, Dr. Vinay, and I compliment him. He would able to identify the zinc nanoparticles. But there are ethical issues are there in the nanoparticles that should be resolved before the make it commercialization. So once again, I compliment all the speakers who has made excellent presentations. But still, there are senior people like Dinesh Singh and Dr. Egde. They made really wonderful presentations. They are given a new thoughts on the disease management. And finally, I am thankful to the organizers giving me an opportunity to chair this session. So after retirement, actually, I moved to the farming. I'm doing agriculture now, and this session may be helpful to me also. Thank you very much.
Thank you, sir. I thank profusely the chairman, Dr. Chaudappa sir, for your valuable comments and suggestions to all the speakers and for having completed the session successfully. I further thank the co-chairman, rapporteurs, all the keynote, invited and oral speakers for their excellent presentation. My sincere thanks are due to all the participants for their active involvement in this session. I'm glad to inform you all that we have successfully completed all the 9 sessions of this international e-conference on multidisciplinary approaches for plant disease management for sustainability in agriculture. I request all of you to rejoin at 2:30 p.m. for the valedictory session of this conference. Thank you one and all. Thank you, sir. Thank you so much.
Thank you. Thank you, Dr. Dinesh Singh. Thank you, Dr. Egde. We are meeting after a long time. Thank you very much.
Thank you, sir.
Hello.
No.
ಕಳ್ಸಿದ್ರಾ?
Good afternoon, sir.
Okay. Okay. Is audible? Is audible? Is audible? 1, 2, 3.
Yes, sir. ನನ್ನ ಮೇಲ್ಗೆ ಹಾಕಿದ್ರಾ?
ಲಿಂಗರಾಜು ಹೇಳಿ. ಜನಾಗ್ತೇನೆ
సార్ టోటల్ రాజకీయంలో
A very good afternoon to one and all. Validated function will start a few minutes at 2:55. So kindly join us by that time. Thank you.
Hello, Dr. Dev.
Yes, sir.
Hello, start ಮಾಡ್ಬೇಕಾರೆ ಎಲ್ಲ ಬಂತ? Everybody has come?
Yes, sir.
I will start.
Yes, sir.
Sir,
Everybody has come.
DDG horticulture has to join, sir.
The DDG horticulture has called me and told he
Yes,
is
sir.
at another meeting. DDG horticulture is not participating in the plenary session.
Yes, sir.
He has confirmed it. So, DDG horticulture, you just leave it. Actually, ಇದು plenary session though. Plenary session to ಬೇರೆ ಇರ್ತದೆ. No. Huh? 9th day, last plenary session.
Sir, we'll start sharp at 2:45, sir.
Okay.
Okay, sir. Thank you, sir.
Mute ಮಾಡಿ. Program sheet close. Yes, sir. Plenary session. Technical session. Session. Good. Hello, Dr. Jagdish.
Start with sir.
Jagdish, our DDG horticulture
Yes, sir.
he is not participating in the plenary session.
We'll start, sir. Yes. Okay. Sangeeta?
Yes, sir.
Start ಮಾಡಿ.
ಸರ್ ನೀವು ಅಲ್ಲಿ ಮ್ಯೂಟ್ ಮಾಡಿ ಸರ್.
Okay.
Afternoon to one and all. I extend a very warm welcome to all of you to the valedictory function of the in achieving sustainability in agriculture.
Oh, thank you, Sangeeta. So we are, good afternoon to all of you. We are coming to the end of the mega event, that is the e-conference on multidisciplinary approaches for plant disease management for achieving sustainable agriculture. So we are organizing this from 6th of this month to the 9th of this month. This is a plenary valedictory program. I welcome to all of you. First of all, I welcome our Honorable Vice Chancellor, sir, Dr. K.M. Indreshji, for his support, and he has given all the support for organizing this e-conference. On behalf of the organizing committee, on behalf of the College of Horticulture, Department of Plant Pathology, a warm welcome to our Honorable Vice Chancellor. Welcome you, sir. Secondly, I welcome our eminent scientist and Deputy Director General Horticulture, Dr. Anand Singh, who is not there, but he is going to join later in this program. He's an eminent scientist in the field of horticulture. On behalf of the organizing committee, I welcome you, sir. Similarly, I welcome our beloved Registrar of University of Horticulture in Palanpur, Dr. T.B. Allodi. He has given all the support for organizing this e-conference, a mega event, and he is going to distribute some awards to the speakers who has done excellent presentation during the course of this e-conference. I welcome all the participants. At present, there are 225 participants are viewing our valedictory program. I welcome all the participants, keynote speakers, the participants who have posted presentation. Totally, we have received the participants from 20 countries, but last 4 days, we have been organizing this in 9 theme areas. Almost all 300 participants have participated. Over 34 speakers and 282 poster presentation. I welcome all the participants from different parts of the world. Those who are joining the online today, I welcome yourselves. Similarly, I welcome all the faculty members, all the staff of College of Horticulture, Bengaluru. They have supported all the 4 days for organizing this e-conference. Also welcome the organizing secretary, Dr. V. Devappa and his team, Dr. K. Shankarappa, Dr. Sangeeta, Dr. Rajeshwari, and all the members of our college for this valedictory program. I welcome one and all the members who have directly or indirectly supported for organizing this e-conference. And I warm welcome to all of you. Thank you. Over to Sangeeta.
Thank you, sir. Thank you for welcoming everyone. Now I request Dr. V. Devappa, Organizing Secretary, to wrap up the International E-Conference.
Good afternoon. It's my pleasure to extend warm welcome to you, all the delegates, valedictory program of the International E-Conference. I'm very proud to say that it was overwhelming response from all over the globe, and we received 1,758 to the conference. And institutes have actively participated in the conference, and we received more than 310 papers and abstracts or research articles for the 9 sessions, out of which we selected only 282 abstracts for the conference. Sir, after inauguration of this conference by Honorable DDG ICR, inaugural yes, delivered the talk molecular cloning of plant disease resistance genes, a way forward. And later, the keynote address as given by Dr. M.K. Naik, Honorable Vice Chancellor, University of Agriculture and Horticultural Sciences. And he has emphasized management. He has given more importance to the biological control rather than chemical control. And later, Dr. R.C. Jagdish, the Dean of the College, in his presidential remark, he has emphasized more on the molecular technique should be the one of the tools in addition to the conventional breedings. And later, the vote of thanks has been organized by the Dr. A. Shankarappa. The totally 9 themes we conducted. The first theme is the recent advances in detection and diagnosis of plant diseases, in which 3 speakers, 2 invited talks, and total oral presentation are the 4, and overall 57 presentations has been taken up in the recent advances in number 1. As in case of plant resistance, the total 50 poster presentations, 6 oral presentation, and 3 keynote speakers from various delegates. In case of plant microbial signaling, there are totally 8 poster presentations, 4 oral presentation, and keynote speakers are 3. In which, in the climate change concern, climate change 3 keynote speakers and 4 oral presentation, in total 9 presentation. Recent molecules plant disease management, 3 keynote speakers and 2 invited talks, oral presentation 2, and 51 that is the poster presentation. It clearly states there's a lot of importance has been given in the plant.
We can't hear you, Dr. Devappa.
Sir, we are not able to hear you. Video constantly. That's ಅಲ್ಲಿ connect ಮಾಡ್ತೀನಿ. ಅದು
Hello. Hello. Yes. Can you hear me?
Yes, sir.
Somebody has muted. Somebody has muted. There is some technical problem.
No, we can
Please,
hear you.
sorry for the
We
inconvenience.
can hear you.
Sir, can you hear me, sir?
Okay, yes, we are hearing.
We are hearing. Okay. Yes. Over to Devappa sir now. There is some technical issue. Sorry about it.
Is it clear?
Yes, sir. Sir, continue, sir.
My wife, I'm audible.
Ambika, can you hear sir's voice? Yes, sir.
Now, I think, good afternoon. It's my pleasure to extend warm welcome to you all, the delegates, to the validatory programs of International E-Conference. I'm very proud to say that there was overwhelming response from all over the globe, and we received more than 1,758 registrations to the conference. Out of universities, bar universities, institutions have actually participated in the conference, and we received more than 310 research papers for all the 9 sessions. Whereas in case of the participation from the different countries, totally we received more than 15 countries participation to this conference. It including Israel, that is Costa Rica, Spain, Poland, then is including Sri Lanka, China, and other countries. Out of which we selected only 282 abstract for the conference. The following is the bird eye view of the presentation. Overall, in the first theme, we received a total number is 37 poster presentation. The wood plant resistance is concerned 50 presentations, oral poster presentation, plant microbial interaction and signaling 8 poster presentation and 3 keynote speakers, climate change and pathosystem 19 poster presentation, 4 oral presentation and 3 keynote speakers, recent molecules in plant disease management 3 keynote, 2 invited and 2 oral presentation, and totally 51 oral poster presentations. Whereas in case of omics and informatics in plant pathology, we received 3 keynotes and 5 oral presentation and 7 total oral presentation. Whereas in case of biological control of plant is concerned, there was overwhelming response to this particular theme, and we selected 77 poster presentation, and total oral presentation were 6, and invited talk is 1, and keynotes also it is a more year, that is 5 keynotes talks have been selected. Whereas today's session is concerned, virus vector relationship, one keynote has been given by Krishna Kumar sir, and 2 invited talks, and totally 12 poster presentation. In the final theme number 9, that is integrative plant health management, there are 2 keynotes, 1 invited talks, and 3 oral presentation, oral and 49 poster presentation. If we look into the total participation and is concerned, there are totally 26 keynote speakers and 8 invited talks, 34 total oral presentation, and 310 poster presentation. Look into this situation is concerned, it was overwhelming response from all over the globe, and because of this response is because of all the speakers, keynote speakers, invited speakers have given lot of coordination for us. Because of that reason, the entire program is successful. Thank you very much.
Thank you, sir, for giving us the summary of the conference. Now it's time for declaration of awards. Now it is over to Dr. Shankarappa KS to declare the best oral presentations under different themes. Over to you, Dr. Shankarappa.
1 minute. Is it visible, madam?
Yes.
It is my pleasure to announce the most exciting and awaiting results. The best oral presentations of the different themes. Best oral presentation under theme 1 goes to Dr. Ravindra Kumar on paper on development of one-step RT lamp or visual detection of patovirus M. Under theme 2, best oral presentation goes to Dr. Girish, who has given a talk on identification of blast resistance lines from Varroa glaberrima accessions of rice. Under theme 3, the best oral presentation goes to Dr. Somnath S. He has delivered talk on a transcriptome analysis of Heterodira saccharine, reveals its adaptation to parasitize Manacot Bose. The under theme 4, the best oral presentation goes to VR Kulkarni, who has given a talk on effect of weather parameters and development of alternaria leaf spot and grey mildew of cotton. Under theme 5, the best oral presentation goes to Mr. Mahesh HS, who has given a talk on colloidal silver hydrogen peroxide, a novel tool for management of soil-borne plant pathogen. Under theme 6, the best oral presentation goes to Mr. Ashok Vasant Rao Kulkarni, who has given a talk on genomic selection of model for blister bark disease tolerance in Casuarina natural population. Under theme 7, the best oral presentation goes to Dr. Gurdatta M. Egde, who has given a talk on potentiality of biopesticides against fungal foliar disease of vegetables grown organically under protected cultivation. And under theme 9, the best oral project goes to Dr. Nidhi. She has delivered talk on a novel and eco-friendly solution for bulleye pathogen infecting tomato. Thank you, one and all.
Thank you, Dr. Shankarpa. Please kindly share. Thank you, Dr. Shankarpa. Now we go to the best poster presentations. I now request Dr. Rajeshwari to take over and present presentations or declare the awards. Over to you, Dr. Rajeshwari. Dr. Rajeshwari. We are not able to hear you. Mute yourself, Shankarpa.
Hello?
Yes, we can hear
Are you able to hear me?
Yes.
Yeah. I'm very glad to announce the awardees under the poster presentation. Under theme 1, recent advances in detection and diagnosis. First best poster award goes to Bindu KG on the title Survey for the Assessment of Prevalence and Incidence of Pigeon Pea Wilt Caused by Fusarium oxysporum Pharma species Udam. And the second best poster award.
Your PPT is not visible.
Yeah, I'm trying to load it. One second. The second best poster award goes to Karthik Pandey and co-authors for the title Morphological and Molecular Characterization of Chili anthracnose. Under the theme 2, Host Plant Resistance, the first best poster award goes to Himanshu Dubey and co-authors for the poster on identification and in silico expression analysis of NBARC domain containing genes in mulberry. And the second best poster award goes to Aruna Kumar GS and his co-authors for the poster on the title Emerging Foliar Fungal Pathogens and Post-Plant Resistance of Mulberry in India. Under the third theme, Plant Microbial Interactions and Signaling, the first best poster award goes to C. Sarathambal and the co-authors for the poster presentation on the title Arbuscular Mycorrhiza Mediated Effect on Growth, Nutrient Uptake, Physiological and Biochemical Activities of Black Pepper Cuttings. And the second best poster award goes to Sinijadas K and his co-authors for the poster presentation on the title Role of Bipolaris Toxin on Bipolaris Sarociniana, causal agent of spot blotch of wheat. And
Sorry, we cannot see your slides.
For the poster for on integration and melatonin incognita in causing wilt complex of pomegranate under climate change conditions. And the second best poster award goes to Dinesh Kumar and his co-authors for the presentation on occurrence of bacterial canker on citrus or Aurantifolia in relation to plant disease management. First best poster award goes to Akshita Kale and his co-authors for presentation on the type
Voice is also breaking.
bacteriophages specific to
व्हॉट्सअप ग्रुपवर पाठवलं तर क्लिक करा त्याच्यात तुमचं दिसेल ते. अरे मी 5 सेमिस्टरचे ग्रुपवर पाठवले सगळ्यांच्या तुझं सेमिस्टर 5 आहे ना.
Dr. Rajeshwari, we cannot hear you. You're not audible.
For the presentation.
Network, namde problem. Nam network problem.
Co-author.
Okay. Illi idu nan connect. Orange ಕಾಣಿಸ್ತದೆ.
Fish. Moonbeam.
Devappa, please check for the audibility, please.
Yes, sir. Yes, sir. We are checking, sir. It's
Okay. Yeah, your voice is clearly audible, but there it is not. Okay.
yes, sir. Network is different. I have taken a different network, sir. I will check, sir.
It's a technical problem. I'm sorry for this. All the participants and the delegates. I'll read the presentation awards once again. Can you hear me? Can you hear me, please?
Yes,
madam.
Go ahead.
Yes, sorry for the interruption. I apologize. I will recent advanced diagnosis of plant diseases. The first poster award has been given to UKG and her co-authors for the poster presentation on the title Survey for the Assessment of Prevalence and Incidence of Pigeon Pea Wilt caused by Fusarium oxysporum forma species udum. And the second best poster award has been given to Karthik Pandey and his co-authors for the poster presentation on morphological and molecular characterization of chili anthracnose. Under the theme 2. Host plant resistance. The first poster has been deciphered to Himanshu Dubey and his co-authors for the poster presentation on the title Identification and In Silico Expression Analysis of NBARC domain containing genes in mulberry. The second best poster award has been deciphered to Aruna Kumar GS and his co-authors for the poster presentation on the title Emerging Folia Fungal Pathogens and Host Plant Resistance of Mulberry in India. Under the theme 3, Plant Microbial Interactions and Signaling, the first best poster award has been given to C. Sarathambal and his co-authors for the presentation of poster on the title Arbuscular Mycorrhiza Mediated Effect on Growth, Nutrient Uptake, Physiological and Biochemical Activities of Black Pepper Cuttings. Am I audible?
Yes.
Am I audible? Yes. Thank you so much. Thank you so much. And the second best poster award has been deciphered to Sinijadas K and his co-authors for the poster presentation on the title Role of Bipolaris Toxin on Virulence of Bipolaris sarociniana, Causal Agent of Spot Blotch of Wheat. Under the theme 4, Climate Change and Weather System, the first best poster award has been backed by R.K.
Not audible, madam.
Investigation on interaction of Ceratocystis fibreatum and Melanodyne incognita in causing wilt complex of pomegranate under climate change conditions. And the second best poster award has been backed by Kumar and his co-authors for the poster presentation on the title Occurrence of Bacterial Canker on Citrus aurantifolia in relation to different meteorological parameters. Under the theme 5, Recent Molecules in Plant Disease Management, the first best poster award has been given to Harshita K.N. and her co-authors for the poster presentation on the title Studies on the Bacteriophages specific to Xanthomonas axonopodis Pathover punicae. And the second best poster award has been deciphered to Rajeshwari and Kiran Babu for the poster presentation on the title Evaluation of Fungicides Price. Under the theme 6, Omics and Informatics in Plant Pathology, the first best poster award has been given to Dattatreya Hegde and his co-authors for the poster presentation on the title SMRT Sequencing of Pomegranate Bacterial Blight causing Xanthomonas axonopodis Pathover punicae to improve microbial genome coverage. And the second best poster award has been deciphered to Sangeeta N and her co-authors for the poster presentation on the title Cultural, Morphological and Molecular Variability in Different Isolates of Sclerotium rosei causing cholera of chickpea. And under the theme 7, Biological Control of Plant Pathogens, the first best poster award has been given to Nageshan SN and his co-authors for the poster presentation on the title Genetic Diversity of Bacillus subtilis from Western Ghats of Karnataka and Field Application for Controlling Potato Late Blight Disease. And the second best poster award has been deciphered to Uma MS and her co-authors for the poster presentation on the title Antagonistic Effect of Selected Bioagents on Ambrosia, a fungus associated with short hole borer of coffee. And the last theme, under theme 8, virus the first best poster award has been given to Ambarish S. and his co-authors for the poster presentation on the title Transmission Efficiency of Moonbeam Yellow-mosaic Virus by Asia 1 cryptic species of whitefly Bemisia tabasi in green gram. And the second best poster award has been referred to Praveen Kumar and Prasad SM for the poster presentation on the title Relationships of Moonbeam Yellow-mosaic Virus with its vector. So I congratulate all the awardees under the poster session. Thank you.
Thank you, Dr. Rajeshwari. Now I request our Honorable Vice Chancellor, Dr. Indresh sir, to give his presidential remarks for the valedictory function. Over to you, sir.
Good afternoon. Good afternoon, everybody. Today's International League Conference plenary session on multidisciplinary approaches for plant disease management in achieving sustainability in agriculture. The president of this today's plenary session, Dr. Jagadish, Dean, College of Horticulture, GKVK Bangalore, the organizing secretary of this International League Conference, and his team, faculty from the Department of Plant Pathology, College of Horticulture, GKVK, all the faculty members of the College of Horticulture, GKVK, and all my faculty members from different colleges, different research stations of the university, all the my dear participants from both India as well as abroad, who are participating from different institutions in this very important international conference on multidisciplinary approaches for plant disease management. First of all, I congratulate the awardees of both oral as well as poster presentations in the different sessions. So I congratulate all of you. I think as mentioned by the organizing secretary, Dr. Devappa, there are about 9 technical sessions and about 141 institutions have participated in this program with a total participants of 1,758 and totally some 310 research papers have been presented both in oral as well as poster presentations. I am very proud that my colleagues from the College of Horticulture GKVK are very successfully organized international conference involving those number of participants from both India as well as from abroad. So the university is very proud for your achievements, especially under the dynamic leadership of the Dean as well as Dr. Devappa and his team members, Dr. Sangeeta, Dr. Rajeshwari, Dr. Shankarappa, and other faculty members of the Department of Plant Pathology and also faculty members from the different departments of the GKVK. It is really a very proud moment for the university that we have achieved and we are successful in organizing this great event. The university is very proud and I congratulate all the team members of this e-conference, International E-Conference, for their very successful conduct of this e-conference. And because of this COVID situation, we have been forced to go for this international e-conference through video conference. And in fact, if we conducted this international conference physically, a lot of efforts, resources, manpower was required. It needs to be diverted. But because of this, even though e-conference was just my colleagues have taken a lot of effort in planning this e-conference, identifying the keynote speakers, identifying the participants, and Yes, it was well planned sessions and covering all aspects of plant disease management. Really, it is a very good move. And because of this COVID situation, we are able to interact with all the scientists from the different parts of the world. That is it is a very, very important. We were able to interact with them, and we were able to exchange our ideas with the many participants from both India as well as from abroad. It is an opportunity for us. Otherwise, we would have not met personally, physically, but this platform has given us the scope, given opportunity to meet and interact with the many experts, authorities on different aspects of the plant pathology through this video conference. Really, it is a very successful program, I say, and university is very proud about your achievements from the College of Horticulture, GKVK. And regarding the presentations on the various technical sessions, a lot of information has been generated, and many new technologies have come out. I hope that the organizing committee will review all these research papers, both was oral as well as poster presentation, and come out with recommendations. Give them recommendations which can directly useful for the farmers. If the technology transfer to the farmers' fields, it will be a very good achievement. So I request the organizing committee to review this presented by different authors and identify the technologies which are having practical relevance and which can be used for the farmers' fields. Ultimately, this the recommendations are very important from this international league conference, and definitely the university will be very supportive to all your progress, all the
पोस्टरमध्ये 3 दिले प्रत्येकाला 1 2 3
by the organizing committee. And once again, congratulate all the awardees and all the participants who actively participated in the International League Conference on Plant Disease Management. And finally, I congratulate the
मला काय वाटलं ते कार्तिक आहे तो कार्तिक वेगळा होता कार्तिक पांडे होता
the College of Horticulture GKVK for their endeavor in very successfully organizing this international conference and bring credit to the university. Once again, thank all of you for giving the opportunity to share my ideas. And I was supposed to participate in the inaugural session. Some communication problem, I could not make it. But however, I had an opportunity to interact with all of you in the plenary session. I thank the organizing secretary and all the faculty members of the Department of Plant Pathology for giving an opportunity to share my ideas. Thank you all.
Thank you, sir. Thank you for giving us your time and being a part of the valedictory function. Thank you, sir. Can you hear me?
Yes, you can.
Yes, thank you, sir. Thank you for being a part of this valedictory program. Now we have come to that part of the International E-Conference wherein we are about to conclude the conference. It is time to now thank each and everyone who has been a part of this conference from the day one. Now, first of all, I would like to thank our Honorable Vice Chancellor, Dr. K.M. Indresh, sir, for having supported us from the beginning. When we took this proposal to the Vice Chancellor and we gave this idea of conducting this international e-conference, he was very supportive and he has given his suggestions right from the title of the conference to the last day, that is today. He has been very supportive to us. Thank you very much, sir, for being a part of this conference. Next, I would like to thank our registrar, Dr. T.B. Alloli, sir, who has been the official correspondent or who has helped us in the official correspondence of all the matters related to the International E-Conference. A sincere thanks from our side to you, sir. Thank you very much. Next, I would like to thank Dr. Rajendra Prasad, Chancellor of University of Agricultural Sciences, Bangalore, for accepting our invitation to be a part of the inaugural session and to be the guest of honor. But due to some of his other urgent engagements, he was unable to be with us for the inaugural session. But still, I extend a heartfelt thank you to you, sir. Next, I would like to thank Dr. Tilakraj Sharma, the DDG Crop Science, who inaugurated our international e-conference by delivering his inaugural session on molecular cloning of plant disease resistant genes, a way forward, which also was a way forward for our international e-conference. I request all the participants to mute. Thank you. So, Dr. Tilakraj Sharma, DDG Crop Science, a very thank you to you, sir. Next, I would also like to thank Anand K. Singh, DDG General Horticulture, who had agreed to be the guest at our valedictory session, but due to some of his other engagements at the last moment, he was not able to be a part of it. I sincerely send my thanks to him. Thank you, sir. Now, on behalf of the university, I thank both the DDGs for accepting our invitation to be a part of this ET conference. Now, next person whom I would like to thank is Dr. M.K. Nayak, the Honorable Vice Chancellor of UAHS Shimoga, for addressing the participants and all the delegates in the inaugural session on the current situation of post-COVID in agricultural scenario. Thank you, sir. Now, on behalf of the university and also from the Department of Plant Pathology and the organizers, I first thank the speakers, the keynote speakers who agreed to our invitation to deliver the keynote address under various themes. First of all, I would like to thank the speakers from outside the country who agreed to even this time zone difference. Some of the speakers agreed to our invitation and delivered the keynote address very late in the night or too early in the morning. A very warm thanks or sincere thanks from our side to you, sir. I also thank the speakers and students from across the globe. A very sincere thanks from our side to you too, sir. I also thank the invited speakers and oral presenters who submitted or presented their various works on various aspects. A sincere thank to you, sir. I also thank chairperson and co-chairpersons of all the sessions who readily accepted our invitation and made good observations and recommendations too. Thank you, sir. And also, I would like to thank the rapporteurs of all the sessions who were very quick in sending us the reports. Thank you. Now, most important people in our international conference were the participants, both the teaching faculty and the students from across the globe who all registered and participated in the conference. A sincere thanks from our side. Next, coming to our college, our dean, beloved dean, Dr. R.C. Jagdish sir, has given us or has helped us in making all the logistic arrangements. I sincerely thank Dr. R.C. Jagdish sir. I also thank the teaching and non-teaching staff of our college, students of our department for helping us in organizing this international e-conference. I also thank Praveen from Digipix Technologies, Prashant from Cisco Web for giving us all the technical support from the day one to today. And of course, I should thank the steering committee of the university for helping us. Now, coming to the most important person during this conference, Dr. Devappa, our organizing secretary and our team leader, who has always been a backbone to the conference, who has taken all our ideas and suggestions very positively and has helped develop this international e-conference, which has been appreciated by many people throughout the globe. All the credit goes to him. We are very fortunate to have him as our team leader because whatever suggestions we have been giving, He has positively accepted all the suggestions and implemented them. Thank you very much, sir. We are happy to be a part of this team. Next, of course, I thank Dr. Shankrapa, who has been immensely involved in passing on this information for all the people throughout the globe and contacting each and every person personally and informing them. So it is only through his calls the message has reached throughout the globe. Thank you, Shankrapa. Next, I like to thank Dr. Rajeshwari, who has put in a lot of efforts day and night, starting from the preparation of the website to the preparation of the registration forms and helping each and every participant in the process of registration. We have received calls in the midnights. You can't imagine how we have worked. So a very big applaud to Rajeshwari for helping us in this process. Of course, now I would also like to thank myself. That is a different issue. Now, a special thanks to our family who has supported us. You don't believe we were on con call at midnight and they never questioned us. They have been very supportive. And we four of us know that we have worked day and night from past 2 months. And though it's not right at this on my part to say, but nevertheless, this pandemic has helped us to come together and exchange our views and ideas from across the globe. Hope we all join together to achieve sustainability in agriculture and So now, at this last moment, I request all the participants to please switch on your videos. A sincere request to all the participants to switch on your videos. Thank you. Yeah, thank you very much. Yeah. Now I request all of you to do one thing with me. Are you able to hear me?
Yes.
Are you able to see me?
Yes, yes.
Yes, but because I'm not able to see anyone. Okay. Kindly clap for a few seconds. Yes, thank you. This is for each one of us. A big kudos to everyone who has put in effort for making this international e-conference happen. Not only as it is you who have also made this conference a grand success. Thank you one and all. Again, we hope to see you someday in the offline conference. Thank you.
Devappa sir.
Yes,
Devappa sir.
thank you.
Devappa sir.
Yes, yes. Hello?
Yes, sir, we can hear you.
That regarding, hello?
Yes.
Special thanks.
Any participant
Hello?
share your views or give any further suggestions, we are ready to accept it. Yes, Shankrapa, I have informed the participants.
One announcement, please. The soft proceedings will be circulated within the e-certificates will be given as early as possible, whereas the hard copy of the certificate will be sent to all the registered participants. Except e-certificates, the award-winning certificate hard copy provided within a week. The soft copy will be shared to all the registered within 3 to 4 days.
Okay, sir. Done. Any delegates wants to express anything can do it.
Thank you. Thank you very
sir.
much.
Thank you.
I was going to say, actually, from maybe on behalf of all the overseas speakers, that this was a fantastic conference. And I'm putting together, 1,700 people. I was telling Shankarappa the other day that it's like almost like organizing a congress. It should have been probably called Congress of Plant Disease Biology and Molecular Biology or something. So it's all the organizers, Professor Devappa, Dr. Shankarappa, Dr. Sangeeta, and Dr. Rajeshwari, you've done really tremendous job. And bringing together so many people and for so little fees, especially. Someone was, I think one of the chairs was making a remark that for 100 rupees, you are able to see the research that's done around the world, on plant diseases. So really, really great job. Really appreciate all your effort, despite all the difficulties. We learned a lot. I'm sure many people have learned a lot. And they made many contacts through these conferences, which is going to help move forward this thing. So on behalf of keynote speakers and participants, I would really like to thank all of you very much.
Thank you, Marathi. It was nice seeing you after so
Seeing
many
you
years.
too. Yeah, nice seeing all of you. I will let others have a chance to say.
Yeah. So this is an opportunity to see our friends and seniors.
Definitely.
Thank you.
Yeah.
Yes,
4 days
special
is
thanks
with
to Dr.
us.
Marathi. Yes. Kulkarni sir wants to say something.
No. I had just unmuted. Congrats and thanks. It was a very successful program from UHS Bagal. And I thank Dr. Devappa, Shankarappa, yourself, and Rajeshwari, as well as the persons behind Dr. Arshi Jagdish and our Honorable Vice Chancellor. I thank one and all, and also the participants. It's the advancement of plant pathology all over the world.
Thank you, sir. Thank you, sir. Thank you, sir.
Let me thank Devappa sir for organizing this wonderful conference. In fact, there was a overwhelming response. I could learn many things. Though I am not from the pathology, but still I could learn many things. So this is helping us in all aspects. And moreover, it is reconnecting our own people. Many of my seniors who are working in this area, I could able to connect with them. So thank you, Devappa sir, for bringing all the people together. Shankarappa sir, Rajeshwari madam, on the Dean College of Horticulture, Bengaluru. Wonderful. Nice to see you. Thank you.
Thank you, sir.
உன் பேரு கிராதன்.
Small suggestion. Take stage again.
Yes.
Hello, sir. We are not able to meet, but at least the local team should go out and celebrate and have a party together today.
உடனே சீன் போடாத சரியா?
Other participants, please mute. Yeah. Thank you. Thank you for the suggestion.
You definitely deserve it after working so hard for the last two months. So you should go out and relax for one evening and together and enjoy the success. So at a safe distance, of course.
Yes. We're waiting for this.
Software.
Devappa sir, shall we close?
Please, you can.
Yeah. Thank you. I think the host can leave.
Thank you one and all. Thank you.
Okay, bye-bye. Bye.
Bye. Thank you. Thank you,
Have,
sir.
have a good day. Bye-bye.
Bye, Martha.
Thank you and congratulations, madam.
Thank you and congratulations.
Thank
it is
you,
very
madam.
well organized conference.
And
Thank
Rajeshwari madam also called personally persons, each and every person she called over phone and asked us information. Really,
Yes.
madams are also very active and really they have done their job very well. Thank you, sir.
Teamwork actually, I would.
Teamwork. Yeah.
Very good teamwork.
Good team. Deva Prasad team is wonderful, sir.
Okay, thank you.
Thank you, ma'am.
Needs to give a party now, madam, for all the teams.
We are expecting
Thank you, sir.
Deva Prasad.
The party and
Sir, we already, sir, already planned party,
yes
sir.
itself.
Already confirmed. That's good. That's the sign of good leader.
Yes, he is a true leader, sir.
Exactly. Just make the team work, but also, look after them. Yeah.
We'll meet. Definitely, we'll host party at the US only for you.
Very good. Very good. Very good, sir. I think you set such a high standard. I think from now on, they will look forward to such conferences in future year also.
For your kind information, 2023 we'll organize offline program without fail.
Good. That's very good. You already have plans. Fantastic.
Yes, 100%.
Bring all the people to GKVK. Yeah.
GKVK only.
By that time we'll
exactly.
I think this is the first international e-conference organized from our university for 4 days. Yes, sir. Deva sir, you agree?
Yes, you are right.
Real advancement of pathology. Really, it's a real advancement.
Not only by your university, I think in India also, as far as I know, ICR
Thank you,
and
madam.
SAUs put together, like yours is the first international conference attended by huge number of participants, and also you brought together the international speakers.
Yes. Thank you.
All credit goes to my team. All 4 people, not by me. I am only the director, that's all. Entire credit goes to 3 people.
Good leader.
He's a very good leader, ma'am.
Everybody can clap.
Very good leader.
Yes. Thank you. Thank you very much. From YSR University, yeah, there was lot of support from YSR University and
We are all YSR, sir.
including
YSR.
Delhi people supported me a lot. It will be there.
Yes.
Okay. Nice.
Very good.
Dr.
Yeah.
Sheshakiran should organize next thing.
Sheshakiran, definitely will organize. We will support.
We will support. He
But
should
he has not
organize.
supported.
I think the students can leave now. Students can leave.
No, he is the best organizer. But for our horticulture university, he has never supported. Sheshakiran is there? Yes, he is there.
Circulated to all the zone members, sir.
Sorry to say that Sheshakiran has not supported for our conference. He is having that capacity, but from his side, nobody has attended. I'm telling Sheshakiran has said.
Sir, I have circulated all your copy, and even you have posted in your Plant Pathology India, and I have posted in IRA Pathology Alumni, and also IPS journal members. I have circulated that copy. That much efforts I have put from my side.
Sir, Sheshakiran, you have put your effort in IPS membership. I know the capacity of Sheshakiran, but you have not supported. I'm directly telling you.
See, without support, you got more than 2,000 participants, Dr. Devappa.
Yes. Sheshakiran is having lot of talent. He is having that capacity, but He is working in the horticulture university, but he is near to me.
ಆಮೇಲೆ ಅವರು
I'm on. Okay. However, definitely we will support for offline conference in 20. Whatever Shashikar.
Sir, we all will meet again for the annual IPS meeting again. We'll all cooperate and we'll organize in much more grand way.
We will meet since 1996. I never missed the IPS program. I will be there everywhere, but I have not participated actively in this program. Really, I am unhappy.
ಬೆಳಗ್ಗೆ ಗಾಡಿ ಕಳ್ಸಲ್ಲ ಸರ್ ಅದು
next time you will do it, I'm sure.
Particularly YSR University, only very few representations.
Sir, we just have 15 members for pathology. Sir, not like your horticulture industry.
It's not there. Who is presented?
Sir, our university has very few pathologists, sir. Sir, that recruitment is pending. If that recruitment is there means we'll have more number of pathologists.
Politics of YSR University is required. Thank you. Definitely you will support in the 2023 international conference, offline conference.
Not only for conference, sir, for everything we will support. I don't know why you are disappointed. You are my good friend and senior for the past 20 years.
Friendship. Okay. Good.
Okay.
ಅದೇ ಅವ್ರು ಹೆಂಗ್ ಮಾಡ್ತಾರೆ ಹಂಗ್ ಕೊಡ್ಲಿ ಆಮೇಲೆ ಏನ್ ಕೊಡೋದಕ್ಕೈತಿ ಏನಿಲ್ಲ ನೋಡಿ
Dr. Silyachalam.
Students can leave. Students can leave. Students can leave the meeting. Yeah, Maruti is still there.
Dr.
Students can leave the meeting.
Yeah.
Only students. I am requesting students to leave, sir.
Yeah.
Maruti is there.
Did you travel anywhere after we met in Korea?
Doctor, this is Ranganath. Hello.
Yes, our Ranganath.
Yes, I went to see my son in Perth, Australia. That is a personal visit.
Okay. Dr. Ranganath, nice to see you. How are you doing?
Please come. Maruti.
My video is on.
Yeah, Ranganath.
Okay. Yeah. So
yes.
Congratulations, sir. Really, it's a good show. Sorry, I could not able to attend your last because of some other overlapping meeting. So just I joined maybe 5 minutes back. I joined. Hello.
Yes.
So it really, really, it is a very good conference and well organized. I think a lot of hard work you and your team has taken. Really appreciable and new experiment. That is a really good thing. And you have saved so many people's money, time. That's a good thing. Even those who are interested will concentrate. Anyway, the slides are coming on our laptops. That is a really good platform to learn also. So it is a really good experiment, new experiment. It was went very well. Although it will happen, especially with this small discorrection, all those things, it will happen. But it is really good. I'm really happy you handled so many people's. And I congratulate you and your team. Great, really great.
Thank you, Bharat. Thank you.
You should consider putting it in the media, maybe some newspaper or some interviews like that.
Yeah.
Right. Articles.
No, before that, I'll leave actually one suggestion, maybe my proposal. Doctor, actually, you have so much organizing capacity and caliber networking things. Maybe my suggestion is maybe you should take one burning problem. maybe whatever is the disease or something related to Karnataka or even entire India, the burning problem, select some few very good people who are working in the same field and organize the meeting. Let it be 25 people, 30 people, that is not important. Have some
Yes.
burning issues or general issues like resistance management. I think Dr. Krishna Kumar can definitely support in this regard. He always tells me, you organize the meeting, I will bring the resourceful persons. So I feel like you are by this you endorse that you are capable of doing much more things actually, and I wish you all the best. And again, we want to see you in many bigger positions.
Definitely,
Sir,
we will
Venkat sir,
Hello.
Sir, your voice
Hello.
is breaking, sir.
Hello. Hello. Hello.
Yes, sir. Therefore, sir, we can hear you.
No, mine is over. I think yeah.
Rangnath, definitely we will take your suggestion. Within short period, we will organize such kind of programs with the help of Dr. Krishna Kumar.
Yeah. Even that day night, actually, Dr. Bidgi sir also called me. Actually, he was telling, no, we should do something differently. We should have some. He was also same opinion. You should take some burning problems which should be useful for the that region or a state like that. Even he was Dr. Bidgi sir was suggested a very good thing was make use of your scientist who are extensive work on that particular projects or research area, and take some industry people also who are working in that line and take some few farmers also. So then it will come out with a very good. He suggested like that. That's a good proposal actually. Even I agreed, sir. If anything is there, I am the first person to join. Actually, I told him. Even Krishna Kumar also was telling so many times we should organize. And yes, I think when Dr. Krishna Kumar sir was in IHR, he has organized some whitefly management, millibug management, I think thrips management, like that. Only brainstorming sessions, morning to evening. At the end of the evening, we should come out with some clear action plans, like that.
Definitely, we will organize such kind of programs in future. Rangnath.
Right. Yeah. Thank you, sir. Thank you.
You can ask next year what is the status of that program.
Yes. No, I understand there are difficulties in your system, but
my team is very strong.
No, I understand. You have proved it. You are sure. No need to. I endorsing. Your team is strong because all this success because of the team.
Mostly network issues. ಇಲ್ಲ ಬೆಂಗಳೂರಲ್ಲಿ ಪ್ರಾಬ್ಲಮ್ ಆಗ್ತಾ ಇದೆ.
No. For me, I don't have any issues. Hello.
ಕೇಳಿಸ್ತಾ ಇದೆ.
Yeah. So, doctor, when you are coming back to India?
Are you asking permanently or just temporarily?
No, both are good actually. Both are okay for me.
Yeah. No, like we all have tied to our jobs. I am kind of tied up with it, so it's difficult to come back there permanently. At least not in the short term. But actually, November, December ಅಲ್ಲಿ ಒಂದು ಮ್ಯಾರೇಜ್ ಇದಿದೆ ನಮ್ಮ ಫ್ಯಾಮಿಲಿಲಿ. So I am looking for tickets to come to India.
Okay.
Yeah. Next month itself. Yeah. I need to find a way. It's not easy. Ella tickets very few flights ಇದೆಯಲ್ಲ.
Okay.
Yeah.
Welcome to our department.
Yeah, definitely, sir. I will definitely pay a visit if I come there. So I never missed visiting GKVK. I'm in India.
ಬಂದಾಗ ಬನ್ನಿ, ನೀವು ಕೂಡ ಬನ್ನಿ. ಬೆಂಗಳೂರಲ್ಲಿ ಇರ್ತೀರಿ, ಈ ಕಡೆ ಡಿಪಾರ್ಟ್ಮೆಂಟ್ ಕಡೆ ಬರೋದಿಲ್ಲ.
No. Yesterday, in fact, I was in GKVK when Ravichandra was presenting. I was with him only. So actually, as per your suggestion, he is prepared that introduction of nematodes. So, hello.
How is Rekha doing?
ಆರಾಮಾಗಿದ್ದಾರೆ ಸಂಗೀತ. Yeah. She's at work today. ನಾನು ಮನೆಯಲ್ಲೇ ಇದ್ದೀನಿ, working from home. So, yeah. be a big relief hangare ನಾಳೆಯಿಂದ.
ಹೇಳಿ ರಂಗ.
Okay, thank you, sir. You can continue. Thank you, Dr. Maruti, Dr. Devpa, and your team. Thank you very much. Thank you, sir.
Nice to see you, sir. Nice to see you. Really, yeah,
Bye-bye.
very good. Yeah. Okay. Okay, sir. I'll take your leave. Yeah, all of you. So I think you need rest now. So go and
Rangnath.
relax a bit.
Thank you for your suggestion.
No problem. Yeah. Okay. Have a good day. Bye bye.
I'm telling you, thank you very much for your kind support.
No problem. No problem. It was very good. Very good. Bye. Bye. Bye, Shankrapa. Bye, Rajeshwari. Bye, Sangeeta. Yeah.